was done between him or among the wife, the children, and him. All right, so this is his DNA. So the first thing you need to do, when there is three people which are involved, the DNA of the mother and the DNA of the husband and the DNA of the kids. Yes, if the kid is the son of this parent of one of these people, basically all the bars, yes, their profile must be matching with this. If here I have 25%, the remaining 75% must match, meaning that there must be no bar which is left out to confirm that that is the parent. Therefore, let's go. Give a scientific reason why lawyer insisted on DNA profile from the woman and their children to determine the parent. Why? Because DNA is unique. Yes, there is no way how you're gonna manipulate it is unique so that you identify the actual parent of these children, or to identify the actual children of this gentleman. You understand? Yes. They're saying that which child will inherit this is wealth. So you have to fight identify. Okay, now let's go. Start with this. This doesn't match. That is just as it doesn't match with the mother and the father, then that is out. Okay, we go to this, out. This matches with the mother. So this matches with the mother. Nothing matches the mother. Yes. So this one is out. This is completely out. What about this? This doesn't match with the mother, but matches with the father. So this is first. This doesn't match with the father, but matches with the mother. Okay. This matches with the mother, but not the father. Correct. This matches with the father. You see? Does it? Yes. It matches with the father. Correct. This matches with the mother. Correct. This matches. Are you sure? Yes, with the father. Yes, correct. This matches with the mother. Correct. This matches with the mother. Correct. This matches with what? With the father. You see that all of them have been utilized. If there is one which is out, that's not your father at all. Then this one, we come here. This matches the mother. Matches the mother. Nothing. It's out. The moment it's out, forget about the rest. You see? So basically, this child, wise child, is going to inherit. Actually, is the son. Yes. Explain your answer using the evidence above in the diagram above. Why is this one a child? Is because all the bands of the child match, all the bands the bands of the child, yes, some match with the mother, the woman, and the remaining one match with the, you see, those which match with the mother is fine. The remaining one must match with the father, or some match with the father and the remaining one will match with the mother, meaning that this is the mother and this is the father. Yes. Oh, if you calculate, if you find it's 50% matching with the mother, correct, and 50% they must match with the father to make it 100. Name one simple source that could have been used to get DNA. It could be blood, it could be hair, it could be saliva, it could be skin, it could be anything which is easy without affecting the mother or the father. Just in case electricity goes, maybe we can be stagnant for some few minutes so that we switch to the system. Anyway, let's continue. They're saying that what is A? This is the chromosome. I told you that when you get this, you have to first, and then this is the spindle fiber. Yes. Then they're saying name the process that is represented. Look, this is single single chromosome, meaning that is must be meiosis 2. So because it is in the middle, therefore is metaphase 2. Metaphase 2. They say name the chromosome that were represented. How number of chromosomes presented in the parent cell, because there are 2 here, so there were 4. You see, one daughter cell at the end, there are 2. And they are saying give the name of the structure. A, we said is chromosome. And then give the function of structure B, to hold the chromosome in what? In a position, yeah? Yes, you see. And also they are used to pull the chromosomes to the opposite pole. Yes, they contract and then they pull the chromosome to opposite poles. Yes. Describe the process of crossing over. I think people okay, anyway, let me not talk about it. Crossing over, basically you look at chromosomes of homologous chromosomes. The homologous chromosomes, they align together. Chromatids of homologous chromosome, they cross over at a point called chiasma, you see. And even they didn't talk about the phase, so you mentioning the phase is also another mark. The genetic material is exchanged, and then after that they split, and then they will have genetically different genes. Yes. So chromosomes of which are homologous, they align together. Chromatids of homologous chromosome, they cross over at a point called chiasma, and then after that what happens? They exchange their genetic material, and then they separate or they split to form 2 genetically different chromosomes. This process occurs in prophase 1. Yes, you can also talk about that, so that maybe if the mark is there. All right, they're saying that draw the phase, the diagram of the phase that occurs after the phase. So here they are looking at anaphase, but when you're drawing, you make sure that, let me show you, make sure that when you're drawing, is actually supposed to be like this. Let me show you. It's supposed to be like this, ne? I don't know whether you can see. It's supposed to be like that. You see, it's gonna split. When it splits, so make sure that these variations are correct. Yeah, so make sure that the variations are correct. So this one is gonna move this side, and then this one is gonna, so meaning this and this is gonna be black. When you are shading it, you must shade it black, you see. And then this one and this is gonna be it's gonna have a black thing there, and then this is gonna have, you see, you're drawing what will happen. It's a black and a white, black and a white, and then this side is gonna be what? A white, based on what it's shown there. And then spindles, and then you write the phase there, the title of the what? Of the diagram. So meaning that you're right, and then you say that because the question they're saying that the next phase, the diagram of a cell and what did they say is the question? Give a functional, okay, describe the process we done, draw a phase, okay, in there. Yes, diagram of a cell showing anaphase 2. Yes, so basically that's what you're supposed to say, caption, and then you draw these chromosomes I've shown you with, you draw them nicely, ne? The way I've tried to show you, and the variation, because they might be marking the variation, but what I want to show you is that they are supposed to be 2 chromatids on each side and then separated, and then the caption, so that when they mark you, they give you correct answers, they give you ticks. Let's go to question 2.4. They're saying that familial this syndrome, whatever, is inherited condition caused by the dominant allele. People with this condition have a high level of cholesterol, fats too much in their blood, and then this increases the risk of their blockage, meaning that you are likely to suffer from stroke. The diagram below shows inheritance of the parents of several. Something is important here. Generation, okay, this is caused by a dominant allele. Look, the person with the, because here they are saying that caused by a dominant allele. So that's a point. as I was explaining yesterday, that this dominant allele, you won't miss it because they know that these days you fail it. So in that case, you come and then you explain. Let's read and find out. Let's read this question. Something is not right here. Is it me who is colorblind? I don't know. Okay, we shall find out. How many generation? You know why I'm saying is because I'm seeing a key here. They're almost the same thing. But if I'm not forgotten, I've not forgotten this was shaded. This was also shaded. It's supposed to be shaded. One shaded. But these ones, some of them are supposed to be shaded. Some of them are not supposed to be shaded. But I remember this was shaded. I think even one of it was shaded here. But it's okay. You'll check on your paper and then find out. Ne? How many generation in this? We count the generation. 1, 2, 3, 4. So 4 generation. Someone is gonna say, no, not this one you don't count. Why not counting the parents? Are they in the same generation with you? Are you in the same generation with your parent? They will say that our generation need to be like this. Okay. Determine the number of males in the diagram above who are homozygous recessive. So in this case, I'm not going to give the answer for this for now, not because I don't know. Yeah, it's because they shaded. I would have been with the original copy, ne? And I don't have original copy, ne? Just use the photocopied one. Okay, I would so count the box. You're supposed to count. Let me just give you a clue. You're supposed to count the box. Yes, the box who are shaded. No. Just a moment. Let me just go get just 2 minutes. 1 minute. Get the original paper. Yeah, I'm back. I've got the paper, the original paper. Let me just find out. Let me just shade it for you so that we are on the same. Sorry, I'm not having a memorandum here. Just creating the memorandum as I'm discussing here with you. Yes. Yeah. So it's like I'm also writing the paper. I'm also writing the paper. All right. Okay, yeah, now I've got it. Yeah, this one is shaded. This one is shaded. Yes. This one is shaded. Yes. This one is shaded. Yes. Cool. J is shaded. Yes. Shaded. N is shaded. O is shaded. P is shaded. Yeah, if I don't shade all of them, then you'll find out. Ne? Yeah, but let me just show you. All right, they're saying that the shaded ones are the one who are with the problem, and then the unshaded are the one without the problem, meaning that the shaded ones, they are supposed to be either capital T, or capital T, small t. Those are the shaded ones. The unshaded ones are supposed to be, don't forget that this is caused by a dominant allele. If it was caused by a recessive allele, then this would have been the shaded and this is unshaded. But because it's caused by a dominant allele, the shaded ones are the one which have the what? Yes. So now let's go through this. The question is saying that determine the number of males. They are looking for males. Oh yeah, it's the males. So the males are the one with the box. They're saying the males with the problem. So it's 1, it's 2, it's 3, it's 4, it's 5. Based on what I've shaded, yeah, it's 5. 1, 2, 3, 4, 5. Yes, the answer is supposed to be 5. Determine the number of males in the diagram above who are homozygous. Okay, we are done with AA. The answer, this answer is 4. Just down here, it's 5. Here I said how many generations? There are 4. Saying, hey, G and are planning to have the 4th child. What is the cross to show? Use the genetic cross to show the percentage probability of child without, meaning that it's supposed to be homozygous recessive. So you guys remember they're saying that they are planning to have another child. Yes. So okay, I'm gonna say P2 F2 phenotype. So you're saying that G is a male who is shedded, who is having the disease. Yes, you put it there, cross with a female which is H without a disease. You also put it there, and then here you say gamete. Sorry, genotype. So if it's a genotype, because this one is having a disease, but this person came from top here. Where is the G? Oh, G is here. It came from top here, of which this one was also sick, meaning that this one is heterozygous. You understand? Yes, is heterozygous for the what? For the condition. Yes. So why? Because this one is capital R, small r. Sorry, capital T, small t. This is small t, small t. Definitely this one to be having a disease must have a capital T. There is no way how it's going to be capital T, because this one was already what? Normal. Therefore now the genotype is going to be capital T, small t, and then GH, H is normal. Remember, is a recessive. Yes, I think now gametes will form as a result of meiosis. So meiosis is going to take place. You form the gametes. It's going to be capital T, small t, small t, small t. Then now fertilization is going to take place. This with that, capital T, small t. This is that, capital T, small t. This with this, small t, small t. And then this with that, small t, small t. So since small t, these are offsprings. You see, F2, it must be in the same line. We come here, we give you a mark. Meiosis and fertilization, we give you a mark. The genotype and phenotype from the question still, we give you a mark. Then we give you a mark here. But there must be a compulsory mark here. A compulsory mark here, the question is saying that use the probability. So in this case, the probability is going to be a compulsory mark. So it's going to be 2 of them, they are normal, and 2 of them, they have the condition. So it's going to be out of 4 times 100, because they are looking for the probability. Yes, the probability, or the percentage probability, which is equal to 25%. So that is the answer for that case. So yeah, let me put it back. I think I'm done with that question. Let's go to question 2.5. These papers were too much reading. Anyway, lactose is the sugar in milk. Lactose is found in milk. It's digested by the enzyme called lactase. Then some people cannot produce lactose, and therefore they become lactose intolerant. And then the condition is caused by recessive allele. This means that the people with the homozygous recessive, they become lactose intolerant. People from different countries, okay, were tested to determine if the lactose intolerant, they were lactose intolerant. They are saying that the table below shows the number of people tested in each country and the lactose intolerance percentages. Okay, let's find out. The population. Here you're having the percentage and then tested people. The population size of country B. Where is country B? Okay, is 190 million. They're saying that calculate the number of people in this country likely to be lactose intolerant. Here they're saying the percentage of people who are lactose intolerant. So here these are the number of people they tested, and this is the percentage of the people, you understand, who are lactose intolerant. So meaning that in this case, because this is the percentage, so I'm gonna say 12 divided by 100. That's the meaning of percentage times the 190 million people. So when you get the answer, I think it's around 22.8 million people, something like that. You can calculate and then find out the actual answer. But that's how you're supposed to do the calculation. There is another method, but let's just go with one as long as you get the correct answer. Which country is most likely to be having the least reliability? Check here. Remember when I was discussing yesterday, I said that people, you need to know what is it. So the answer is E. Why? Because we use the same fewer number of people. Remember, if you want to increase the reliability, you increase the sample size, the number of people who are in this investigation. You understand? Yes. So we go to the next question. Yes, we go to the next question. Just a moment. Yes, let's go to the next question. Give a reason for your answer. We said that because you have used this fewer number of people. Provide 2 ways in which the validity of this experiment can be improved. Validity is you are keeping things constant. Yes, use the same number, maybe the age. Remember, age can also affect the people. Same kind of individuals. Yes, you are busy increasing the what? This validity. Keep the same type of the food you are giving them, because they might have different concentrations. Yes, meaning that the concentration of food or the type of food you are giving these people, which has the milk, sugar. Yes. Then there are so many. Describe the planning there a lot, because we are using different countries. It depends on where. If you are going to fly to different countries, then therefore you have to plan in advance, maybe requiring permissions or visa things, a budget. Look at the budget. Ask for permissions for these people. Are we going to be experimented about? You have to look at the suitable time for these individuals. So there is a lot of planning steps you can consider. Then let's go to question number 3. Yes, question number 3. Yeah, we are almost done. That's question number 3. Okay, it's too much of talking than writing. Flies lay eggs in urine and fish that stuck on the wall of the what? Of the sheep. The eggs hatch and then larvae damage the sheep's skin, leading to the infections. To reduce this problem, farmers in New Zealand have been developing sheep with no wool growing on their legs. Then they're saying that the back side, that's the bottom, or the under side of their abdomen. Farmers have crossed different breeds of sheep to produce offsprings with desirable characteristics. And they are saying that the characteristic of other breeds of sheep are shown in the table. Okay, they're saying that use the evidence from the table to describe how the farmer could use selective breeding to develop. So if you look at our distinction material, it shows you what exactly you're supposed to do when you are dealing with the selective breeding. Yes, it's here. Sorry. Yeah. Some there is basically what you're supposed to do. In this case, the first thing you're gonna do is to select the individual who have what you want. So basically, you want these legs. You want bare legs, ne? Did they say bare legs here? Yes, the farmer crossed breeds, okay, of springs with desirable characteristics, and then with other sheep, okay. They're saying that produce. Yes, he said it here that use the evidence to describe how farmer could use selective breeding to develop the sheep with bare legs. You see, bare legs, and then backside, bare backside, not abdomen. They're looking for backside. So meaning that this individual is gonna cross this because this has but they doesn't have the bare backside. Therefore, it's gonna cross the one which has bare legs and then the backside, and then it will deny sex to this. So meaning that this one will not be given sex. Only this one will be interbred, and then yes, you keep interbreeding. This is what I'm trying to say. So what happens is this is what you can say. For example, to increase the quality of is here. Suppose you want a variety of cow to produce a lot of milk. Now, in this case, suppose you want to have this sheep with these characteristics they are looking at. You see now you are writing. Choose the cow in the herd to produce that can produce most milk. Choose the sheep that is this, the one which can have bare legs, and also so these two, because they asked you how did use the evidence describe how the farmer could use. So we did select this and this because they have these desirable characteristics. So after that, then allow these offsprings. Let only allow these cows to reproduce. These ones which have the desirable characteristics, these sheep, you allow them to interbreed. That's why I'm saying that you are denying this one sex. And then after that, what happens? The one when they get the offsprings, yes, the offsprings that produce these desirable characteristics, the one who has bare legs, and then this, you cross them again. You see, you cross the offsprings that can produce this characteristic. You let this offspring reproduce again. You understand? So at the end of the day, you have animals with this and this together. One animal with those two characteristics. That's how you're supposed to explain. Then give two tabulate. Here you're supposed to draw a table. I told you guys, you draw a table, complete table. You don't draw a T table. This is wrong. You get a 0. So don't cry when you find a 0 there. So tabulate the difference between selective breeding and natural selection. Selective breeding, basically, this one occurs naturally. So the nature will select. Here it is human needs. Yes, then basically it's like artificial selection versus natural selection. Environment will do the selecting. Human will, is the selective force. You understand? Yes. It occurs within a specific species. This one can occur in multiple species. Yes. Because you are just crossing. Yes. So you only select what is best, what you think it is good for you. So basically this is the differences you're supposed to write about. Explain one disadvantage of selecting breeding of sheep in this question above. Basically, when you do the selective breeding, this sheep might lose important characteristic which might help it to be able to survive in the wild. You see? Yes. Maybe the hair, and then you don't want this hair, and the hair is there to protect this sheep so that it can sustain in different temperature conditions. And then when now you just want to wipe it out because it is doing the caca on the wool, because you don't want it. So at the end of the day, that character might get extinct. So that's whenever you're doing selective breeding, you are not leaving the nature to select against those who are not best suit. Then you are doing some disadvantages to the animals. Yes. Then when we go to question 3.2, the diagram below shows the modern swordfish. The ancestor of had short sword that yes, the modern swordfish have long sword. Yes, the modern swordfish have long swords. Yes, the swordfish use their swords to kill the prey, meaning that they're using it to kill the prey too much. The injured prey is easier to eat, you see, is easier to eat and is easier to catch and eat. Something is important here. Okay, they're saying that the diagram below shows one theory of how the length of the sword it got evolved. Here is many generation saying the sword grew longer each time the swordfish uses its sword more and more. Oh, the more you use the structure, the more it becomes bigger and stronger. So it means that that's Lamarck's theory. Oh, they're asking a question. Suggest which scientist suggested a theory above. That's Lamarck. Explain one reason why this theory is rejected by most of the scientists. Because organisms don't evolve because they want to evolve. This sword did not evolve because the fish wanted it to evolve. And then there is not enough evidence to show that this acquired characteristic during lifetime is passed to the next generation. You understand? Yes. Describe how natural selection could result in modern sword. So here you're using Darwin's theory. So what are you going to write? So here you talk about the variation, that there is variation among the swordfish. Then you tell us the variation. Some swordfish had long snout. How did they call it? They call it sword. The sword, yes. Some swordfish had long swords and some swordfish had short sword. Yes. Those with the long swords, here they said it. They say that they could. Yes, check here. Those with the long sword, yes. What happened here? The one with the long sword, what happened? Yes, they are able to kill the prey and catch it and also eat it. Therefore, they survived in large numbers. While those with the short sword, they couldn't get or catch the prey and eat. Therefore, they died in large numbers. Those with the long sword, they passed their genes. This is called natural selection. Those with the long swords, they passed their genes to the next generation of the long sword. After many generations, all swordfish had long sword. See? Yes. So when you're explaining it, you're supposed to link it to this example. Yes. Then the next question they're saying that describe. Yes. So we are done with that. So variation. Some swordfish had short sword and then long sword, and then those with long sword could catch the prey and kill it, and then they survived in large numbers. While those on a short sword, they couldn't, and then they died in large numbers. Those with the long sword, they passed their genes. This is called natural selection. Those with the long swords, they pass their genes to the next generation. Yes, after many generations, all the swordfish had long sword. So that's what you're supposed to explain in that regard. Then when we go to 3.3, 3.3, they're saying that the table below shows the average cranial capacity in hominids fossils. Okay. State one Australopithecus africana species. you have learned about and give. Yes. So what are you going to give? And give the common name, and then so basically you just supposed to choose maybe Australopithecus sediba, that's Carabo, who discovered it. So all those things, they're saying Australopithecus africanus. They're saying did they say Africanus? It is wide. Afarensis. So it's only Adipithecus. So Australopithecus Lucy Johansen. Then Australopithecus africanus, you can says place Robert Broom. Yeah, Littlefoot, Clark. And then Australopithecus sediba, Carabo, Idebega. Then you all this you can choose whatever you want to choose, and then just tell us Australopithecus. Then what is it? And then you tell us who discovered it. So you choose any of these Australopithecus from here up to there. So you choose any. Yes. And I was emphasizing how do you memorize that. Step 2, advantage of having a large cranium. Large cranium is going to bring about more brain cells. And then these more brain cells are going to link, is going to bring about intelligence. And then if you have more intelligence, meaning that you can think more, you can reason more, you can have more skills. All those are some of the advantages of having a large cranium over a small cranium. Then that's what you're supposed to write. Then you're saying draw a bar graph. Draw a bar graph to compare the average cranial capacity of Homo. Guys, this question was a little bit not supposed to be rushed on. You know why? Let me show you. They're saying Homo species. Some people are busy touching on their head. Oh, I drew everything. That's why I say that don't rush. Don't finish the paper in 1 hour and then you say, yeah, I'm done. You think that the examiner is a fool. No. Okay, they're saying Homo species. So it means that your bar graph was supposed to be only those individuals, because those are the Homo species. You see? So from there, after drawing that, then what are you supposed to do? You come here and then you tell us, you draw. You're supposed to draw the bar graph, but now what goes on x-axis, what goes on y-axis? Hominids, they go on the x-axis. So you tell us here the hominids, and then the y-axis, they're saying number of fossils, they're saying cranial capacity, because they're saying cranial capacity. So meaning that the cranial capacity is supposed to be here. Did you give the caption? Did you give what is here? You put the names, the spaces are correct, the scale is supposed to be the same, depends on the scale used. Yes, the small one is, the smallest is 640, the biggest is 1350. Maybe you can start from 0, 500,000, 1, something like that, or you can be 250. Yeah, 0, 250, 500 to 550, 600, 750, something like that. Yeah, so like that, 0, 250, then 500, 750, 1000, 1250, 1.5. So like that, it must have the same range, so that we come here, we say caption, we give a mark. We say correct type of the graph, we give a mark that is a bar graph. Yes, it doesn't matter. And then caption, you put the caption, you give a mark. The spaces are correct. It must be correct, same spaces, same size. And then scale, we see the scale on x and axis. The scale, we give a mark. Have you labeled it correctly? X and Y, you have labeled it correctly, give a mark. And then plotting, maybe we can give you 2 marks there, 1, 2, 3, 4, 5, 6. So maybe that's how we're gonna be marking it, so that now you get the 5 marks, all of it. Then they're saying that explain the genetic evidence to support the out of Africa hypothesis. I was explaining it even yesterday, and then I was giving an example with these kids, and then people. So this is what we are supposed to say, that inheritance of only inherited only from paternal line. Analysis of mutation of this mitochondrial DNA show that the oldest female, yes, the oldest female, yes, the oldest female, what happened to it? The oldest female ancestor. Just a moment. Thank you for, yes, I was explaining that the mitochondrial DNA. So here, if you look at this question, check how the question is saying that explaining how genetic evidence support the hypothesis that ancestor of Chinese Homo erectus fossil originated in Africa. So here you are showing that this mitochondrial genetic evidence, and we have mitochondrial DNA. It is inherited only from the what? The maternal line. You understand? And then the analysis of mutation of this mitochondrial DNA show that the oldest female ancestor with the were located in Africa, and all look, and all now look, and all these Chinese, and all these this came from what? From Africa. So basically, that's what they are looking for in that regard. Yes. So the next question is saying that study the phylogenetic tree. It was clear yesterday when I was trying to explain this. Study the phylogenetic tree showing the evolutionary relationship between the birds. These are the birds, the eaves and the crocodiles. Ne? Answer okay. The following questions. When did this branch, when did Eve or the birds or branch crocodile, when did they branch from each other? So it will depend on this will depend on, if you look at this, I don't know whether I see it properly. I don't have the original again. If this is 1 year, 2, 3, 4, 5, 6, from here to here is 1 year, 2 years, 3 years, 4 years, 5 years. Basically, they separated here. This is when they separated. If it is in terms of years, they separated in million years, meaning that it's 5 million years. But this is where they separated from. I don't know whether they forgot to put the numbers here, 0, 2, 4, or depends on what we're supposed to be here. So they will see how they're gonna mark it out. But it was supposed to have a mark there. Yes, it was supposed to have figures there. Let me check from the original paper. Then the original paper has nothing. Okay, it has nothing. So I think they will put a concession there. But if it is regarded as 1, then we would have said that then it would have been 0, 1, 2, 3, 4, 5, meaning that they separated 5 million years, meaning that is 5 million years ago. That's when they separated. But they're saying that give the term that describe the structure shown in the symbol of the forelimb that was inherited in common ancestor. Which structure? Oh, this one. Yes, forelimb, this one. How do we call this? How do you call this? We call it pentadactyl limb. Let me show you where I put it in the booklet here. The distinction material. Yes, it's here. We call it pentadactyl limbs. You see? It's here. Pentadactyl limb. This one. This is what they are looking for. So pentadactyl limb. And then they are saying that that is the answer for that question. Then the next question, the second last question, they are saying that too much reading. Okay, that imps and ostrich are both large flightlet birds. Imps are only found in Australia continent, and then ostrich are found in Africa. All right, tell us the story. We are listening. Okay, this is the imps, this is ostrich. The original hypothesis about the evolution of the imps and the ostrich was that they share a common ancestor. They share common flightlets. This is a very important thing. Share. Okay. The sea separated the ancestral population during this. So meaning that this is the force which separated the continents. The Australia and Africa, yes, more evidence has resulted in a new hypothesis that the ancestral population of imps and ostrich was highly flying small ancestral birds that could fly long distances and migrate from Central Asia to continents in the southern hemisphere. The new genetic analysis show that mutations in DNA cause modern birds to lose the ability to fly eventually. And then describe the question: how the high-flying small ancestral birds evolved into different species of large flightless birds, ostrich and imps, on the other continents. So how are you going to explain this? So basically here this is speciation. Yes, and this cardinal speciation is the allopatric speciation because there is a geographical barrier which is separating them. So what are you going to say here in the so you can say that at first there was one big mass with a common ancestor of flightless birds, because they said it the originals, they're saying that we're sharing a common flightless bird, and then separated into this by this, so meaning that at one time there was one large mass with a common ancestor with a common flightless ancestor. You understand? Yes. Or this flightless ancestor lived on one big mass at the beginning. And the mass was divided into Australia and Africa, whatever. Yes, due to continental drift. I see. Yes. And then these organisms which were in Africa, where exactly? Which organisms were in Africa and Asia, they could not interbreed because they experienced different environmental conditions, and because of the no gene flow, the natural selection occurred independently. Therefore, they became phenotypically and they became genotypically and phenotypically different. If you remove this water mass, they say the sea, they say it somewhere. Yes, yes. These birds cannot, yes, interbreed. When they mix, they cannot interbreed to produce a fertile offspring, therefore resulted into large flightless birds, ostrich and emus. Yes, so this must be written at the end, because this might be a compulsory mark, because this is what you are looking at. And also a common ancestor of flightless birds could be a compulsory mark, meaning that if this question is 7 marks, you find out that 5 marks is for free, meaning that you can score it anywhere. But you saying a common ancestor is a marking point which is compulsory, and also you saying that mentioning these birds, yes, because that's what we are looking for, might be a what? It might be a what? A compulsory mark. Yes. So if you're a teacher and you are here, please try to look at it on the memorandum and then find out where to put a mark, not just giving marks. Yes, so that's how you explain that. Then the last question, but not the least, is the last one. Identify. Here you have yesterday I was at one of school. I was in 3 schools, but there is another school where my colleague called me. It's called Wiseman. All right, yeah. As I was about to leave, a student asked me the same question about punctuated equilibrium. How do we differentiate between punctuated equilibrium and gradualism? I don't know why this always disturbs students when it comes to explain, but let me explain it. Here it is punctuated equilibrium. The diagram you see here is punctuated equilibrium. It has periods of rapid change and long periods of no change. Short period of rapid change and then no periods of change. You see, short period of rapid change from here to there, and no period from here. No periods of what? Change from here. You see, rapid change. Rapidly they change without seconds and seconds. You understand? And we say that this is caused by a mutation. Imagine from here, from this kind of rat to white, immediately in the morning you find yourself that. And then from this kind of rat to a dark one, from a gray to a dark one. We don't have, it's caused by a mutation. So that's number 1. Punctuated equilibrium is caused by a mutation. Number 2, it has no intermediate at all. Because how is this linked to this? Nothing. It's just rapid change. Then number 3, it takes place within a short period of time. Short period of time. If you compare it with gradualism, which moves slowly, you'll find out it's caused by natural selection. The nature must select, and the nature, it's slow. Select small. Then when you talk about it has intermediate. From here to there is an intermediate, you see them. And then it takes long period of time. So now, if you come here, identify the concept of evolution illustrated above. So here it is punctuated equilibrium. And then give the definition of the concept evolution as shown in the diagram above. Basically, this is evolution with a rapid period of change and long period of short period of rapid change and then long periods of no change. Yes. So this also is a type of speciation. It's trying to show you how speciation occurred. So guys, I think that was the paper. How was the paper? How was the paper? Yeah, I think the paper was not that bad, but it had too much of reading. Even paper 1 had too much of reading, and we know that our kids, they don't like to read too much. They are not straightforward like other provinces I've seen. Yes. So if I look at the analysis of these papers they have written in terms of life science, paper 1, it was not that difficult, but too much words. was too much words. Paper 2, the same story. It's not too much. You know, our students don't like to read. Ne? And I know you are there, you know that you don't like to read. Keep reading. It's not, you don't like it. Ne? So you guys, thank you for being here. Don't forget to get a distinction material book from the description below. Tell us what you need us to talk about. If you have any challenge, let us know so that we can help you out. And then also, don't forget to tell your teachers to help you out. We are always here to help you out, you guys, so that at least you have a better life. Yes, so that when I get to my retirement age, at least I know that I helped maybe 1 billion people. Yeah, 1 billion people. People maybe in South Africa, at least 50% of them, at least they pass through my hands or somewhere. I helped them a bit. At least I will be in my retirement with happiness. Anyway, I wish you the best in your finals. I'll be here still to help you. I've not stopped from helping you. I'll help you even if you have done writing your paper. Very much, guys, for those who have been supporting us. Thank you very much for those who tried to be on our channel, who shared our content. I really appreciate, or we appreciate for that. Natasha, please don't be scared. Everything has the beginning and has the end, and this is today's end for this life science. Let's start, guys, without wasting time. Are you ready? All right. All right, guys. Don't be scared, ne? All right, guys. Hey, question 1. Various options should be provided. I don't read these instructions because everyone knows that. The base pairing in DNA was discovered. Base pairing. Don't forget that. Here they are talking about that DNA is made up of complementary base pairs. Who discovered that? When I was teaching this, I tried to show you exactly what they said, and I talked about this, that they would just try to mix up these names. So basically, it is James Watson and Francis Crick. So number 1 is going to be the best answer. I'm not going to go to the distinction material all the time, unless there is a challenging question which talk about maybe you don't understand something, and then that's when I'm gonna talk about the distinction material. So this is what you're talking about: James Watson and Francis Crick. Yes. So basically, it is Crick and Watson. So number 1 is yes, the gene code for production of the gene. The gene code for the production of what? For the production of protein. DNA code of life. I was explaining that, the DNA code of life, that DNA code for life because it codes for proteins. So the answer, the best answer here is D. I hope you guys, you can hear me very well. No echo, no what. Everything is right. If it's not right, let me know. And then they're saying that which one of the following is a characteristic? Which one of the following is a characteristic of stem cells? We talked about stem cells yesterday. And then we say that stem cells have ability to change into other organs. So let's look at that point. They are easily obtained from any other organ, not any other from any organ. No. They're saying that because we saw the sources, division divided by meiosis. No. They are haploid. No. Then they're saying that they can be stimulated to form any other type of cell needed. Correct. So basically it is D. Then someone is saying that the chance of having female in a human, guys, the chance of having a female in a human is always the same. Boys, girls, the same thing, 50%. If you want to do the calculation, we can do the calculations, or you go the distinction material. So basically this one becomes B. You see, now I'm thinking about something and then I'm writing something. Okay, B. Which one of the following is a part of reason why color blindness is more common in males. We talked about this yesterday, and then we said that the allele for colorblind is recessive and located on X chromosome. Yes, don't forget we said that males have one X. If a recessive allele appears on it, it cannot be masked. So the individual is already sick or affected with that disorder. While females have two X chromosome, if a recessive allele appears on one, can be masked by a dominant allele. Therefore, it is very common in males, because the moment it is there, definitely the person is sick. Colorblind is made of two copies of in male, because Y doesn't carry, so it's out. The allele for colorblindness is recessive, located on Y chromosome. Never. Y doesn't carry. They're saying the father pass the alleles of colorblind to their sons. The allele, no. Remember, the father give them the Y, doesn't give them X. So this is out. So the most correct answer is A. I think you are counting your marks, né? You are counting your marks. We are doing for the distinction. I think our prediction was 95% correct, né? Yeah. So I expect you to get 95%, because that's what we talked about. That our prediction is always 95% correct, né? Yes. So make sure that you count until you get 95%. Okay. We talked about this yesterday, and I showed you how exactly you're supposed to indicate it. DNA profile of 4 individuals are given below. Okay, sharp. Let's look at it. Saying that which one of the individual is possible members of the same family. So it means that they have to be with something which is common almost throughout. Okay, first one is correct. First one is correct. Correct. Second one, no. Correct. Third one, correct. No. Correct. Fourth one, correct. The fifth one, correct. No. Correct. No. Correct. Correct. No. Correct. I think now you can identify. Correct. No. They're saying correct. I see. Then this one, no. Okay, if you say I take this, no, nothing is there. All right, let's see. This one is 1 2 3 4 5. 1 2 3 4 5 6 7 8 9. You see, 9. 1 2 3 4 5 6 7 8 9. You see, so it means that this has 9 bars which are similar to this, So the similarity here is only 5. Therefore, we can conclude by saying that it is W, Y, and Z are family members. Do we have W, Z, and Y? W, Y, and Z. So yeah, so C becomes the most correct answer. Kill. When two plants homozygous for the characteristics are crossed, expected ratio is, remember, for example, capital T, small t. So when you talk about that, it's capital T. They're saying phenotype. This with this, it's going to be capital T, small t. This with that, capital T, small t. This with that, small t. Therefore, these are tall, for example, tall, 3 of them, and then 1 short, for example. So the ratio is 3 to 1. We look for it. So A becomes the most correct answer. Then we go to the next question. Next question. Someone said that in Thunder we trust. I think, guys, Thundereduc we trust. Yeah, if you want a distinction, tell your friends, tell your brothers. Next year they must join, subscribe, so that they don't miss out the everyday classes we release on this channel, so that we help everyone. Subjects which we have not tackled this year, for example, agricultural science. Next year it will be here full. Fortunate enough, I'm the teacher for the agriculture science, so we will be dealing with that subject also. Thank you. Let's continue. 1.1.8. The diagram below represents the structure of chromosome at different stage of meiotic cell division. Which one of the following chromosome would be found in the cell during late? They're saying about late and anaphase 2. So remember that in anaphase 2, you expect the chromatids. The chromatids are supposed to be what? They are the one which are moving to opposite poles. Yes, but because they are late, it means that they could be straight. So we are looking for chromatids. Therefore, the answer becomes S. Therefore, we look for it. So it is C. So it is 2 marks there. Why not this? Because they are not chromatids. Why not this? Homologous. Why not this? Homologous. So it couldn't be the what? The answer. So they're saying that scientists who discovered Littlefoot. I talked about this yesterday, and then we say that the person who discovered it must be starting. Remember this all the Australopithecus africanus species. The people who discovered them, they start with R. So we say that it must be among these ones. So it's not Lideberger, no. So now it is Ronald, yes. Littlefoot, actually it's Ronald, the one who has the L, Littlefoot, Ronald Clark, or Ron Clark is the same thing. Ron J. Clark, whatever. It depends on what they have given you, but here they have given you Ron Clark, but actually it is Ronald J. Clark. All right, so C becomes the most correct answer. I hope you guys, you made it to 18 out of 18. Those who made it, congratulations. Yes, so we say that thank you very much for bringing about that. All right, so let's go to the next question, which is terminologies. I think let's talk about terminologies and then we see. Then they're saying that position of a gene on a chromosome, we talked about this yesterday, locus. The type of evolution where there is characterized by a long period of change and a little period of no change. We talked about this yesterday. Punctuated equilibrium. Yes, punctuated equilibrium. Yes, they're saying that the natural shape of DNA, yes, we talked it about yesterday, and we say that is double helix. And we emphasize that, please don't say double helical. No, it's double helix, nothing else. Type of bond found in amino acids, we talked about it yesterday. We said it's peptide bond, and we said that don't say polypeptide bond because we cross it. Peptide bond. Yes, so we talked about it yesterday. Then you're saying that the type of vision shared by but these people, they just did what we did yesterday. But thank you very much at least, because that was our prediction yesterday that these things are coming. Make sure that you go through them. All right. They're saying that the type of vision shared by primates and allowed deep perception is stereoscopic vision. Vision. Someone can ask me, what about binocular? I hope so. They will also credit about binocular, because stereoscopic and binocular, they mean almost the same thing. But here, because they talked about deep perception, then you have to talk about stereoscopic. But let me just show you in the distinction material what I'm trying to say, that they could credit you. However much, this book is not written by the department, it's written by MSID. So it's the same thing. I hope, let me check here, not yet there. Let me just go through here. I'm sorry about your eyes if it is hurting your eyes. I just want to show you something here. Yes, here, yeah, 2 minutes, 2 seconds, 2 seconds, 1 second, and boom, we are here. Saying that the eyes in front, stereoscopic vision. This is what we are talking about, because they didn't say that eyes in front. So the word binocular might be a little bit of not being credited, but stereoscopic, definitely it will be credited. But if they go under memo discussion, they could credit it, because they are talking about the perception. So I think those who wrote binocular, you are halfway, but those who wrote stereoscopic, 100% correct. Because they didn't just say eyes in front, they talked about the perception of, yes. All right, the type of however, binocular also brings about that the perception. So yes, someone is saying that you wrote binocular, don't be worried, so you could be credited about that, né? Yes. The type of dominancy which result in intermediate, when we talk about the intermediate phenotype in heterozygous condition, here we are talking about intermediate. The moment you talk about intermediate, you are talking about incomplete dominancy. Remember, you form, for example, capital R and capital W, meaning that if you have maybe red and white, you produce maybe gray, whatever. Yes, an intermediate, not red and not white. So that's what they are talking about. But because it is in this form, therefore it is in heterozygous state, different letters. We go to the next question. Just a moment. Just want to check here something. All right. Yeah. All right. Okay. We are writing. Someone is saying that wrote codominancy. The reason why they might not give you codominancy is because they say the intermediate. And there on our distinction material, let me just clarify for you, so that, because also this one could be a disturbing factor, that people could mix it with the codominancy, and the supposed to be at the beginning here of yes, at the beginning here. Okay, yeah, supposed to be around here. Yes, so you see codominance in complete dominancy. No, that one is fine. In complete dominancy, here we say that they say that this word, you see this word, it's what they said, intermediate. You see, intermediate, and then the title here is incomplete dominancy. And then if you talk about codominancy, it produces a mixture. It produces a mixture. Both allele are, yes, expressed. So it means that here you produce a mixture of two colors. So that's why I'm saying that incomplete dominancy is the best correct answer. Then we go to the next question. We are saying that the fluid fluid of the nucleus where free nucleotides are found, we call it the nucleoplasm. Plasm means fluid, and then nucleo from the word nucleus, therefore is the fluid of the nucleus. Then you are saying that the tangled mass of chromosome located within the nucleus. So we call this one chromatin network. So here is chromatin network. If you have any challenge, I can just show you on the distinction material. As I told you that distinction material, there is no question which cannot be answered by that book. Yes, so I wrote that book with the great time so that it can tackle any question they will set before and after you have written. So if you use distinction material, now I'm talking about those people who will be watching, not you now, definitely you will get a distinction. All right. Division of the cytoplasm, we call it cytokinesis. We talked about it yesterday. Say that and they're saying that the name of the, someone is saying Saying that is talking about the chromatin network. Talking about the chromatin network, it is here. We talked about it here. You see, entangled. You see, this is what you call chromatin. So it's a chromatin network. Yes. So that's what they are talking about. Here is the thing. Chromatin with chromosome consists of DNA and protein. So that is what they are looking at. This entangled. Yeah. That's what they are talking about. Ne? Yes. All right. We go to the next question. The name of the X and Y chromosome in male. We talked about this gonozome. So I think we are clear there. Someone is giving me the answer already. Gonozome is correct. Let's go to, I hope you guys, you got 10 out of 10. If you got 10 out of 10, congratulations. If you lost one, at least you tried. Yes, we're still in the range of 95%. Genetic disorder characterized by chromosome mutation. I don't know how many people got this right. Let me just say it because I have no choice, because I have to tell you what is the most correct answer here. They're saying that genetic disorder caused by chromosomal mutation. The answer is none. I'm sorry if you wrote something or both. Ne? It's none. Yes. Why none? Because these are gene mutations. They are gene mutation, not chromosomal mutations. When you talk about the chromosomal mutation, we are talking about the whole chromosome changing. Therefore, we go back to Down syndrome. And if you check on Down syndrome, sometimes we call it chromosome mutation, sometimes we call it chromosomal aberration. So when you look at Down syndrome, that's where you can find chromosomal mutation. Let me show you what I talked about Down syndrome. When you come here, yeah, I think I didn't indicate it here, but it is chromosomal mutation. Eh? Yes. I don't remember where I wrote it, but it's there, whereby you talk about the whole chromosome is changing. Yes, when you change the number or the structure of chromosome, then we call it chromosomal mutation. Sometimes we call it chromosomal aberration. Yes, aberration or chromosomal mutation. But if you just change the base, then we call it gene mutation. So these two are gene mutation because as a result of exchange of a base, a specific base. Then we are saying that importance of meiosis is both A and B because there is formation of gametes and then having the chromosome number. Yes, so this is both A and B. And then I'm saying that the organelle where DNA is found, both A and B. We talked about this, and yesterday I was tricking you, and then someone got it right. I asked a question that give me any other place where you can find DNA other than a chloroplast. No, other than a mitochondrion. And then someone saying that chloroplast. And then the person couldn't tell me that nucleus. Ne? Yes, so but what I wanted to tell them that think about mitochondria and think about the chloroplast. You understand? Good enough that this question came, and I hope that person tried to get the answer right. So the answer here is both A and B. Don't forget that in paper 1 I told you that you must say that both A and B, not A and B, not both. If you wrote both, I'm sorry. If you wrote A and B, I'm sorry, you did not follow the instructions. However, maybe with the mercy of God, then maybe they might correct it, but you have to follow the instructions. Now I'm talking about those who are going to be writing the next paper. And then we are going to question 1.4. I hope, guys, you got 6 out of 6. Ne? Yes, question 1.4. Oh, blood group. Talked about blood group yesterday, but let's see what was here. Name the type of diagram represented above. This is pedigree. Yes, so this one is correct. We talked about it, and someone was asking me what's the difference between pedigree and the phylogenetic tree. So now we are clear there. Give the number of the allele that control blood group. We talked about this yesterday. We said there are 3 alleles. Yes, alleles or alleles, whatever case you call it, is fine, man. Just a moment. Sorry, this Samsung is here disturbing me. Just a moment, guys. I don't want to this Samsung. Yes, I'm back. I'm back. I didn't want this Samsung. Okay, so we are saying that the diagram below represents the blood group in the family, or blood group O. Okay, sharp. Then we have name the type of diagram represented above. We said that the answer here is pedigree, so it is fine there. Then they are saying that give the number of allele. We said it is 3 alleles. How many generations are represented in the diagram? We said we count 1, 2, 3. So the answer becomes 3 generations. Yes. They are saying that Lena's genotype is this. Lena's genotype is A. Yes, you remember what we talked about? Okay, now let's go to the question. State the possible phenotype of Vusi. Possible phenotypes. Phenotypes or genotypes. They want the genotype of Vusi. So if you are talking about the genotype of Vusi, where is Vusi? Vusi is here. But now, if Lena is A, small a, Vusi, this person I don't know is small I. Remember, I asked a question that how is a person of blood group A and a person of blood group B can produce a kid of blood group O? Then I said yes, because if they both have heterozygous, they're in heterozygous state, they have the recessive allele, therefore they can produce a kid of blood group O. Therefore, how many marks they give it to Vusi is 2 marks. And they're saying possible. Therefore, Vusi can be A, small I. Yes, that's number 1. Vusi can be B, small I. Vusi can be I, small I, like that. Whereby if this combine with this, you produce O. If this combine with this, you produce O. If one of these combine with this, you produce O. You see that any of that can lead to Vusi. So they want 2 of them. If you wrote this and this, cool. If you wrote this and this, cool. Any of this, they wanted 2 of them. So I think, yes, you will get it right. Ne? Yes. Someone is saying that 4 generations. I don't know where did you get the 4 generations, but here is showing you 3 generations. Ne? Yeah. All right. Then they're saying that give the genotype of Bob. Where's Bob? So if this person is O, if this person is also O, definitely there's no way how Bob will be A or B. It will be O. So they're saying genotype of O, of Bob. Therefore, Bob is gonna be II. So I think that one is also correct. Give the name of the individual who displayed the codominancy. Codominancy means that you have blood group A and blood group AB. That's what they are looking for. So the first person I can think of here is you see here now, this person is blood group O. You see, blood group O. But the kids they produced is blood group B and blood group A. Definitely Anna is blood group A, B. Do you understand? Yes. So this one is here, can combine with this to produce B, to produce A. Sorry, this can combine with this to produce A, and this can combine with this to produce B. Therefore, Anna is definitely answer. But here it's showing you, so it's Anna. Is it Anna? Anne. Ne? Yes. So here they're showing you 2 marks. then you have to look for another one. If they don't want one, you have to look for another one. Another one, do we have another one? We can get another one just in case that Lena and Anne are kids of these two. You understand? But Lena is having blood group A, and then Anne is having blood group AB. Definitely, if this is blood group A, ne, and this is blood group A, how will they produce this? Maybe one is blood group AB. You understand? Whereby this combine with this to produce blood group AB, which is Anne. You understand? What if, because they said, did they say give the name of individual which displays codominancy? So the Anna is the perfect answer. However, based on this and this, so either this, because I can have this here, or I can be, maybe this can be A, because if I combine this with this, still I'll produce this person. You see, if I combine this and this, I'll produce this person. So is either any of this is either A like that, and then B like that, or is like this. Either of these could be. So it means that based on the analysis, deep analysis, not just easy analysis, it could be Curis and this, one of the two, not both. You understand? Not both. Both can't be AB. One of them must be AB. Then the other one has to be a heterozygous. You understand? Yes. So one of them could be. So the next answer, if Anne is the obvious answer, then the person who wrote either this or this could be also another correct answer. Someone is saying Vusi. What about Vusi? Let me see. Vusi is having blood group AB. No, there is no way how you can get blood group AB when someone is having blood group O. You understand? Because you don't have the second recessive allele. You understand? Yes. Yeah. Someone is telling me name. Thank you very much. Yes, I know that you are saying name. This is the obvious answer. Yes, it's a name. But don't forget that this could also be one of them correct. So the memo could have an extended opportunities after the discussion. However much, they could not see this maybe before they do the memorandum. But when you go to deep analysis, then you find out that this and this, there could be some student who wrote it and it could not be discredited. So should be credited if it results in that. You understand? Yes, obvious answer is that. However, even those, either of this could be an answer. We go to the next question. You're saying that the next answer is question below is a representation of protein. Okay, protein synthesis. We know that what is here is being protein synthesis. So now remember guys, we discussed about this and I told you that please don't make a mistake to go without knowing this. I told you that one of the paper, depending on which section, which paper they have brought, for example, if set 1, set 2, set 3, whatever, if it happens, one set must have at least transcription, maybe another must have translation, another must have DNA replication. One of it must come. And then that's what I said, and say that please don't go to the paper without knowing it. All right, let's continue. They're saying that 1, 2, 1, okay, sharp. Let's go to the question and then we see Z. What is Z? Name, identify Z. Where is Z? Am I lost? Where is Z now? Oh, sorry. I couldn't see Z. The process Z. Process Z is, what is this? Remember, this is uracil. And then this is DNA. Definitely, this is transcription. transcription. Remember, transcription is the formation of messenger RNA. So molecule R, where is R? Oh, molecule R is messenger RNA. Then they are saying that organelle Q, organelle Q, where is this? Where is organelle Q? Okay, organelle Q is the ribosome. Yes, ribosome. So guys, I used to tell you that whenever they think of organelle, think about two things, either nucleus or ribosome. So that the two organelles we talk about here, don't have any other organelle. Give the collective name for nitrogenous base O. This is O, ne? O is gonna be anticodon. Remember, we said that code for DNA, codons for messenger RNA, anticodon for transfer RNA. And I told you how to identify that this is a transfer RNA. Just write a T, transfer RNA. It's a T, and then you call it messenger. When you see it's a T, it's a T. So it's a transfer RNA. So the codes which found there, we call them anticodons. Determine the sequence of the nitrogenous base at S. Where is S? Oh, it's here. And remember, this is, let me check, this is T, and also let me check this T. This one, it has T, even this one has T. Therefore, this strand is for DNA. Therefore, I've just said T goes with A, and then C goes with G, and then A goes with T. So basically, that's what you're supposed to write there, here, the sequence. Which strand 1 or 2 is used as a template for formation of R? So we come back to R. So we have to mirror this. So this A goes with the T, U goes with the A, C goes with the G. So I have to look for what is this. I can continue. TAG. So strand 1 is the one which is used as a template so that this can be formed. Some people could have, maybe they have written strand 2 because this thing is facing that side, but that's just a confusion. Just a confusion to see that some people are off. All right. So the answer here is gonna be strand 1. Strand 1. Yes. Someone wrote spinal cord. Wait. Okay, sharp. Someone said it's a spinal cord. Aibo. No, man. You guys, you're acting in the paper. All right. AGT. Okay, sharp. Then they're saying that which amino acid 3 or 4 will be brought to area P. Area P. Where is area P? Okay. Guys, there is something which was hidden here. Let me show you that hidden part of it. And I hope you guys got it right using the elimination method. Okay, all right. If we follow this, following this, yes, I hope we can get it. But if you just follow what is here, because there are 3, and here you have the bases. So definitely this goes with A goes with U, because this is transfer A, U goes with the A, then this goes with the G. So UAG, so this one is gone, so we don't talk about it. G goes with the C, G, C, and then A. G, C, C, A. C, C, A, then this is gone also. Definitely this one will come there. You see? So you can see how you can identify it correctly. So basically here we come here and we say that amino acid number 4, amino acid number 4. So that's the correct answer there. Then they are saying that name the type of sugar that forms part of structure molecule R. Part of sugar molecule R. Part of sugar. Name the type of sugar, not part. Molecule R is messenger RNA. So here they are talking about sugar. So it's gonna be ribose. Don't say ribo. No, it's a ribose because they are looking at the sugar. So guys, I think you got 50 out of 50. Congratulations if you got it. Ne? Yeah, so it looked to be a little bit simple. Is there anyone who got 50 out of 50? Let me know in the comment section. Someone is saying 1.5.2. The collective name of nitrogen. They say the collective name of nitrogenous base. The collective name of. They are not talking about what is this. No, they are saying nitrogen. The key point here is nitrogenous base. So someone is talking about tRNA. tRNA is only this. But they're talking about the collective name of these 3 bases which are found on the tRNA. Therefore, the answer becomes the nitrogenous bases. I think I'm clear there. Let's go to question 2.1. Uh, 48, 35, 40, 18. Still, 18, you can still do it. You can still do it. Let's go to section B, and then you see how much you're going to collect with the 50. Yes. Section B. Then they are saying that name the structure. Name the structure. Name the structure. So here is, I told you about it. Yes. Name the structure. B. What is B? We come here, we look for B. Is a century. Remember yesterday I told you that please turn it so that it runs east to west. Is it east? Is it east way? Is it west? Is a? I'm not a geographer, guys. Ne? Don't quote me wrong. Ne? I think it's east this side and then west this side. I hope so. Ne? Yeah, so it must be running east west or in the horizontal way so that you see it clearly. For example, it's supposed to be like this so that now you see it must be running in that direction, ne? So that you see the actual phase. Yes, so I hope you guys you did it and identify the phase correctly. Okay, let's go through it and then we find out. Okay, now we are saying that B is centriose, spindle fibers. These are homologous chromosomes, but I don't know whether they're asking you. Okay, spindle fibers, we are done. Identify the phase in diagram 1. Diagram 1, what is happening? You are seeing here crossing over is taking place. There are 4. It is prophase 1. Prophase 1. Give 3 reasons for your answer. Why are you saying it is prophase 1? First of all, you are forming here a bivalent. Chromosomes are under crossing over. Homologous chromosomes are under crossing over, you see. Yes, that's number 1. The nuclear membrane is starting to disappear, you see. Nuclear membranes are starting to disappear. The centrioles are moving to the opposite poles. So you can talk about that. Yes. Yeah. If there is any other, it could be accepted. I don't know what you wrote, but as long as it is correct. Describe the process that takes place at A. They're talking about the process of crossing over. Homologous chromosomes come together or align together, and then chromosomes of homologous chromatids, chromatids of homologous chromosome, they cross over at a point called chiasma. Yes, they exchange the genetic material, and then they split to form two genetically different chromosomes. So basically you just explain the process of crossing over. As I say that you cannot go to the paper unless you are insulting me to when you don't know the process of crossing over. Yes, I talked about that. I said don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing the Metaphase. Don't go to the paper without knowing Anaphase. The rest you can skip, but those three, don't make a mistake to go there. I think those who read, you did not. Someone is telling me that spindle fibers are formed. Spindle fibers are formed, they are moving, centrioles are moving to opposite poles. Spindle fibers are not formed. Spindle fibers are forming. They are not yet, you see. Yeah, it's also correct. It's also correct. Spindle fibers are forming. Yes. Yeah, because if they are formed, they have already formed like this. So they are forming. Remember, in prophase, they are in the process. Everything is in the process of forming. All right. That's English, but in most cases, we also mark English. All right, let's go to the describe the process. We are done with that. Identify the phase, represent diagram 1, diagram 2. This I said you swap it because they're in pairs. Therefore, it is meiosis 1. Therefore, because they're in the middle, you see they're in the middle, you swap it, you turn it. Ne? Yes, as I told you, east to west. Ne? Therefore, it is metaphase 1. So the answer there becomes metaphase 1. They're saying that describe the difference in the event that takes place in phase mentioned above. Describe the differences in the events that take place in the phase mentioned in A and the same phase during mitosis. Or they want the differences between this phase, metaphase 1 and metaphase. Remember, mitosis doesn't have metaphase 1, metaphase 2. Here, chromosomes align along the equator, yes, in homologous pair randomly. You see, randomly. Chromosomes align along the equator in homologous pair randomly. While here, individual chromosomes align along the equator singly. In actual sense, they're asking you meiosis 1 and meiosis 2 of the same phase. Remember, we said that meiosis 2 looks like mitosis. Yes. So basically, chromosomes align along the equator randomly in homologous pair. That's the key point. Homologous pair. Yes. While here they align along the equator singly. Yes. So they're saying differences. They're not talking about tabulate. So they don't need you to write the table or to draw the table. Describe the results at the end of meiosis if chromosome D fails to separate. Chromosome D. Okay, let me just turn it so that you see it clearly. What happens if chromosome D does not separate? So basically, this is not a human cell. So don't talk about 47 chromosomes. Here, what you say, okay, definitely it's going to be like this. Technology is jamming. Okay. Still jamming. Okay, it's supposed to go that way. Just a moment, just 2 seconds. As it's back. It's back. Okay, now you're saying that it's gonna go through like this, and the whole thing, this becomes one cell, and then this one becomes another cell. So it means that what you describe here, say that one cell we have 1, 2, 3, 4, 5 chromosomes. Yes, one cell will have 5 chromosomes, while the other ones instead of 4, because it's supposed to be 1, 2, 3, 4. Then the other one will have 1, 2, 3. So you have 3 chromosomes instead of 4. You see? Yeah. So basically that's what they are looking at, or they are looking for. Yes, that's what they are looking for. So that's why they ask, describe the result at the end of meiosis. At the end of meiosis, yes, if D fails to separate. Yeah, so it means that one cell will have an extra chromosome, 5 chromosomes, while the other ones will have 3 chromosomes instead of 4, and then the other one will have 5 chromosomes instead of 4, meaning that you have one extra chromosome, and then the other one will have one short of chromosome. That's it. Then number 2, those people who wrote about 46 chromosomes, they didn't talk about here that this is a human what? They didn't talk about that this is a human cell. Yes. Number 2, look critically here. Let me show you something here. If this cell, these two cells, this one is going to receive X, 1, 2, 3, 4, 5, and then this one is going to receive 1, 2, 3. So the fourth one is going to be empty. I see. Yes. So it means that now when this one divide, this cell is going to have 1, it's going to have 5 of them, and then the other one, when it divides, they're going to be 2 of them, but each will only have 3. While this one, each of them, remember it's meiosis 2 now, each of them is going to have 5 because it's supposed to split equally. Each of it is going to have 5, and then this one, each of it, because there are 3 now, this one is 0, it's going to be 3. You see, it's going to be 3. That's another option, but still it gives you 3 here, 5 here, 5 here. 2 cells will have 5 each chromosome, while instead of 4 each, and then this one will have 3 each instead of 4. I think I'm clear. If you wrote it in that way, still they will credit you. Describe, so we're done with that. Describe the process of DNA replication. Perfect. I think I was clear here, emphasized, and even say that if you could see my face. All right, DNA replication is a process by which DNA makes identical copy of itself. You understand? Yes. So you now tell us that DNA double helix unwind, weak hydrogen bonds break, and then weak hydrogen bonds breaking is the same thing saying unzip to form 2 templates, each. So to form 2 separate strands. What you want to hear the word separate. 2 separate strands. Each is acting as a template for formation of new strand using free-floating DNA nucleotide. Remember, if it is transcription, messenger RNA nucleotide. But here we say DNA nucleotide from the nucleoplasm, whereby a complementary strand is formed whereby A goes with the T and the C goes with G. Is it clear? And then 2 new DNA strands are formed. 2 new DNA are formed, each with 1 original strand and each with 1 new strand. This process is controlled by enzyme. It could also be right. So this question has more than 6 marks. They have given it to it. So it means that the only credit 6, and then we write maximum, meaning that you have reached the maximum, therefore you got it. People are always asking me about the spelling. Okay. Tando is asking me about the spelling. Tando, what if I call you Tondo? Will you feel good? Instead of calling you Tando, I call you Tondo. I replace A with O. Will you feel good? Will you be the same person? If you are not the same person, why do you want to make mistakes with the spellings here? All right, it will depend on them, whether it means something else or it's the same thing. But we always encourage you to write correct spellings. All right, let's continue the next question. Read the passage. Gene, okay, code for blood clotting in humans. This gene made up of that 163 amino acids. The mutation occurred and result in amino acid 123, 128 sequence. This changes to this, and then this changes to this. Mutation has been transmitted as autosomal. Yes, it has been transmitted as what? Autosomal. And they are saying that they're saying that the mutation has resulted in a resistance of warfarin drug. Warfarin drug in human. Warfarin is used to treat prothrombosis. I'm used to the process of clotting. Prothrombin, fibrinogen, fibrin. Okay, prothrombin is the one which produce thrombin. So here thrombosis is the disease. Okay, treatment of thrombosis. Thrombosis results in the formation of blood clotting in the arteries, and then this causes the thin of blood to break down and the blood clot. Name one piece of evidence in the information that show that mutation for this gene occurred in DNA molecule. How do I know that mutation occurred in DNA molecule? Check here. Yes, it changed from here to this and here to this. Where do you find the T? Definitely find T in DNA. Therefore, that is the correct answer. So you talk about that, yes, the sequence change of this, change to this, and also this change to this. This shows that it was DNA. You understand? Yes, because DNA is the only one which has the thymine. Yes. Then number 2, they're saying that how many nitrogenous bases code for this gene? Hey guys, I talked about this. I even showed you how to calculate these nitrogenous bases. Remember, each nitrogenous base, each amino acid is coded by 3 nitrogenous bases. Therefore, I will say that. Yeah, someone is saying it has thymine. Yes, correct, because of this thymine. Yes, presence of thymine. Yes. Okay, saying that how many nitrogen? You're gonna say 163 amino acids times 3. So the answer you get is what you call let me see, how much is it? Okay, you calculate. Let me know the answer in the comment. Yes, describe what is meant by autosomal dominant allele. Autosomal, it means that is a non-sex chromosome. Non-sex, it is not carried on X chromosome. You understand? It is not carried on X chromosome. This allele is not carried on X chromosome and is dominant. Yes, it is dominant. That's what it means. It's an autosomal dominant allele. It's dominant and is not carried on X chromosome. So this disorder is caused, or this condition is caused by a dominant allele which is not carried on X chromosome. Yes. Then they are saying that the table below shows the amino acid and their corresponding codons. Hey guys, when I was trying to explain to you these mutations, I told you that you need to take note of this. Yes, I said it. The table below shows the amino acids and their corresponding codons. So meaning that this is messenger RNA, because they said that codons. They didn't say transfer RNA or anticodon. How will the mutation on this result in resistance of this. Check, 5 marks. What are you supposed to do now, man? What you are supposed to do here is you come back here and read here. A mutation occurred affecting the amino acid this, and the sequence of this to this is changed, becoming this. This mutation has been transmitted as an autosomal dominant characteristics throughout the generation. This is the problem now. It has formed another protein. You understand? But how do I know that he's forming another protein? Okay, now the first step to score those 5 marks, you had to tell us what we started with and what we ended with. So we started with G, T, G, which changed to C, A, G. You understand? And then you have T, A, T. Someone said that is 489. Here is 489. Guys, if it is correct, you calculate it, then that's the perfect answer. Then you have to tell us that this one changed to from what? Okay, let me just make a big space so that we can know exactly what we are trying to show you. Okay, this is the first amino acid, this one, né? So first one, and then the second one, let me write it here. It is T, A, T, and then T, C, T. But now, what do these ones indicate? They're saying these are codons. Yes, so I don't know, I can't read it here unless I change this. So change it. So this is the first mark by changing G, A, C. And then this one is G, U, C. What does GAC mean? GAC, let me first remove this so that I see clearly. GAC is leucine. It indicates leucine, this one. And then GUC indicates glycine. Yes. Now, what about this? Let's convert this. This is gonna be it's got A, U, A, and then this one is gonna be A, G, A. So what does this one indicate? This one indicates this one indicates AUA. AUA is phenylalanine. Phenylalanine is this one. And then this one, what does it indicate? AGA is arginine. Guys, these are just the names, né? Yeah, you don't need to know them by head, né? Me, I'm saying them that arginine, what? Yeah, because we find this in biochem, né? Yes, in biochem. So this is it. These are the amino acids, which you know that, yeah, the essential and non-essential amino acids. So as long as you write what they gave you. All right, now we have translated. You see, we have what? Translated. Here we will get a mark. We will get a mark. It's 1 mark, 2 marks. Then also changing this is another mark, 3 marks. Then changing this to this is another mark, which is 4 marks. You understand? But now put them in words. The codon, the first codon, AGA, CGA was changed to ACG, cool, which indicates that this was changed to this, okay? And then this produced leucine. It produced glycine instead of leucine. So you have changed the amino acid. And then also this one, the second phase, we changed the amino acid from phenylalanine into arginine. So different amino acids will be changed. This will be changed into this, and also phenylalanine will be changed into arginine. You understand? which makes a different protein. It won't be the same protein because these codes which change, they change the amino acid. So therefore, a different protein is formed. That's what it means. So you guys were supposed to show us that this change to this change to this, meaning that new amino acid is formed. So it will be coded for glycine instead of leucine, and then here it will be coded for arginine instead of phenylalanine. Therefore, a different protein will be formed, and then this protein is resistant because they said the mutation has resulted in resistance. Yes, this protein which has been formed result in the resistance to this drug. So basically that's what they wanted. I explained it yesterday, but now when I'm seeing this question, the way how they try to trick it, when I was explaining it, I explained using one example, and I told you that don't make a mistake to go to the paper without knowing this, because I know they're going to bring it, and I know that people are going to have a challenge to explain this. And these people, you know what they do, that if they find out that this question is hard for these students, they keep on bringing it. I don't know why, but they keep bringing it. Maybe they want to make sure that everyone knows that concept. But if they see that now you can know this one is easy, they don't bring that one anymore. They bring more, which is a little bit difficult to see that, yeah, you can apply. But I hope you guys, you got this question correct. Let's go to the next question. Let's go to the next question. They're saying that they're saying that the effect of this mutation has explained the effect of this mutation has with the prothrombic. This one you just come back to the extract and then you read here. The mutation has resulted in resistance, you see? Yes. And then in the human of treatment of used to treat this, the thrombosis result in the formation of blood clot in artery. So this will result in the, yes, you see, the effect of this mutation on human with this. So meaning that it will result resistance. First of all, the mutation will bring about resistance. Number two, it will also bring about this formation of blood clot in the artery, you see? Yes. And then also will cause this to break down, you see? So basically, no. This one is out because this is a drug. Né? So it means that it will not allow this blood to break down, the clot to break down, because now it is now resistant. So meaning that the blood clot will remain there. Why? Because of the mutation which has developed. Therefore, you talk about the resistance, and then you talk about what this you see here, this formation of the blood clot in the artery. Therefore, these clots will not be broken down because of the resistance which has been developed. So basically, the answer was supposed to be obtained from there. Hemoglobin. Someone is saying that if I could say that it's okay, has deleted it. Okay, it's fine. So guys, I think this question was one of the challenging questions, just challenging questions, and I knew that it's coming. And then when I was emphasizing it, yes, I tried to see that everyone can pass it. Yeah, we go to the next question. That is question 2.4. Question 2.4, we are saying that polydactyl, these are the people who have extra finger. Maybe they have 6 instead of 5. Ne? They have 6 instead of 5. Lead to extra finger on extra finger or toys. It is caused by a dominant allele. This word, you remember I told you guys when you talk about dominancy? Yes, a man who is heterozygous for this, meaning that is capital R, small r. Yes, because they said use R. Ne? and a wife who is not, meaning that it's a recessive, so is homozygous recessive. Use the letters R and the RR, do the genetic cross to show the percentage chance that their children will have polydactyl. The percentage chance of having the polydactyl. So here, what are you going to write? So it's simple, guys. Here is very, 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 very simple. Let me just talk about it here. P1, F1. I told you about that, and I said we gave you a mark. Then you say phenotype. I think you guys wrote it that. You go back to the question, you find the phenotype. They say that homozygous, a man who is having the condition, a man with polydactyl. Yes, polydactyl. So you're gonna have to say a man with, yes, polydactyl, crossed with a lady without polydactyl. Yes, meaning that here I'm gonna have a lady, wife, with no, yes, here with, yeah. So you finish. Then here is genotype, but they say the man is what? They say the man is heterozygous. Therefore, I'm gonna say capital R, small r, crossed with a lady who is recessive. So here I'm gonna say meiosis occurs, and then I'm gonna say capital R, small r, small r, small r. I say these are gametes. If you didn't circle them, it's fine. If you circle them, it's fine. Yes. Then now you can use the Punnett square method, yes, or you can cross. Either way, as long as it's correct, but you write the word fertilization if you use the Punnett square, or you write fertilization here, fertilization here, and then you say C, and then you start capital R, small r, this with this, capital R, small r, this with this, small r, then this with this, small r. So these are offsprings, you understand? Yes. So basically, this is what you're supposed to do to get it right. But now we come here, we mark. If you don't know, if you got this wrong, then automatically we shall come here, we say fertilization, it will give you a mark. And also writing this, you see now you have 3 out of what? Out of 6. But if you got all these correct now, but the question is asking you, there might be a compulsory mark. Let's find the compulsory mark here. They're saying that use the letters, do a genetic cross to show the percentage chance. Yes, the percentage chance of so percentage is a key point. Is a key point of having the kids with the disease. So basically, we have 2 people, normal, is caused by a dominant allele. 2 people with the disease and 2 people without. So basically, it's gonna be 2 out of 4 times 100, which gives you 50%. You see, this is a key point. We mark you. So we're gonna mark you here. We mark you here. We mark you here. But we're gonna first count this. How many marks are there? How many marks we are given? Yeah, we're gonna give you this one plus any other 5. Yes, or it will depend on, but this is the compulsory mark which they're gonna give. Someone is asking me about agriculture science. So many people asking me about agriculture science. Guys, I told you that unfortunately this year I'm not sure that you're gonna work with agriculture science, but next year 2024 we will work with agriculture science. Yes, it's because currently we don't have a school which has which does the agriculture science in this area. Unless you send it to me, you scan it, you send it to me, then we'll do a memorandum for you. Yes. All right, yeah, saying that below shows the graph below shows, I think guys you got all the marks here. I'm skipping, I'm sorry. Okay, so we are done with that, so let's go in summer squash plants. White fruit color is white fruit is dominant, né? So we need to know that white is dominant over yellow, and then round is dominant over oval. So this is dominant, white over yellow. Okay, now let's start. Summer squash plants that are homozygous for white and round is crossed with plant that is homozygous yellow. Okay, sharp. Homozygous. Remember, yellow is recessive, ne? And then this one is white, is dominant, and they are saying that dominant around. Oh, dominant over crossed with recessive. Okay, give the genotype of parent one, parent in the first generation. Okay, parents in this case they say that white, therefore capital B because they said homozygous, and then white, and then round is capital D because they said homozygous. So that is the first, and then the first mark, and then the second mark could be is the crossed with the homozygous yellow. Yellow is, we said, is small b, and then oval is small d. So that's the second mark. So that's what they are looking at here. The phenotype of F1 generation, because all of them are dominant, therefore is going to be capital B, small b, capital D, small d. So the phenotype both is going to be white and yellow. So it's going to be white, correct, and oval. Did I say? Not oval, man. It's not oval. The one which is dominant, because I have capital D. Capital D here indicates round, right? Round. So it's going to be white, a mark, and round, another mark. Then they are saying that two plants that are heterozygous for both characteristics were crossed. Okay. What if I use, someone is asking me, what if I use X and Y? Guys, I told you that in this generation, in this, you only use X and Y if they have told you to use X and Y. So they didn't tell you that this is sex linked, né? So the answer will be wrong. But if you wrote this, and this, then automatically you will get the answer. Those remarks, and if you got the percentages 50, you'll also get another answer. At least maybe you'll get 4 out of 6, né? At least you did not come out with 0, né? Yes. They're saying that the two plants that are heterozygous for both characteristics were crossed, meaning that I have capital B, small b. I have capital B, small b, capital D, small d. So this is what I have. So if they are crossed, all possible genotypes of the gametes. So here I'm going to find out the gametes. This gonna go with this, as I told you yesterday. This is exactly the same thing I told you yesterday. This we're gonna be go with this. It's gonna be capital B, small d. Then this with this, it's gonna be capital small b, capital D. This with this is gonna be capital B, small d. So these are the gametes which are possible. So 4 of them, maybe 2 mark by each. So you get that correct. Then we go to the next question, guys. If there's any question I've skipped, you let me know, né? Sometimes I skip when I don't see. All right, how many plants in, okay, so how many plants in the next generation are likely to have yellow and oval fruit? How many plants in the next generation are likely to have yellow and oval fruit? Yellow and oval. So we come back here and look at for yellow. Where is yellow? Yellow and oval. Yellow. Okay, now they're saying that, okay, both were crossed, heterozygous. So automatically when you cross, you'll find out that because both are homozygous recessive, meaning that they are going to be small b, small b, small d, small d. So the answer here is going to be one plant, yes, or maybe 1 out of 16, yes. So one plant, or one plant out of 16. I think let me just show you here from the distinction material what they are trying to show you without doing the genetic cross. Yes. This is what I'm saying. You see, they are looking at this. It's only one which has both letters when they are homozygous recessive. So basically that's what they are looking at. When you come back here, that's what they are looking at. It is small b, small b, small d, small d. That's what they are looking at here. Capital small b, small b, small l, small l. Yes. So and the percentage here, if you count all of them, they are 16. Therefore it is 1. It's only they are looking for this. Yeah, one individual, or 1 out of 16 individuals. So I think I'm clear there. Then we go to the next one. Yeah, someone got it right. I think, guys, you are collecting marks now. You are collecting. Give a possible genotype of both parents that must be crossed. A farmer wants squash, okay, that way, white and oval. Yes. So the two, if you want white, basically, if you want, all of them are supposed to are white, which is capital letter, and then oval is a small letter, meaning that the resultant must be capital B. For example, it must have a capital B, and then it must have 2 small d's, because they say the oval, and the oval is recessive, you see. So basically the genotypes of the parents must be this: capital B, small d. That's the first person, and then the second person can be capital B, small d. That's the second person. Or the first one can be like this, and then the second one can be capital B, small b, capital D, because this B will be somewhere. Yes. So basically, if you wrote this, they will give you a mark. I think they want to, let me see, possible genotypes. They want to this with this. Yes. Or this with this is cool. Or this with this is also cool. So yeah, that's what you were supposed to write, so that at least you get the correct mark there. So there are options there. If I have not mentioned another option, then automatically it means that it will be marked correct. Who got 50 out of 50 in section question 2? 50 out of 50. Let's go to question 3. Someone is asking me, you describe, I don't know, what was that question? I don't remember what question was it. Okay, let's go to the next question, and then they're saying that the graph below shows the results of artificial selection for protein content in the meal plant over 50 generations. Describe how the farmer did artificial selection. Here you just had to explain the artificial selection. Remember, in artificial selection, you identify the character, describe how this farmer did. So he identified, you see, they identified the character of producing protein in the meal, and then after that he he identified those plants with that character, yes. Then he crossed those plants with those ones which have that character, which have that character. He crossed them, yes. Then after that, he selected only those which produced more in the first generation, and they were allowed to cross-pollinate because they don't do sex here. This is close pollination. It's pollination. So he he cross-pollinated those which could produce large number of protein, you understand. And then at the end of the day, he found out that the plants were able to produce large amount of protein. What was the average percentage of protein content in the meal in at the 15th generation? So here you just had to draw a line touching there, and then you draw a line touching there, and then you find out maybe 12.8, and then you put these units, kernels. Yes, so that becomes the answer. And then someone is going to ask, what about 12.89? Or what about 12.9? Yeah, they will give a range. Yes. Someone got 38. That's still good mark. 42, still good mark. Is good mark. All right, guys, thank you very much for getting those good marks. At least you are promising. But by how many times did the average percentage of protein content in meal grain increased between this, meaning that they are looking at for percentage increase. They have brought the question again. Eh? By how many times? By how many times? By how many times? Okay, the average percentage of protein, yes, increased between 40 and 45. I come here, I find what is final. The final is 20. 20 minus the initial, which is 14. So you get how much here? You get 6. 6 divided by the initial. The initial, which is 14, times 100. So the answer you get here is the number of times it increased. Yes. In terms of percentage, saying that by how many times did the average percentage of protein, yes, okay, you had from 14 to 20. So someone might have a different version. So you guys, you have to calculate for me and say it's only 1 mark. So you how many times from 14 to 20? Basically, that's what they are looking at. So when you do the percentage increase, then you'll be able to know how many times. And then here, you guys, you calculate and you get the answer. Yes. Describe one way in which the process of artificial selection is different from genetic engineering. We talked about this. Artificial selection, there is no manipulation of genes. You only have to select those organisms which are desirable by the human, and they are given a chance to reproduce. While in genetic engineering, you don't have to cross these organisms. You just have to select using, there is manipulation of genes. That's why it's called genetic engineering, manipulation of genes. While here, it's only you cross the organisms. You don't touch the genetic composition of an organism. Yes. Number 2, this one is a little bit of expensive. This one is a little bit of cheap, in which this process they differ. Okay. This one takes short period of time, while this one takes generations and generations. Yes. Someone is saying 12.5. Someone is asking me, is it fine if I don't put the units there? Hey guys, those are the discussions we do, or the discussions they do when it comes to the discussion. Should we correct this? Should we not? So basically, what you need to do is you must not always forget about the units, because the units are very, very, very important. All right, let's go to the next question. Darwin's theory of evolution by natural selection. I explained this and I told you that unless they bring it as like this, then I also explained to you what you're supposed to say. Natural selection, you talk about there is variation among organisms, those which are favorable, those with favorable characteristics, because they didn't give you here an example. So you don't pick an example. Those with favorable characteristics and those without favorable characteristics. Those with favorable characteristics, they reproduce in large numbers. They survive and reproduce in large numbers. And they pass their genes to the next generation. And those without favorable characteristics, they die in large numbers. This is called natural selection. So those which pass their genes to the next generation, yes, they reproduce in large numbers, and then after here you also supposed to talk about, just in case, because it's 7 marks, maybe you can talk about natural selection is what is a survival of the what of the fittest, which organism is best suited. Sometimes when students, they are trying to define this, because this is a block question whereby it is no example, no what, you just have to explain just what you know. Yes, you on natural selection. So basically, this is what I'm trying to say. Yes, I'm almost there. Yes, here. Natural selection. Yes, it's natural. Organism produce large number of offspring. There is a great deal of variation. Some have favorable characteristics and some do not have those. When there is a change in environmental conditions, yes, if there is competition, the organisms with favorable characteristics will survive, and then those with unfavorable characteristics will die. Organisms with those which will survive will reproduce in large numbers, thus pass their alleles to the next generation, to their offspring. So basically, that's what they are looking at. Just describe the process of natural selection. That's it. Then we go to the next question, guys. That's what they wanted from there. 3.33, investigation was done to determine the relationship between height and the head and the bite force. The head sorry guys, there was some technical problem with the sound, but now it's back. Okay, they're saying that what I was saying that the investigation was carried out to see the relationship between height of the head and the bite force in lizards. Ne? Yes, the bite force in lizards. Scientists, okay, collected all of that, so you are supposed to read there. Ne? So now, what is the first question there? What is the independent variable? So the species will determine the head and also determine the force of the bite or bite force. So the species becomes I, D, X, Y. I told you that. Ne? So this becomes the species, becomes the independent variable. And the independent variable, then the bite force. The bite force. Yeah, someone can ask me that, what about that? Height determines the force of the bite. You understand? How much force do you need in neurons? Yes, the height will, because as the height increases, the force also increases. Yes, but what determines this is this the type of the species. So yes, if it was this and this would have been the independent variable, dependent variable. But now we have this which determines this and this. You understand? Yes. So the species will be the independent variable, and then this bite force is the most correct. But I hope they also credit for those people who wrote the height of the head. Yes. Because if you are to plot a graph, you'll find out that this is A, this is B, this is C, and then these two will have two graphs in that case, whereby this is the independent. And someone talking about full marks by Darwin, that great man. Yes, state two factors what were kept constant. Factors which were kept constant here, they talked about, there are a lot, so you have to talk about in a factor, but here they talked about that each species were kept in a cage with environmental conditions similar to that of the habitat, meaning that you kept maybe same temperature, same humidity, same light, same, talk about environmental factors which you could keep constant, or any other factor which you read there and then you find out that it was kept constant. So they're saying that apart from the sample size, state one way in which the reliability of the experiment was ensured. So when you come back here, apart from the sample size here, what did they do? Did they repeat the experiment? Yes, they say that the height, was measured for each lizard and the average. So they calculated the average height of each lizard. You see, this is also increasing reliability. Yes. Then they are saying that school name the type of variation which is displayed in this characteristic. I told you that whatever you are born with, and it changes since you are born up to where you are now. How big was your head before when you are born, and how big is it now? How big is your size, and how big was it when you are born? So things which change like that, then it is continuous variation, because it's all individuals, they have the same. But if it does not change, like blood group doesn't change, so it means that it's discontinuous. So in this case, name the type of variation, because the size keep on changing, therefore we will say that it is a continuous variation. Describe the relationship between height and the head of the lizard in terms of the bite force. As the height of the head increases, the bite force also increases. You see, 10, you see, so the bite force also increases. Which species A, B, C would be expected to be feeding mainly on rough fibrous plant? So we are looking at the one which has the force which is strong enough. So we have this. So its answer is C. Why? Give a reason for your answer. Because it has a strong bite force, so that it can chew these tough fibrous plants. Yes, so it has a strong bite force. Yes. Then they are saying that explain, so we are done with that. Which species A, B, C would be the most suitable to live in narrow areas of the rock? With the one which has the small head, because it can go through the narrow or the crevices. So basically it is species A. They didn't ask you the reason, but if they had asked you the reason, then you'd have said that because it has a small head. Yes, it has a small head. So that is the correct answer. Another question is 3.4. 3.4. Guys, are you getting it? If you have any challenge, let me know. Let me know. Maybe you are not so sure about the answer I talked about. Yes. Let me know. Let me know. Let me know. Eh? Yes. Let's go to the next question, and then we try to do the last one. Okay. Which species in the diagram? What is this diagram? Is a phylogenetic tree. Yes. Which species in the diagram above existed or survived for the longest period of time? You look at how long the length of the bar. Check. It's this one. So the answer is Homo erectus. Then we come to calculate the period of million years. Australopithecus afarensis and Australopithecus africanus coexisted. So here, remember I told you this and I gave you those questions that you will never be asked a question and it will never be fail to answer it. Remember when I was going through this and I told you here period of existence, you see, competed for resources. So that's the same question which they are asking you here, that which give two pair of species which competed for resources. So now what they did here is they gave you that. Now they're asking you here, you see, they coexisted. In other words, they competed for resources. But how long did they do that? They did that if this Australopithecus africanus is here, and then this is afarensis. Did I say afarensis? Wait. Afarensis and africanus, yes. So this is the time when they competed for resources. So basically that's the time. So I have to calculate here. So it is 2, 3, 4, 2, 1, 2, 3, 4, 5, 6, 7. So it's 2.7. Remember we count from this side because the 0 is this side. And then this one is 3, 1, 2. So it's 3.2 minus 7, 2.7. And then the answer you get here is million years. Not million years ago, because here they are talking about the range. It's million years. So guys, I think when you subtract here, what is it? What is 2? Yeah, make sure that you subtract correctly, né? But it must have that word million years, because in most cases we mark the what? The units. Name the species that was also known as handyman. Homo, I told you this. Homo habilis. Remember when I was going through many schools trying to explain these skulls and stuff. I hope you guys you got it right, ne? Got it right. State 2 uses of tools in developed. State 2 uses of tools that was developed in 2.6 million years ago. 2.6 million years ago. 2.6. So it's 2, 1, 1, 2, 3, 4, 5, 6. So something which, yeah, it's this one. Yeah, the other one tools. Yes. So what is it? You just copy from here, because they said this for So there is no cramming there. Then so we got the answer there. Identify 2 species that use the most complex tool. 2 species which use the most complex tool. The most complex tool is this one. So if you look at it here, it is the Homo sapiens and the Homo neanderthalensis. Homo sapien and Homo neanderthalensis, or Neanderthals. Sometimes you call them Neanderthals, but Homo neanderthalensis. Yes. Is there anything I've skipped? Yes. Okay. Let's continue. Name 2 species. So we have done that. Explaining how the change in the brain size over time relates to developing tools. I explained this also. that increase in the brain size increases the number of cells. Yes, so this number of cells, if they are increased, then automatically it means more intelligence. And these sophisticated tools require thought. They require what? Thought. Therefore, they will require more intelligence. So basically, that's what they are looking for is here. We talked about it here. Cultural evidence. Using tools in cutting or improving in nutrition, developing of tools become more sophisticated as hominids evolved. Tools require planning and thought. You see? Yes, this is what they are looking at. Tools require planning and thoughts. This is what they are looking at. Development of tools become more sophisticated as the hominids, yes, as the hominids develop. You understand? Yes. Or as the brain size changes, yes. And then they become more sophisticated, and these tools, they require planning and thought. Therefore, you have to increase the brain so that you can increase the intelligence, so that you can plan and you can also think about what you are planning. So basically, that's what they are looking at there. Then we go to the next question. They're saying that name two species used to, okay, explain the size. So we are done with that. Let's go to the last question. Yes, the last question. Someone is saying that what if you calculate it differently but you got the answer? It's cool. It's cool. Maybe they will give you a mark. How many marks are there? Which one is it? I don't know. But if you got the answer correct, maybe, because here they're saying show your answer, you're working, maybe you just only get one mark for the correct answer. Yes, and then you lose the 1 mark for the Let's go to the next question. 3.5. State the genus of Adipithecus, Australopithecus sediba. Australopithecus. It's this one. You have to write it in full. Australopithecus, because they're looking for the genus, the first name. Yes. Describe, it must start with a capital A. Describe the shape of the spine. We talked about this. Is S shape. They're saying describe. Just is S shape. That's all. S shape. Then they are saying that state what is Hosi Dube. Thank you very much. You are also the best because you kept yourself on the channel, and we pray that you get that distinction you are crying for, and I know you will get it. You will get it. There is one person I want to thank. Has been a member since the beginning. Oh, I've forgotten the name. Maybe I'll thank her in the next video. Yes. Yes. Okay, state what is meant by the transitional species. We also talked about this. Transitional species is a species which has both characteristics of the present species and the most recent common ancestor, and the most recent species. You understand? And the most recent species. Don't say and the ancestor. Ancestor where? And the most recent what? Species, or before it. This is what I'm trying to say. When I was yes, here is transitional species. This is a transitional species. It is having characteristics of this and the recent ancestor, or the most recent what? Ancestor. Yes, so that is a transitional species. That's how you're supposed to connect it both in front and backward. So you're supposed to explain that in 2 versions. That's why it is 2 marks to talk about in front, and 2 marks. Yeah, but in most cases here we use the all or nothing law. You either get it all or you lose it all. What if you misspell the Australopithecus? Yeah, you guys, spelling. Maybe they will correct you if it doesn't mean something else. But if it means something else, they won't correct you, ne? So what is this? And then what is this? Do they mean the same? They have the same letter, but differently. So it depends on how you have spelled it, ne? Yes. And they're saying that give the letter of the pelvis that would be represented by, okay, give the letter. Remember, I said that Australopithecus sediba is a transitional fossil. Remember, I talked about it and I emphasized it here. Check. Australopithecus sediba is a transitional species of fossil. You see the distinction material? Yes, those people who used it correctly, I hope you're going to get a distinction, because there is no way you're going to fail to get it. No question can be failed to be answered by that. Okay, they're saying that give the letter of the pelvis that would be represented by Australopithecus sediba. In this case, you look at this is more wide, but it's not that wide. This is more narrow, but it's not that narrow. So this is like 1, 2, 3. So this is in the middle. So this is Australopithecus sediba. Therefore, the answer becomes A. Explain your answer, because now you have to relate these two. That Australopithecus sediba, yes, this A, the pelvis is not as wide as C and is not as narrow as B. Is not as wide, yes, meaning that is wider than this, but still is not as narrow as this. This one is much more narrower and not wider compared to this. So it's not as wide as this, but it's not as narrow. So sorry, it's not as it's not as narrow as this, ne? Yes. Or how many marks are there? 2. Yes. So basically you would have said that is not as wide as C and is not as narrow as B. That's the first mark. If you also wrote like this, you will get a mark. This is not as narrow as this. No, not narrow. This is not narrow. Is not as long. This skull, this pelvis is not as long as B and is not as short as this. You understand? This one is shorter compared to this, but this one is longer compared to this, which means that this is in between these two. If you look at how long it is, how long it is, how wide it is, how wide it is. You either use wide to explain, you have to compare this with this and this, or you have to use how narrow it is. So not narrow, how short it is, short and wider, isn't it? So it must be not as wide as this and not as narrow as this, or you say not as short as this and not as long as this. This is not as short as this and this is not as narrow as this. So basically either way or whatever you have written, then you will get those 2 marks. We go to the last question, the last bullet in the gun. The last bullet in the gun to see who got 150 out of 150. Explain the significance of change in the prognathism from Australopithecus sediba to Homo sapiens. Prognathism meaning that protruding jaws. But there's something which you need to know. When there is protruding jaws, check if this from here to here, check if this protruding jaws. Basically, the prognathism is reducing. Yes, because we said that this is prognathus, this is not prognathus. So it's reducing. So they are not saying increase or decrease. They are saying explain the significance of the change in the prognathism from Australopithecus sediba to Homo sapiens. So the prognathism here is reducing. So when you become more straight, flat, it means that it creates more space for the cranium to accommodate more brain. But here, because here it is not, because he has to have too many muscles so that this is being supported. Therefore, the first answer is to create or to have more brain, which indicates brain cells, and then you link them to the intelligence. Also, when it is more pregnant like this, the position also keep on changing, the position of foramen magnum. You see, if it is straight, if it is flat straight, then the position of foramen magnum indicates like this. And if it is more protruding, then automatically this position is going to have to go a little bit back so that it brings about the shape. You see, it brings about the what? The shape. But here, the shape is straight. Therefore, it also allows the shift of the foramen magnum so that it can bring about bipedalism. Bipedalism, which means that you're walking on two hind limbs, and then this will create more faster. The organism can move more faster. So the significance here, you have to talk about the cranium becoming more bigger, creates more space for the cranium to be bigger, meaning that more brain cells. So the intelligence is linked here, while here is small. And then number two here is you talk about you can talk about it makes the position of foramen magnum to move a little bit forward, so that it can allow the organism to walk on two hind limbs, or to allow bipedalism to take place, so that now the organism can walk faster. So basically it brings about changing from the slope face to a flat face. Yeah, so that's what you are supposed to explain there. Guys, I hope you got. So guys, what about their deity? You're talking about, how about the deity? Yes, there are some things which I've not talked about, but you look at when you are changing from prognathism, meaning that even the jaw, the shape of the jaw will change. You understand? And if the shape of the jaw changes, because it is like this, let me show you in the book, the distinction material. You see here, ne? So the more it's checked, ne? So even this is gonna change. You see how it is slanting. When this thing is out, then automatically this one, you see. So the position is cool. So the position, we have seen it, because now it's not protruding out. So it means that the position is cool. You see now, when it is straight, it creates more space for the skull. You see? It creates more space for the skull to accommodate more brain. But here, because it is a little bit flat, so it means that the skull, the cranium becomes smaller. You understand? Yes. And then also, prognathism is basically you change. Remember, it's the jaw. This jaw, which could be also from big teeth. You can talk about the teeth becoming more smaller. So it means that you are shifting away from the rough, tough food to rough, raw food to one which is more cooked. So that, yeah, so you have to link. Your answer was supposed to be linked. You know, I told you yesterday that once it comes to this describe thing you don't know, but you think about something which is correct. Describe thing, you write everything, and I caught it that write all your rubbish. I call it all your rubbish. But in other words, I mean that write everything you think it is correct. When you're marking, we will look for what is correct in these things whereby they tell you that describe what. Yes. So you guys, thank you very much for attending our class. You have been good people. You have been supportive. And then I wish you the best. I wish you the best in your life. If you have sisters, brothers, and stuff, please refer them to this channel. If you have not subscribed, subscribe, like the video so that it can move and then others can also benefit. Yes, benefit. They can judge themselves so that they go for these holidays when they are happy. If you have not made it, just this is a simple advice. If you have not made it, guys, listen carefully. Not the best in class will be the best in the world. Is it clear? So it must not make you to be too much stressed. No, I don't think, I'm not insulting, but I'm just trying to show you this. Ne? I don't think Mr. Cyril Ramaphosa, our president, ne, was the best in class, but he's the president. So that's what I'm telling you, that not the best in class is the best in the world. You understand? So that must not influence you. You understand? Keep focused. I don't think Mr. Let me say Elon Musk, whatever, yes, was the best in class. I don't think so. You understand? So not the best, I repeat, not the best in class is the best in the world. Those are two different things. I repeat, those are two different things. However much we want us to be best, but please don't lose your hope. Yes, you should not lose your hope. Is it clear? Lastly, don't treat others bad because you are the best in class. Because you don't know that this person you don't treat right might be your boss. It might be your boss tomorrow when things say. Describe the structure of DNA. Structure of DNA. Guys, when talk about the structure of DNA, look at the characteristics of DNA. Ne? So I think that's done. The characteristic of DNA is double helix is made up of 2 strands. It's made up of the nucleotide. It's made up of nucleotide, yes, which are made up of phosphate, the oxyribose sugar, and nitrogenous bases. We talk about the nitrogenous bases. Talk about all of that. Ne? Yes. Challenge. Don't go to the paper without knowing DNA replication. Yes, don't go to the paper without knowing translation. Don't go to the paper without knowing transcription. And then describe the description of the structure of DNA. So one will come. I won't tell you which one, but one will come. You'll come. We're going to discuss the paper after. You'll know. And then now, if they ask you a question that give the difference between DNA and RNA, that one is simple. You can talk about everything you know. Eh? Make sure that you draw the table. Don't leave it hanging like this. You want a complete table. You mentioning this, you writing like this, and you say DNA, and the other side you say RNA. Guys, put DNA, put RNA, close the table. If you don't know anything, at least you got a mark. Ne? Yes. If they ask you to tabulate, draw a table, put the title, close the table, leave. If you don't know anything, yes. If you know something, then write something. Yes. I'm done with that. But now the challenge comes in when they ask you that give the difference between DNA nucleotide, nucleotide, and RNA nucleotide. Here, you only look at the nucleotide of DNA, the nucleotide of DNA and the nucleotide of RNA. And there are only 2 differences in there. DNA has, don't talk about the phosphate because it's the same. DNA has deoxyribose sugar and the RNA has ribose sugar. And then DNA has thymine, RNA has uracil. Done. If they ask you about what? About the nucleotide. Is it clear? If they ask you the difference between DNA replication and transcription, you have to know the differences. DNA and transcription. We're going to spend a few minutes here. I'm only going to focus on the things which you fail. Which you fail is what I'm going to focus on. Yes, too much. Then we are saying that DNA, you talk about DNA, in this case, in DNA replication, 2 strands act as a template. The other side, you talk about 1 strand transcription, 1 strand act as a template. Guys, only know at least 3, but we want 2. In most cases, we want 2. We won't ask you more than 2, the differences. Ne? Don't go to the paper. If there's a lot, just know few things. Ne? Don't on each, ne? At least go have a dose on each, whatever I'm going to talk about. Ne? Yes, then you'll be able to make it. Then we are saying that yes. Then another thing which you go off is about the mutations on DNA. About the mutations on DNA. So when they ask you about the mutation, let me just use my book here so that you understand. Excel in Life Science Distinction Material. Here, let me just, I don't want the structures, okay? I don't want the structures. What I want is this. What I want is at the end here. Yes, at the end, the mutations. Yes. Guys, let's answer this question. That what will happen if a mutation changes triplet number one? Triplet number one, it's this. From this, the same thing. Ne? From this to this, it means that this has been changed. Is it clear? You changed G to A. So now what you do when they ask you such a question, you first identify what is here. It is a codon. What is given here is DNA. Therefore, I have to convert this into messenger. Remember, we say that. Let me do like this. Yeah. Remember, we say that when you talk about code, it means DNA. DNA. When you talk about codon, it means messenger RNA. 3 bases on messenger RNA. When we talk about anticodon, we mean 3 bases on transfer RNA. So now the question is asking it means that it's changing from DNA to codon. It means that it's talking about messenger RNA. Is it clear? Yes. So now what you do is to change this into messenger RNA. So change this into messenger RNA. So when you change into messenger RNA, this is going to be A goes with U, T goes with A, G goes with what? G goes with the C. So I'm going to CUA. I come here, look for C, C, CUA, C, no, AUC, AUC, where is it? US is triosine. So this is triosine. UCA, isn't it? Then what about this? Now I change it again. Also is U. It's gonna be. It's gonna UAU. What does it mean? UAU is also triosine. So it means that a change from this to this, yes, will lead to this USC, from USC to UAU. That's the first mark. It will, you converting this into codons. You understand? Into codons. Is it clear? Which will code for the same amino acid, triosine. Therefore, there will be no change in the protein. Why? Because it's the same amino acid which is being coded for. Can I repeat it? I said, when they bring such a question, I know when they bring it, and I know it will be there. When they bring such a question, students lose these marks. Change in A, this, first find out, changing, because here it might be tRNA, and here is codon. So you change into the codon, which is messenger RNA. Is it clear? So change from ATG, basically result in this, and the change in this result into this. Is it clear? And these two, they code for the same amino acid. Therefore, it does not code for a different protein. It's like me calling you, let me check. Let me check here. It's like me calling you Lerato, Lo, Lailo, whatever. Ne? Sorry if I pronounced you wrongly. Ne? I can call you Lerato, or I can call you whatever, ne? The second name. I don't want to pronounce it badly. Ne? Yes. So if I call you the other way around, you say yes, sir. When I call you with another name, you also say yes, sir. You understand? So it means that they are coding for the same amino acid. However much the codes are different. Is it clear there? Yes. Then, what about base triplet number 4 from, yes, base triplet number 4 So this is the first instance. The second instance, what will happen to a protein if the base triplet number 4, ATA, is changed to ATT? So ATA, I have to convert it because the DNA, I need them to be in messenger RNA. ATA is going to be, T goes with A, U. And then this one is going to be U, U, U. So now I say that this will change into this. Okay, this will code for this and this will code for this. Cool. That's point number 1. But what does this code for? AAU. AAU is valine. What about UUU? UUU. UU, is it valine? No. Where is it? A. We have UUU. No, man. It's not UU. Yeah. What am I doing? It's AAA, ne? It's AAA because T goes with A, T goes with A. So it's AAA. So what am I looking for? Is the AA is proline. So it means that if you look here, this one code for valine. Valine. While this one code for proline. Proline. You see? So it means that these two, they are different. Is it clear? So it means that even the protein will change. It's like calling you Carabo, and then I call another, I call again Cabero. And Cabero is different from Carabo. You see? Yes. So it means that now this will code for this will code for this, of which this codes for valine, and then this, you write this in full, that AAU, AAA, which code for proline. Therefore, a different protein will be formed because of the different amino acid. That's the scenario number 2. Scenario number 3. Yes, one scenario will come. Explain what will happen to a protein if DNA base triplet, or if DNA base has changed. Yes. There, they have not talked about you have to interpret it automatically. If the DNA sequence has changed, one base has removed or has been replaced, whatever, it will lead to a different sequence of messenger, which will lead to a different sequence of transferrin A. Yes, which means a different sequence of amino acid. You understand? Hence a different protein. Different sequence of DNA will lead to a different sequence of messenger RNA, which leads to a different sequence of transferrin A with its amino acids, which leads to a different protein. That's a study scenario. The first scenario, it might not change the protein. Therefore, the protein is not going to change because the amino acids are being coded by different codes as we saw it. The second scenario, they can code for a different amino acid, meaning that valine and proline, for example, therefore a different protein. Or they can ask you what will happen if DNA base sequence changes or messenger RNA sequence changes. So you start from messenger RNA sequence. If it changes, it will lead to a different sequence of transferrin A with its amino acid, hence a different protein. Guys, don't go without knowing those things. Lastly, on DNA, it's this. Let me just use this example, but you still have questions, ne? You still have questions we can use. Please, don't go to the paper without knowing that. Ne? Someone is telling me T, they have given you T or C. Just in mind A and C. Yeah. So guys, make sure that you guys get the things right. Yes. If you have any challenge, let me know. Ne? Yeah. Lucy, Lucia. Yes, you say adenine John. Yeah, I made a small mistake, but yeah, I saw it when I was trying to. Yeah, I'm always human. I can make a mistake. Ne? Yeah, but when I do it, ne, sometimes when you talk, you can talk about something else, and then what in mind is something different. Ne? Yeah. All right, but I'm sober today. Ne? I'm sober. I don't drink and smoke. Ne? So I'm sober. All right. All right. Let's continue to the next. Guys, don't go without knowing how to label these things. What is this? It is amino acid. What is this? Is ribosome. What is this? What is this? It's transfer. You see, it is a T. Guys, check. It's a T. tRNA. If you don't know it, try to see if that T will come up. Check. You see? It comes out. It's a T. So it's a T transferrin. Guys, this is not a protein. It's a polypeptide chain. Protein is 50 amino acid and above. Is it clear? So this is DNA molecule, this is messenger RNA, this is nuclear pore, this is nuclear membrane. Please go when you know how to label these simple things. Is it clear? Yes. This is the transcription. It occurs in the nucleus. This is translation. It occurs in the cytoplasm. Therefore, if they ask you what is this space, is cytoplasm, and what is this space, is the nucleus. Ne? Yes. Lastly, but not the least on DNA, so that we go to meiosis. Two things on here. The first thing is if DNA is 30%, if adenine is 30%, what is the ratio of guanine to cytosine? What's the ratio of guanine to thymine? Let me just say like that. Guys, you have to know how to do the base calculation. A goes with the T, C goes with the G. If this is 30%, automatically also this is 30%. So 30 plus 30 gives me 60. So remember, this is percentage, né? Percentage means 100. So 100 minus 60, it gives me 40%. So this 40% is for these two. So divide by 2, so it's going to be 20, it's going to be 20. That's not the answer. They're saying the ratio. So it's going to be G to T, whereby G, where's G? G is 20, and then T is 30. So I divide by 10, I divide by 10, so I get 2 here, and then I get 3 here. You know what we're going to do? The first objective we're going to do, we're going to say that 3 to 2. Because when you saw 3 and 2, you just say that it's A. And the last one is going to be 2 to 3, because you don't want to read everything. Make sure that you read everything, because there is most correct answer. I remember in paper 1, whereby they gave a correct answer halfway correct, and there is the most correct answer just down. You understand? We always try to trick you to see that, yeah, you are off. But I know since you are here, you won't be off. Ne? Yeah, we are here to kill the paper, not the paper to kill us. Ne? Yeah, tomorrow we will make it. We will make it. We'll make it. I know. All right. Yeah, if do you have any question, let me know. Let me know if you have any question. Let me know so that I smash that question without. That's number one. That's number one. The last thing which I need to talk about is about profiling. Is profiling. Someone might say that, profiling is easy, man. Yeah, I know that profiling is easy, man. But there is a time when they are talking about comparing the paternity test. Ne? Let me see if this question is about paternity test. Identifying the criminal. What is this? Okay. Sample, DNA sample. Okay, let me just read it. Detective investigating the scene of broken window, suspected blood was found in the criminal identified. No, it won't give me what I want. Blood. And it won't give me what I want. Okay, now let me design my question so that now you understand what I'm saying. If they say that two guys, they are crying, or they are saying that, okay, you have a boy, and then you have two guys, ne? And these guys, they are saying that it's each is saying that is the father of this boy. But now the mother doesn't know who, because this mother slept with these guys, ne? These guys are the same day, ne? You understand? So now we don't know who is the father of this boy. So what you need to do is, if this is the DNA profile of the boy, guys, listen until we end this, ne? Because sometimes this question, when it comes, you are off, ne? Okay, if this is the profile of the child, yes, if the profile of the child, I know DNA profile is very easy, but once it comes to this, you lose the marks. Yes, you lose the marks. Okay, this is the boy, ne? And then now you have the profile of the mother. You have the what? The profile of the what? Of the mother. You have the profile of the mother. Is it clear? Yes, you have the profile of the what? Of the mother there. And then now you have the first profile of the guy. Well, the first profile of the guy. You have the first profile of the first guy. You have the first profile of the first guy. Ne? Have the first profile of the first guy. Yes. And then you have the second profile of the second guy. You have the first, you have another profile of the second guy. You have another profile of the second guy. Yes. Yeah. So now they ask you, this is the mother, this is X, this is Y. Is it clear? Who is the father of this guy? And after that, they can ask you to explain, and they say 4 marks. Explain 4 marks. That's it. That's what I'm saying, that's where students go off when it comes to this. Listen carefully what you're supposed to say. Why did I divide with the 10 so that I get the simplest ratio? Ne? Yes, I divided by 10 so that I can get the simplest ratio. Yes. To simplify the ratio, yeah. Wholly. Yes, correct. What you said is correct. I divided by 10 so that I can get the simplest ratio. Equilibrium, I'll reach there, don't worry. So, guys, look here critically. What you need to do here is to first, because I know the mother. So take all the bars which correspond with the mother. So this corresponds with the mother. Yes. This bar corresponds with the mother. Yes. This bar corresponds with the mother. So it means that, let me just use a different color so that we see exactly. So this bar is out. This bar is out. This bar is out. So now the remaining bars, not that one. I'm saying this bar is out with the mother, this bar out with the mother, and then this bar is out with the mother. So the remaining bars, these remaining bars, this, and this, it must correspond with one of the guy. If there is one bar which is not corresponding, then that's not a father. Is it clear? Yes. Now I come here. I look for this. What does this one correspond with? This. If there is no one who is corresponding with it, then it's out. You understand? Then is what? Is out. So this bar correspond with the, with this, ne? Yes. So it corresponds with that. This one is a little bit down. And then this bar corresponds with that. This bar corresponds with this and this. So however much this one has one which is corresponding, but this one and this one did not match with it. So the remaining bars, you see that all the remaining bars, yes, have corresponded with this guy X. Therefore, guy X is the father. Explain your answer. Why are you saying that X is the father? X is the father because the bars of the boy correspond, the remaining bars which of the boy which do not correspond with the mother correspond with this guy X. All the remaining, the word all must be there. All the remaining bars of the boy, yes, after matching them with the mother, correspond with guy X. Is it clear? Because they always ask you this. Yes. And then students tend to go off. These bars must correspond with the mother. The remaining bars must correspond with the father. If there's one bar which is not corresponding, we will ask you, where did that boy get that gene? Understand? Remember, we get these chromosomes as a sperm and ovum. Where did that boy get that gene from? Are trying to mean that's a mutation? So that's why all of them must correspond with the father, so that we conclude that that gentleman X is the father of that boy. Yeah, guys, unless you have any question concerning about describe the process of translation according to the messenger RNA, the transfer RNA with the complementary anticodon match according to the messenger RNA, the transfer RNA with the complementary anticodon, yes, bring the required amino acid to the ribosome. The amino acid is joined together by a peptide bond to form a required protein. Guys, those are just processes I'm not going to go through. Maybe I can ask you a question. What is DNA replication? When does it occur? When does it occur? When does it occur? It occurs during before the cell divides, during interphase. Where does it occur in the nucleus? Why does it occur to double the chromosome number? Yes. How does it occur? That is the process of DNA replication. So there are so many questions. I'm not going to go through them, but make sure that you know those processes. Unless you have a question about DNA replication, about DNA code of life, let's go to meiosis. Yes, when you talk about meiosis, guys, there are some things which you need to talk about. See, this is a transfer. Okay? It makes it. You see, this is amino acid. This is transfer. We said it. This is a ribosome. This is messenger RNA. Sometimes it's up, sometimes it's down. This is what? Codons. Hey guys, how many? This question. Two questions. The first one is how many codons in 12 bases. So remember, 3 bases is equal to 1 codon. So it means that 12 divided by 3, so makes them to be 4 codons. How many bases, how many amino acids in the 4 codons? It's gonna be 4 because each codon code for 1 amino acid. Understand? How many bases code for 5 amino acid? Remember, each amino acid is coded by 3 bases. So I'm gonna say 5 times 3, which gives me 15. You understand? So you have to know how to interpret that. And then, let's go to meiosis. Yeah, we try to explain this. They say left, right. Make sure that you translate it left, right. Ne? Yes. The first thing you need to know about meiosis are the phases. But I'm not going to go through the phases. Use IPMAT, you'll be able to know which phase is it. But guys, something is itching me. And that itch makes people to lose marks in meiosis. Number 1, identify phase in diagram 2, diagram 1, diagram 3, and diagram 2. Guys, whenever they ask you about the phase, please turn your paper if these centromere, the centrioles, they are not on the sides. So turn your paper to be like, yes, you see, it must be, I don't know, I think this is east and this is west. If I'm not a geographer, I hope this is the correct. Yeah, but it must move east to west, It must not leave what? East to west. Don't leave these, they must be in east to west so that you identify the perfect what. If it's not straight with a paper, turn it. Because if you turn it, you will be able to identify the actual what? The actual phase. So in this case, because there is space here, it means that is anaphase. But because it is single, therefore is gonna be anaphase 2. So it's anaphase 2. What about here? The chromosome, you see there in the equator. So it's gonna be metaphase 1. Yes, because the chromosomes are double, you see, therefore it's gonna be metaphase 1. If they're single, then it's gonna be meiosis 2, which is you identify the phase and then you put the word 2. That is the first thing which you need to know before you go for the paper. The second thing which you need to know about before I tell you what to focus more on this is the number of chromosomes. How many chromosomes are found in diagram 1? You will find out someone saying 46. Did they say this is a human cell? No. If they don't talk about human cell, don't talk about 46, don't talk about 23, because they didn't talk about human cell. How many do you see? 1, 2, 3, 4. It's 4. How many chromatids found in diagram 1? It's 1, 2, 3, 4, 5, 6, 7, 8. It's 8. Chromatids they're talking about. How many chromosomes will be found at the end of division of diagram 2? Yes, because this is meiosis 1, therefore it's gonna be, since there are 4, therefore it's gonna be 2. Yes, that's the number of chromosomes which will be found at the end of meiosis 1. Now the question comes, how many chromosomes will be found in diagram 2 if this was a human cell? Now they said if this was a human cell, because this is meiosis 1 human cell, therefore there will be 46 or there will be 23 pairs. Yes, I think I'm clear there. That is point number 1 about meiosis. Point number 2 is about the karyotype. Point number 2 is about the karyotype. This is where also you lose marks. Describe the structure, describe the karyotype. It's fine. You can describe that from this karyotype is for a male. If you see this is for a male, this is for a female. Because here the chromosome number 23, they have the same size and shape. While this one is big, one is small. Yes, therefore it means one is X, one is Y. But now, if they ask you to describe the karyotype, from is made up of 46 chromosomes, which are arranged in pairs. From pair number 1 to pair number 22, we call them autosomes. The 23rd pair is called the gonosome. The gonosome has X and Y. If it is a male, has X and X. If it is a female, the X is longer than Y. So you describe what you see. But if they ask you how many autosomes are found in P, please guys, don't just say that is 23. Sorry, don't just say that is 40, 44 or is 22 pairs. No, count them, because sometimes maybe chromosome number 6 we put them 3, whereby you say 1 2 3 4 and you don't see that, you don't recognize. Look at chromosome number 21, but it's better to count them 1 2 3 4 5, because you'll find out you won't just ask you a question, how many chromosomes are there? No man, they will have to hide something. There must be a reason why they ask you that. Yes, there must be a reason why they ask you that. Maybe there is a chromosome which they have they have done the hiding, and then when now you have to identify where they have hidden that. Guys, give it or don't panic. Take your time, read the question 3 times exactly. If you feel like you don't understand, skip it. Yes, why write what? Start, if you see that things are not working out, start with what you think is right. I'm telling you, don't panic. Don't panic because you have not seen the diagram, Yeah, the diagram is known. This time they have brought it in terms of what? So why are you skipping? Skipping what? I'm not skipping anything. Yeah, I'm going to come back. I'm trying to talk about the things. After that, I come back to those questions. Yes. So guys, make sure that you concentrate on those karyotype. Make sure that you know how to identify them. Give a reason. Give one observable reason why. State the gender of P. P. Where is P? This one. We say that this P is what? Is a what? Is a male. Why? Because the chromosome number, the pair number 23, it has 2 different chromosomes. One is X, one is Y, or one is big, one is small, meaning that one is X, one is Y. Yes, that's number 2. That's where people fail. Number 3, non-disjunction. Non-disjunction. You see this? This is a non-disjunction question. Yes. Whereby when they ask you to which phase is it? Is Anaphase. It could be Anaphase 1 or Anaphase 2. Yes. But looking at the diagram, you can identify that it's Anaphase 1 or Anaphase 2. Yes. Describe Down syndrome. How is it? How is non-disjunction lead to Down syndrome? Guys, you just talk about what is extra. That Down syndrome is as a result of non-disjunction, leading to one cell having an extra chromosome. Yes, on chromosome number 21. Yes, meaning that it's gonna have 24 chromosomes instead of 23. Yes. When this cell fertilizes a normal ovum, the resultant individual is going to have 47 chromosomes instead of 46, leading to Down syndrome. Is it clear? Down syndrome is caused by non-disjunction. Is it clear? Either in Anaphase, it could be Anaphase 1 or Anaphase 2, leading to a cell having an extra chromosome on chromosome number 21. In brackets, you can say 24 chromosomes instead of 23. Then, when this gamete, yes, fuses a normal gamete, meaning that it is having 23, it will result in 46 chromosome, a zygote with the 46 chromosome, instead, no, 47 chromosome instead of 46, leading to Down's syndrome. It's simple. I don't know why people make it difficult and complex and what. No, man, it's simple. Is it clear? Yes. So that is another thing where you lose marks. Number 3, give two, let me check here. I hope that question is here. Metaphase 1 and Anaphase 1. Those ones you can do. Okay. Describe 2 ways in which, this is Down syndrome. Yes. So we have answered that one. Yes. Name the organ in male that where meiosis occur. Okay. Meiosis occurs in the testis for the case of male, and then female occurs in the ovary to produce ovum. Yes, but there is something which I want where students fail. Give 2 causes of variation in meiosis. In meiosis, guys, they are saying in meiosis. In meiosis, they're not talking about the causes of variation. No, they're saying about in meiosis. So what are you supposed to talk about here? You talk about the the random arrangement of chromosomes during metaphase. Yes, random arrangement of chromosomes during metaphase, and also talking about crossing over during prophase 1. Remember, when you talk about prophase, crossing over, you are specific to prophase 1. Prophase 1, not anything else, is prophase 1. You don't talk about something else. It's supposed to be prophase 1. That is crossing over during prophase 1, which leads to exchange of the genetic material. So please don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing anaphase. Don't go to the paper without knowing metaphase. Yes, metaphase 1, metaphase 2, anaphase 1, anaphase 2. Those we might likely to bring it. Division of the cytoplasm, what does it mean? Is called cytokinesis. This division of a cytoplasm is called cytokinesis. Lastly, on meiosis, what you can't go without knowing is know how to label. What is this? In most cases, people, they call this nuclear membrane. No, nuclear membrane cannot be seen. You see, the nuclear membrane can only be seen in telophase. Yes, yes, in telophase. And in prophase, early prophase. So this is cell membrane. This is chromatid. This is centriole. The dot is centromere. This is spindle fiber. Ne? This is, what is this? This is a cytoplasm. So now I think you know. The last question whereby students get lost is draw the diagram of a cell at the end of meiosis, or draw the diagram of a cell after crossing over. Let me just show you. Let me just show you using this. Using this, I think I have the diagram here. Give the collective name, give the gender, name of observable reason, the main result of segregation, describe the character of Down syndrome will result differ. Okay, sure. That's not what I want. Yes, here. Look at that diagram. Look at that diagram, guys. If they ask you to draw the cell or this after this process, guys, look at where crossing over took place. In this case, the crossing over took place up, while here is took place here and here. So if they ask you to draw this cell, maybe they are saying chromosome X and chromosome Y, whereby you say structure of chromosome Y after maybe crossing over, then you have to draw it nicely. You have to draw it nicely. Yes, you have to draw it nicely. But where did the crossing over take place? Crossing over took place on this chromatid, but halfway up. So you mean that when I'm shading all this, remember this is the black thing, so I have to shade all this. I have to shade all this. Yes, I have to shade all this to be black. And then the crossing over did not start from here, it started from here. So this variation of shading will give you a mark. Writing a caption will give you a mark. And then don't forget to label. Even if they have not asked you to label, label. Even if you don't ask you to label, at least have 2. Because in most cases, when they ask you to label, at least 2 points. Yes, they say at least 2 points. You have labeled it at least 2 points. At least then you get those marks. Yeah, please don't forget to label. If they don't say If they say draw a label diagram, or they'll say draw and then just leave it like that. Guys, make sure that you draw and label, because it's not losing you. It's not taking, they won't cross it because it's labeled. But you get a mark if you label, just in case, because you are not a memorandum to know which one is not going to be credited or is going to be credited. So you just write. Lastly, before I forget, if they ask you to describe, write everything you know in the head. At least for us, we look for what is correct for you. If they say name, that one you only name. You don't give alternatives. But if they say describe, just write everything you know. Write everything you know. Or sometimes I call it write all the rubbish. I'm not saying that write the rubbish, but write everything, whatever you have in the head. Don't leave it, because when you leave it, you come out of the paper and say, I thought about it. I thought about it. I thought about it. No, don't do the regrets, because this is your last time to change this subject, the mark. So just write everything. If the correct answer is there, we look for it and we'll give you a tick. Yes. Yeah. Do you have any question, guys? Comparative anatomy. Someone saying comparative anatomy. Okay. Thank you. Guys, if you have any question, you let me know, because I'm here to make sure that you get a distinction. You get a what? A distinction. Distinction. Yeah. Make sure that the distinction clicks, because we want it and we will get it. We will get it. Yes. God willing, you know me, when in my region we say, Inshallah. Inshallah, we will get a distinction. God willing, we will get a distinction. That's what it means. All right. Check this. Check this. It's simple. You guys, Meiosis, I'm winding up now. Make sure that you know how to label those diagrams. Make sure that you go when you know the process of crossing over. Make sure that you know the karyotype. Make sure that you know the prophase, anaphase, telophase. Make sure that you know when you go there, when they give you a diagram, it must be east to west. Those spindle fibers, they must be east to west, meaning that you have to turn the paper. Turn the paper. No one is going to ask you, why are you turning the paper? It's your paper. No one is going to ask you whether you turn it, whether you write upside down. Doesn't matter. Turn it so that you identify the things correctly. You understand? Yeah, these phases, they are confusing people to identify. Please don't make a mistake. It's the last time for you to identify what is it. Let's try to label this and then you see. How do you call C? Is the centromere. How do you call this? The point where crossing over takes place is chiasma. How do you call this? We call it chromatid. How do you call this? Because they are chromosomes and crossing over, we call it bivalent. Yes, we call it a bivalent or bivalent. Yes. Or you can call them, sometimes you can call them, they can correct you when you say homologous chromosomes. Yeah, but scientifically we call it a bivalent. Bivalent, or bivalent. Someone might ask me the spelling. Yes. So identify, okay, we have done that. Name two ways in which structure A and B is similar. Structure A and B. How are they similar? How are these two structures similar? They have the same size, same size and shape. Cool. Number two, they have the same location of the chromosomes. Not same chromosome. They are not the same chromosome. If they're same chromosome, you'd have looked exactly the same as your father. Yes. So they have the same position, locus, same position of the genes. They have the same position of the genes. Describe the process at D. So they are looking at describe the process of crossing over 5 marks. The homologous chromosome come together. They cross over at a point called chiasma. They exchange the genetic material between two homologous chromatids, and then they separate, and then they form genetically different chromosomes. So basically, that's what you need to explain when it comes to that. I'm not going to use too much of the distinction material, but I know that you guys, you will make it. Lastly, before I go, where does meiosis occur in male? It occurs in the ovary to form ovum, testis to form spermatozoa, then in plants, ovary to form ovules, anthers to form what? Grain. That's it. Hey guys, I feel good when you guys are feeling good. Ne? Don't panic, ne? Because when you panic, you make me panic. Ne? Yes. So make sure that you don't panic. Ne? Yes. Make sure I repeat. Don't panic. You're gonna make it. Let's go to genetics. Genetics. What are you supposed to talk about? Genetics. Genetics. So we have used 1 full hour. Remember, we said that we're gonna discuss this paper for 2 hours. Eh? Yes. So we have used 1 full hour. So let's go to the second hour, that is genetics, and 30 minutes, and then 30 minutes for evolution. And then we know that we have smashed. Guys, make sure that you get all the marks in DNA, all the marks in meiosis. Yes. Guys, in genetics, we'll talk about genetics. Terminologies, you know, I'm not going to talk about the terminologies. But go when you know that a circle means a female, and then a box means a male. Is it clear? That's number 1. Number 2, when they give you a question, identify whether it's a sex linked or a non-sex linked. I repeat, identify whether it's a sex linked or a non-sex linked. How do I differentiate between the two? Sex linked, they will mention it that is sex linked disorder. Always read what is on top here. Is a sex linked disorder. Is it clear? Make sure that you read it. If they say sex linked disorder, definitely you have to use X and Y. Male, there will be X and Y. Female, there will be X and X. If it is not a sex-linked disorder, please don't use X and Y. However much they have talked about same and carabo, and you know that this is a male, this is a female. Please don't use X and Y if they have not mentioned that this is a sex-linked disorder. I think I'm clear there. Number 2. Let's do one example on sex-linked recessive allele. One example, sex-linked dominant allele. One example, non-sex-linked. The first one, identify whether it's a sex-linked or non-sex-linked. Okay, now we have identified here that is sex-linked. The second thing you need to know, guys, I repeat, and I'm putting down the pen, the mouse. I wish you could see me the way I'm begging. Please, let me come closer to the mic. Please identify whether is caused by a dominant allele or a recessive allele. I repeat, identify whether it's caused by a dominant allele or recessive allele. Because if it is caused by a recessive allele, the one who is homozygous recessive is the one who is going to be affected. If it is caused by a dominant allele, the one who is homozygous recessive is going to be normal. Let me show you using here. This diagram saying. So it means that it's caused by a recessive allele. Ne? They said it here. Ne? So it means that the one who is shaded, they're saying that is affected with the disease. Therefore, this person is a male because the sex linked is going to be that way. While this is a female, is going to be small d, small d. Is it clear? This one is a male, is going to be capital D, Y. Okay. Question. What about if it's caused by a dominant allele? Perfect. This is caused by a dominant allele. Yes. Okay, let me first finish here so that I don't confuse you. Now, let me go to the questions and see how many generations are shown here. It's 1, it's 2, it's 3. So there are 3 generations. How many kids listen carefully. How many kids are sons of 1 and 2? How many kids are sons of 1 and 2? 0. How many kids are 1 and 2? 2. Remember that this is not a kid of this. This is just came and married this daughter. And this one came and married that daughter. Therefore, there are how many? there are two kids which are coming from these two. Is it clear? You see that this thing is not linking down. Is it clear? Yes. I'm done with that. Let's go to the third question. How many family members are in this phylogenetic tree? Family members. It's 1, it's 2, 3, 4, 5, 6, 7, 8, 9, 10. They are saying family members. Is it clear? They're saying family members. Don't say that this is not a family member, unless you're telling me that your father is not among the family. It's a family member. But if they say how many kids for this, then I think now it's clear you know what you're supposed to do. Yes. That's the third thing. If they say that this cross with this, or this kids, do a genetic cross, because we are in the second generation, so it's gonna be P2F2. Is it clear? If they say this, it's gonna be P1F1. I think I'm clear there. Yes. So I will talk about when I go to the crosses. Then we go to the next, what you're supposed to know. Yes. Can you please do a live letter? Today I join. Can you do the live again later today? I join. No, what you can do, if the live is, start from where you have started from, and then after that, join, you will play the video, ne? Yes, before. Lindo, Putle, why are you removing? I didn't see what you are saying, sir. The sound at the back. Hey. Someone saying there's a sound at the back. Serious, there's a sound at the back. Okay, let's go to the next question. Okay, how do I find the unknown one? I'm gonna talk about it, my daughter. Yeah, guys, I hope the sound at the back is removed. Let me know. Let me know if the echo sound is removed. I don't want those echo sounds. Yeah, it must be nice. All right, let's go to the next question. How do I identify? Okay, now someone is asking me. I'm gonna go there so that we finish this. How do I identify one? To identify one, remember you have to first identify that this is caused by a recessive allele. Yes, if it's caused by a recessive allele, so what you need to do is okay, what you need to do is, okay, this one is XX, but this person is normal. Therefore, it's going to be capital D. I don't know whether it's going to be a capital D, small d, or capital D, capital D. But they produced a kid who is sick. Therefore, this kid is sick with the, don't just say sick with what? With the disease, ne? So automatically, one came from the father, one came from the mother. Therefore, this mother cannot be like this because it gave rise to this. You see? Whenever they ask you to identify, make sure that you look at the kids, ne? Yes, which kind of kids they have produced. Therefore, we have identified that because this kid is small d, therefore one came from the mother. So therefore the mother is like this. Né? Now what about the father? Remember this is sex-linked disorder. Yes, if one came from the mother, then definitely another one came from the father. Therefore the second X, the first X is like this, or the X is like that. Therefore the father is affected with that disorder. Don't just say affected. Affected with the what? HIV, sickle cell, flu, corona. Is affected with what? With the condition they have talked about. Don't leave the answer hanging. Please, 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 please. Don't leave the answer hanging. Then the second one. Okay, someone saying, ish. Hey guys, just give me 2 minutes. Let me check why is it. I hope the sound is gone. I don't know whether. Okay. I hope the sound is gone. Yeah, you let me know. Please slow down. I hope. It's okay now. All right, let's continue, guys. Let me know if the sound is fine. I don't know because I've tried to see that everything is right. I've tried to check. Let me check here. I've tried to see that. Yes. Let's continue and see. Hey guys, I think that sound, I don't know, because we changed the system and the system I'm using is a different one from the main one. Yes. So I had to change the stations. All right, let me just continue. I hope at least you get something. Yes. Okay. I think now you have got it correctly. How do we identify this? Yes. Now let's go to the question, the next question. Then we are finding out. Identify the phenotype of individual 2. These ones I'm going to do the questions and then we see. The phenotype of individual 2. So we have said that this person is normal. Yes, is normal. A normal male or female is a normal female, or female without C. Yeah, that disorder. Then the phenotype of 1, we have identified that this person, because they have produced a kid who is sick and the sick is like this, therefore one recessive allele came from here, another recessive allele came from here. And remember, this is a male. This is a male. Therefore, this individual is affected with seed. And then they're saying that identify the genotype of number 3. Genotype of number 3, yes. remember this person is sick, né? Therefore is going to give remember is a female, né? Is going to give one chromosome from there, but because it's normal, therefore the mother gave her this, therefore is going to be that way because they say the genotype. They say explain how individual 5 inherited disorder. Individual 5 is small d. Therefore, we're going to say that individual 5 got one recessive allele from the mother and then one recessive allele from the father. No. Individual 5 is a male, ne? Is a male. Can't be from the father. Father is normal. The individual is male. So you're gonna say that the individual 5 is a male who is sick, and then therefore obtain a recessive allele from the mother. Where is these 2 marks? You have to tell us that individual 5 is affected with this disorder and is received one recessive allele from the mother, who is number 4. Therefore, I think that's how you can try to explain. But now let's go to A. Yes, the problem comes in when it is caused by a dominant allele. Okay, let's look at it. When it's caused by a dominant allele, Putti, whatever, is syndrome sex-linked. You see, they have talked about it, but it's caused by a dominant allele, isn't it? The diagram below shows this syndrome in the family. Okay, now we are stating how many generations are there? There are 3. 1, 2, 3. Sharp. How many females are in this phylogenetic tree or in this family? Females 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. Is it clear? How many females were produced by Patty and Gabby? It's gonna be females 1, 2. This one is not family member, not a kid of this. It's a family member, but it's not a kid of this. So it's only these ones who are the kids. I think I'm clear there. Then now we go to the next question, whereby they're asking you. They're asking you. They're saying that okay, name the all right, let's find out. Remember now they're saying that this is affected, né? And then this is also affected, this is unaffected. But don't forget that this syndrome is caused by a dominant allele. Therefore, these people who are affected, they're going to be dominant. Capital D. Can it be capital D, or capital D? Sorry. X, capital D, X. Is it capital D? No, they used G. Sorry, guys. They used G. Please use the letter they have given you. Don't just use what you think is correct. Therefore, it's going to be X, capital G, X, capital G, X, small g, no, capital G, and X, small g. Yes. But now I need to find out, what about this one? This one is normal. So if it's normal, therefore it's going to be small g. It's going to be small g. Therefore, this person can't be this because this person, the kid, has a small g. Therefore, came from here. Therefore, I have to say that this individual is like this. Capital, small g. No. Capital G, small g. So that one is done. What about the female? The male. Remember, this is normal. If it's normal, therefore automatically is having small g. That's why this and this, they combine to produce a kid who is a female with the 2 recessive alleles. We are done with this. What about this? Is a female who is sick. Therefore, this female can't be homozygous dominant. It's going to be capital G and then small g. Why? Because one X came from here, then another G came from there. You understand? Yes. Now, how many, name the type of diagram is phylogenetic tree. They're saying how many female in this family? We talked about them. 1, 2, 3, 4, 5, 6. Male in this generation have how many male in this generation? Generation 1 have what is syndrome. Generation 1. Where is the generation? The first generation. The first generation. Remember, this is a generation 3. 1, 2, 3. Because they are looking for male in generation 1. So this 1, 2, 3. They're talking about generation 1. They're saying with the disease. 0. Answer is 0. Because it's caused by a dominant allele. And then they're saying that give the male in generation 1 have what is disease? Give the genotype of Gabi. Genotype of Gabi. We talked about it because they produce a kid who is not sick. Guys, please take note which one is dominant, caused by a dominant allele, which one is caused by a recessive allele. They will have to specify that is caused by a dominant allele. Is it clear? Males in okay, we're done with that. Aju and Pirusa have another 4th child. Give a phenotype of their sons. And Pirusa. Yes, so of their sons. Remember this is caused by a dominant allele. The moment it's caused by a dominant allele is easier to find. For the case of what? A son. And then remember this one, son. Ne? It's a Y, it's a Y. Therefore, because this one is normal, remember is recessive, is normal, therefore this one is going to be, both of them are going to be normal. Don't forget it's caused by a dominant allele. If it was a recessive allele, then they would have been affected. But the one who has the recessive allele is the one who is normal. And the one who has a dominant allele is the one who is affected with that disease. Is it clear? Yes, that is the only trick you need to know about that. The one is normal. Yes, the one who is normal is having a recessive allele for the dominant allele, if it's caused by a dominant allele. The one who is sick is the one who is having a dominant allele. You understand? Yes. Explain your answer above. Why? Because they are normal. Because Anju, who is a female, is normal with the 2 recessive alleles. Therefore, it gives 1 recessive allele to the males. You understand? Yes. Therefore, they are normal. So basically, that's what you need to talk about. When you go to a cross which involves an autosome, a non-sex linked, ne? Yes. Please, even if they talk about same and carao, please don't use X and Y, ne? Yeah. If they say that this diosa is caused by allele R, so you just say R. Ne? It is homozygous. If it's heterozygous, is R, small r. You don't put them on X and Y. That is the major difference between the two. Yes. Okay, lastly, about blood group. Then after that, we talk about the cloning. How many blood groups, how many genes control blood group? How many genes control blood group? How many genes control blood group? Someone is asking me, sir, it started confusing you. Daily interesting videos. How am I confusing you? Let me know. Let me know. Huh? How am I confusing you? Oh, where are you confused? Yeah, in this video description. You're saying I'm confusing. Let me know where am I confusing you so that I can sort that out. Ne? Yeah, just tell me that maybe this question or this question. Yeah, so that I can sort it out. How many genes? Yeah, correct. Correct, guys. Correct. Thank you very much. Yes. How many genes control blood group? It's 1. It means that you guys watch my videos. Thank you very much. And I'm seeing live as Mbali, as a member. Yeah. Thank you very much, Mbali. Yes, for being a member. And I saw another member here coming. Oh, Lindy Wayne Love. Thank you very much also for joining us. So you are on the team, ne? Yes. All right. Never be saying that X and XRR. Okay. Let me explain to you what I meant there. Someone is saying that X and XR. I'm saying don't use X and okay. Let me just look for a question here. Yes, here is a question. Ne? Mbali. I think it's Mbali. Ne? Yeah. Here they're saying that this syndrome in dog is caused by a dominant allele, capital H. You see? The pedigree diagram below shows the offspring produced by 2 dogs. You understand? They're not saying that this is a sex link. You understand? Yes. How many offsprings are affected by this disease? How many offsprings? You see now? Is caused by now this one is also caused by a dominant allele. Is caused, meaning that the one who is sick, né? Check here. Even here they are saying it that dogs with the disease and dogs without the disease. So these ones which have the disease, it means that they are going to be capital H, or can be capital H, small h. Even this one is going to be capital H. You see, is a male, but I'm not using X and Y. Capital H, or capital H, small h. Whenever there is a capital H, because it's caused by a dominant allele, this individual is going to be sick. You understand? Yes. So, however much is a male, I'm not using X and Y. You understand? I don't use that. Why? Because they didn't say it is caused by a sex link. Is it clear? Yes. So in this case, but I can identify because they say that how many individuals are affected. Okay. That one is simple. But now this one is what? Is not affected. Meaning that this individual is going to be small h, because it's caused by a dominant allele. Whenever you see a capital H, it means that that individual is what? Is sick. If they produce a kid here who is having these two, then automatically one came from the father and one came from the mother. Therefore, they can't be this, they can't be this. So this is like this, so that one H comes here, and then this one is like this, so that another H comes there. You understand? So when they give you a question and they don't talk about sex linked, please do not use, I repeat, do not use X and Y. Use the letters the way they have given them to you. Yes? You only use X and Y if the statement says that is caused by a recessive allele is sex linked. You see? Is sex linked. Even here they said it here is sex linked. Even they gave you what to use that is carried on X chromosome. Is it clear? Yes. So this situation is caused by a dominant allele, but it can be caused by a recessive allele. So I think now I'm clear when it comes to that difference between phylogenetic tree and the pedigree. Phylogenetic tree, guys, phylogenetic tree is explaining the evolutionary relationship among the members. You understand? Evolutionary relationship, like how did you evolve? But a pedigree is describing a character in a family. Is it clear? Is describing a character in a family. Yes. Okay, guys, let's go back to blood group. I think I've got it. You have got it. I'm no longer confusing you. How many genes control blood group, guys? Is one gene. Is one gene. How many genes control blood group? Is one gene. How many alleles control blood group? One gene, and that gene is blood group. Do you have any other gene? Tell me if there's any other gene. It's one gene, blood group. Yes. But how many alleles? There are 3 alleles. Yes, that is allele for A, allele for B, and allele for O. Guys, please don't make a mistake and you say allele for A like this. This is not A. This is a Roman numeral, which means 1. Allele for B is supposed to be like this. Allele for A. Don't put at here. Don't put them, make italics. Don't say allele for O like that. What is this? We will cross it. I is supposed to be like that. Is it clear? Yes. So allele for A, allele for B, and allele for O. So there are 3 alleles. Yes. I don't want to go to the distinction material to show everything. No. We go to the next part, of which the next part they're asking you how many genotypes. Genotypes. Oh, before I go, guys, back, there's something I forgot. Before I go back, I don't know what took me off. Why are males more affected by sex-linked disorders than females? Why? Or they can say, why are males more affected by hemophilia than females? Or they can ask, why are males affected by color blindness than females? So the answer you give here, if they say 4 marks, you say that males have one X. Yes. If a recessive allele, for example, is capital small h, appears on X, yes, the individual is affected with the disease, whatever they have talked about. While females, they have 2X. Yes. If a recessive allele appears, for example, on 1X, it can be masked by a dominant allele on another X. Therefore, the individual is normal. Males have 1X. Yes. If a recessive allele appears on the X, the individual is affected with that disorder they are talking about. While females they have 2X. If a recessive allele appears on 1X, it can be masked by a dominant allele on another X, and the individual is normal. Sometimes they can ask you a question that why males have higher chances of becoming hemophilic than females. So guys, it's the same thing. Please, you need to know about that. Ne? Yes, you need to know about that. We go to the next question. Because they have 1X. Yes, because they have 1X. This question they can come for 3 marks. Ne? Yes. Is it possible for a person of blood group A marrying a person of blood group B and they produce a child of blood group O? Is it possible? Yes, it is possible. How do I know it's possible? You look at the genetics. Ne? Okay, here the question is A. Before even I go there, let me just answer this question, at least one question, so that, yeah. A man who is blood group AB and then who is heterozygous marry blood group B and then have a children. How many alleles control blood group? They're now talking about the alleles. Yes. Gene 1 is 1. Allele, there are 3. Genotype, there are 6. Phenotype, there are 4. Why am I using 4? You have A, B, O, and AB. A, B, A, B. Do you understand what I'm saying? A, comma, B, comma, A, B, comma, and O. Those are the 4 phenotypes. But genotypes, there are 6 because A is homozygous or heterozygous. Let me show you. Let me show you in the distinction material, because some people, in just talking, they might not take it, and they might take it long to understand my ideas. See, you see, here is what I'm saying in the distinction material. Blood group are as a result of multiple alleles. Ne? It's a multiple allele. Yes, meaning that it has more alleles, more than 2. There are 3 or more alternative form of yes, this. There are 3. Ne? All right. So phenotypes, there are 4. 1, 2, 3, 4. You understand? And then the genotypes, there are 1, 2, 3, 4, 5, 6. That's what I say. Because this is homozygous and heterozygous, homozygous and heterozygous for dominant, and then homozygous recessive. Yes. Then they're saying that 3 different alleles, which are this, and then 4 blood groups or types, which we call the phenotypes. No, the genotype, the phenotypes. Yes. So it's A, B, O, A, B, and then O. That's what I say. A, comma, B, comma, A, B, comma, and O. So that you understand. Yes. So now we go back to our question. Describe the type of dominance that occur in the inheritance of blood group of woman B. Describe the type of dominance that occurs in the inheritance of blood group B. Now they're saying inheritance of blood group B. Check. They're saying that a man with blood group AB and a woman who is heterozygous for blood group, yes, heterozygous for blood group B, meaning that this lady it looks like this. See, yeah, meaning that obtained one dominant allele from the father or mother and another recessive allele from the father or the mother. So it can come either from the father or mother for A, and this either father or mother, because they are not specific in that regard. Yes. Use the genetic cross to show the possible genotypes of the phenotype of their children. So you guys here, this principle works to all of them. Start with P1, F1. Please, F1. Don't just say P1 and then you leave, you finish after you say F1. You will forget this F1 and then you lose a mark. So you say P1, F1, at least we shall come here and give you a T. Is it clear? Then you say P1 phenotype, because P means parent phenotype. Ne? Yes, you tell us. The phenotype is always obtained from the question. Check here. You use, because these 3 marks, if it is 6 marks, 3 marks are mahala. You understand? Use the genetic cross to show the possible genotype of phenotype of what? But what did they say? They said a man who is blood group. So you say man of blood group AB, times a woman of blood group B. You don't need to tell us heterozygous, because when you look at someone, you can't say that he's heterozygous. We just say heterozygous, homozygous, so that you know what you're going to cross. So you're saying P1, F1, we give you a tick. Now you tell us phenotype, you tell us a man of blood group AB times a lady of blood group B. That's all. Then we give you another tick. Why? Because you have identified it, but you always get it from the question. So that's the second mark for mahala. Then you talk about genotype. If you don't know the genotype, if the space, talk about meiosis. If you don't know the meiosis, if the space, talk about gametes. If you don't know the gametes, leave the space. Then you talk about fertilization. Don't know this fertilization, leave the space. But you shall come here and give you a mark there. So you have got 1 mark, 2 marks, 3 marks. Meiosis and fertilization, they must go together. If you know now, that's where you're going to obtain the other 3 marks. To know the genotype, because most of the cases, you people, you get lost on the genotype. When you get the genotype wrong, the remaining thing here becomes what? Becomes wrong. Someone is asking me, please, I answered that question. You have to rewind the video. Yes, I answered that question. Eh? Yes, for case of the DNA changing the triplet. Yes, I answered that question very well. Please, you have to watch it maybe when I'm done with this session. All right, guys, let's go to the next question. That is dihybrid cross. And then I show you what you do or what you focus on dihybrid cross. Dihybrid cross, we are not going to ask you to cross. No. What we might ask you about is identify the genotypes of the parent, the phenotypes, and the gametes. Ne? Yes. So guys, you need to know first identify which one is which. Ne? But now if they ask you what is the genotype of the parent here, for example, ne? Don't repeat the letters before you finish. For example, the genotype of this can be capital P, even capital L. Then another one is capital small p, small L. So the one which are the same, you combine them. Capital P, capital L, small l. So this becomes the genotype of the parent. I'm just trying to show you how do you identify. And if they ask you to identify the gametes from here, then it still is fine. You will say that P goes with L. P, capital L. So this one is done. Now P again with the small l, capital P, small l. So it's done. This with this now, small p, capital L. Then this with this, small p, small l. Sometimes people, what they do is not a cross. It's not a cross, but what they do this. They do this, capital P, small p, capital P, small p, capital L, small l. They just cross with this, capital P, capital L. They cross this now again with this, capital P, small l. And now they cross this with this, small p, capital L. And then now this with that becomes small p, small l. See, they form the same gametes. You understand? Yes. So at least you have done. Now they can ask you, they have given you the gametes. They want you to write the genotype of the parent. Yeah, that's where the story comes from. So you start with you start with one letter, capital P. Do I have any other capital letter? Yes. So you first leave it. Ne? Do I have a small letter of the same type? Small p? Yes. If I have a small p, I write the small p there. So they are supposed to be 2 letters. But if I don't have a small p, then definitely I'll have to write a capital letter. Is it clear? I'll have to write a capital letter here. But because I have a small p, therefore I will have to write the small p, small p. Then I go to L, capital L. Do I have another capital L? Yes. Do I have a small l? Yes, I have a small l. Therefore, it's going to go with a small l. Therefore, this becomes the genotype of the parent. Check. It's the same thing. Capital P, small p, capital L, small l. Then when you come here, it's capital P, small p, capital large L, or capital L, small l. So that's how you can identify that. Difference between transcription and DNA replication, I talked about it, guys. I talked about it. Just wind up. Just wind up. I know that you will find everything you have asked me. Ne? You are trying to ask me. Yes. Lindo, yes, I talked about it. Difference between transcription and DNA replication. Yes. And even the similarities also talked about. Then that both the DNA, double helix, unwind, they talk about both there is unzipping. Yes, when you talk about differences, you can talk about DNA is formed, RNA is formed, 2 templates are formed, 1 template is formed. So you guys, we talked about it, né? Yes. All right, let's continue to the next one. How many genes control? Once you see the dihybrid, then there are two what? How many genes? There are 2 genes, né? Dihybrid is controlled by 2 genes, né? Yes. How many alleles? Yeah, in most cases, there are 4 alleles. Yes, you'll find out the capital letter and the small letter. So depending on whether you see they are here. It's 1, 2, 3, 4. But there are 2 genes. Genes for the number of seeds per pod and then the shape of the what? The leaf. So guys, the hybrid is gonna be is simple, né? Simple. Just make sure that you do it. Né? Yes. So it's the same thing here. No one is gonna ask you to cross. Before I leave this point, I want to ask you a question. Is it conclusive to use blood group to identify the paternity, the paternity of the individual? Is it conclusive? I want yes or no. Is it conclusive? Then once I see the answer, then I will give the reason. If the answer is correct, I will give a reason why. Mm-hmm. 3 factors of, okay, the experiment, that's one you must know, guys. Lastly, let's go to, before I go to, please read about the stem cells, read about the cloning. Yes, cloning, the process of cloning. I designated a full video on cloning. Eh? Stem cells, what are stem cells? The cells which have ability to modify and form other body structures. Yeah, basically you just need to read this and the examples of these stem cells. Yes, let me just show you here from Excel and Life Science distinction material. Yeah, see sources of bone marrow, umbilical cord, adipose tissues, amniotic fluid, their uses, yeah, to generate new or replace the worn out cell or damaged cells, or to generate new cells, be used for research purposes. Yeah, so cloning, please know how to use this cloning, but most especially, let me just talk about, yeah, it can come like this or it can come as a doll. What? Do you know that this is the name of a person? Is a singer. Dolly is a singer from, I think it's from America. Do you know that? that is a singer from America. You just listen to people's songs, but you don't know that they're asking you questions and you don't know. I hope he's a singer, eh? I'm not a fan of. Okay. Is it conclude? Okay, thank you very much, guys. Everyone is saying, sir, can you please touch on stem cells? Okay, sure. Someone is yes, I'm saying that. No. Yes. It's no, it's not conclusive because many people share the same type of bloody group. Therefore, it's not conclusive. It's not conclusive. Yes, it's not conclusive. Someone say, yes, I'm going to talk about them. Let me just talk about stem cells, then we shall go to, not stem cells, I'm done with stem cells. We talk about the cloning. Guys, this is what I want to clone is this. I want to clone, let me just go like this so that it gives me the full version. I want to clone this. Therefore, I have these two. I get sheep A. If I want to clone this, I have to keep its nucleus. Is it clear? Why? Because the nucleus contains the genetic material. So I keep the nucleus, I throw away the shell. Then this one, I remove the nucleus because I don't want its genetic material, but I use the gamete, the ovum. But here I just get any cell from anywhere else apart from a gamete. So I throw away the nucleus because I don't want it, but now I keep this. But why do I need to have, why do I have to be with a casing of the cell? Why should I be with the casing? Yes, it's like you drinking water in a bottle of alcohol. What are we going to say? We shall say that you're drinking alcohol, Baba. Yes, that's what you're going to say, that you're drinking alcohol. Yes. Why? Because you are putting water in a bottle of alcohol. So when I put this nucleus into a cell, yes, this cell, it will look like a gamete. Remember, this is a diploid cell from anything else. So if I put this nucleus to this, then this one is going to look like a zygote because this is a casing of an ovum, and I've put a nucleus which is 2N. You understand? Therefore, it's going to look like 2N. So if they ask you how many chromosomes are here, these are, if they say the sheep has 24, therefore it's going to be 24, or it's 2N. Yes, you understand? Yes. So now, we are saying that which method do we use? We also ask you about the method is mild electric shock. Then you have here the foster mother here. And then we bring this, we put it in here. And then after putting it here, then this one starts to develop, develop that embryo, and then it forms the baby. This baby is exactly the same as this. You can ask you a question, why do we remove the nucleus? Why do we keep the nucleus? Because we wanted the genetic information from sheep 1. Why did we throw the nucleus away? Because we wanted the casing of the ovum. We are not interested in the genetics here. Yes. So which method we use? Mild electric shock. And then which sheep will this, which of these two sheep will this one resemble? Sheep 1. Give a reason for your answer, because we use its the nucleus, we use its nucleus or the genetic material to produce the sheep. Yes. So when they ask you such questions, they're not that difficult. You'll be able to answer and get it right. Paternity test, blood group. So it's the same thing we are saying. This is the question I was asking. Is it conclusive? It's not conclusive because many have the same type of blood group. So it's not conclusive to confirm or to determine that this is, yes, the parent of the child. Guys, now we are going to welcome back to our class. Today we're gonna be looking at the paper 2023 final exam. Yeah, so share the video so that everyone is here. We can discuss this paper. No one should miss out. This paper was really a very easy paper for those people who did the study, and for those people who did use my book for distinction material. Yeah, if you want it, you always get it from the link below. So this paper was a little bit easy, was just a little bit of easy, and we're gonna see how we can really see that those people, they can mark themselves and then they see, and also those who want to prepare for the next exam. All right, let's start without wasting time. Msaidi, as usual. According to Lamarck's theory, I know most of you don't like Lamarck because of his theory, but me, I like him because he started the story. All right, according to Lamarck's theory of evolution, or for evolution, acquired characteristics are not inherited. Obviously, we say that Lamarck says that acquired characteristics are inherited to the next generation. If you have a person, maybe your father gets an accident or is a bodybuilder, and then you're going to produce kids who are having muscles already. Acquired characteristic is inherited. So here the key point is not. So you have to take note of that. So this one was off. And then they are saying that organisms evolve because they don't because they adapted to their environment. No, here they are saying that organisms evolve because of the adaptation to the environment. How do organisms evolve due to the adaptation of the environment? Is it natural selection? Because adaptation will require that those organisms which are able to adapt will evolve, and those organisms which cannot adapt will die. Yes. And then number 3, they're saying that there is variation among organisms. Lamarck doesn't talk about variation. Yes, so this one is off. Then they're saying environmental change has no influence on the species diversity. Here, they're saying that environmental change has no influence on the diversity of the species. And then here they're telling you that organisms evolve because they adapt to their environment. So how are these two different? Here it means that environmental change have no influence. For example, that if there is scarcity of food, yes, and then the one which is on ground die out. This is opposite of this, né? And they die out, and then there will be no influence on the species, how they diverse they are different from each other. So that is wrong, because species will change based on the what they want, but still based on the environment. If they want to be, for example, they want to stretch their necks, they stretch their necks until they reach, for example, the tortoise. If we are using the giraffe, yeah, you call them giraffe, yes, because that's the best example most of the people are looking at. So the best answer here is gonna be B. And then they're saying that genotype for species characteristics. When you talk about the genotype for species characteristics, what does it mean? They're saying that contain 2 chromosomes. No. Physical appearance, they're saying genotype. Physical appearance is a phenotype. No. Is the composition of a gene pair. Look, they're not saying that genes. They're saying gene pair, meaning that you have, remember that when you talk about an allele, is also a gene, but is half of a gene or alternative form in which a gene can exist. For example, tallness is an allele, and also shortness is an allele. For example, capital T, small t, for example. Yes, so this is an allele, this is an allele, but this is a gene, meaning that it's a pair which makes a what? A gene. And then is represented by the genotype of a specific characteristic. here, characteristic. Here they're saying it is represented by one allele. It's not represented by one allele. Even if it is shortness, it will be represented by the two alleles. Yes, for example, capital R, or small t, they are saying a characteristic. Yes, so when you talk about the characteristic, it means that they are looking for the complete set of that characteristic. So the best answer here becomes C. And then they're saying after discovery of fossil, scientists classify it as after discovery of a fossil, scientists classified it as an African ape because it had, okay, what do African apes have? Large cranium? No. Small cranium? Yes, they have small cranium. Small cranium? Yes. Large cranium? No. So meaning that A and D are out. So these two are out. So let's look at this. Small cranium, non-prognanthus, meaning that they don't have a protruding jaw. Apes have protruding jaws. Yes, prognanthus means protruding jaws. So prognanthus, this is correct, this is wrong. So it means that the best answer is C. They're saying that large canine. This one is correct, this one is correct, but this is wrong. Therefore, B is wiped out. The best answer is C. So C is going to be the most correct answer. Yes, let's go to 1.1.4. The statement below describes the step in the process of cloning. I explained this, the process of cloning yesterday very well, but let's ignore that you are not there. So now let's look at it. Remember, they are saying that embryo, okay, let me just show you here so that number one, you get one cell, you get a cell A and cell B. So this is animal A, animal B. Get a cell, ovum. I'm just summarizing it very fast. Somatic cell. So here is somatic cell, meaning that is 2N. After that, remove the nucleus of the ovum. Yes, throw it away. Take the what? The casing. And then here, remove the nucleus because I want the characteristic of this. Bring the nucleus, throw the casing. So bring the casing here and bring the nucleus, fuse them together with electric shock. Yes, after that, bring these two, once it is fused together, it looks like a zygote-like structure. Therefore, it becomes, I'm just explaining so that because this is the process they are trying to show you, it undergoes mitosis. Bas. Then, let's go back to the question. They're saying that the statement below describe the steps involving in the process of cloning. Embryo in the implanted is implanted into the uterus of an adult female. Embryo is implanted. This must be around this stage 4. Okay, we shall find out. Nucleus from the somatic cell of the donor cell is extracted. This could be one. Nucleus. Check. Nucleus. They're saying the nucleus or the cell. Nucleus from the somatic cell. That's not step 1. You can't extract the cell before you get the cell. You can't extract the nucleus from the cell before you get the cell. So we need to first get the cell. We are looking for these two steps of getting these two cells. All right, we go back. They're saying that the nucleus from the somatic cell is inserted into the ovum. The nucleus from the ovum of another is removed. The ovum from, okay, now we have to look for it. They're saying that the nucleus from the somatic cell, meaning that, okay, they obtain the cell, yes, but now after obtaining the cell, in this statement, they're not talking about this. So meaning that they're starting from this. They're not talking about this. They're starting from 1, 2. Okay, let's go back. They are saying that the nucleus from the somatic cell of the donor is extracted, meaning that the nucleus is removed, is brought here. So this could be step 1. Okay, we shall see if it is the most correct. Step 1, so this is covered. They're saying that the nucleus from, the nucleus from the somatic cell is inserted into the ovum. the nucleus, this is done. The nucleus is of the somatic cell is inserted into the ovum. Do we have the ovum? The nucleus from the ovum of another individual is removed. This is step 2. Yes, this should be step 2. Because I removed here the nucleus, so I remained with this. So now I inserted this into this. This should be step 3. The nucleus from the ovum of another cell is removed. It's done. The ovum of the new cell is given. The nucleus from the somatic cell is inserted. Now this is step 3. So meaning that now I have 2, 4, 3. Then I'm remaining with this and this. So they are saying that the ovum is now, the ovum with a new nucleus is now given electric shock. Now we are here. We are giving it electric shock, meaning that now this becomes 4. Yes, and then now the embryo is implanted into, so meaning that I'm going to have, yes, it's going to be 2, yes, 4, yes, 3, 4, 1, meaning that 2, 4, 3, 5, 1. So this is the most correct answer. So the answer is A. Let's go to the next question. Then they are saying that 1.5, shrubs, these are short grasses, of the family. This family, they are saying that can be found in Australia, South America, Indochina, and parts of Africa, as shown in the map below. It is hypothesized that all continents were once one large continent called Pangaea, and that they separated due to continental drift. They said, okay, sharp. They're saying that this is evident. This evidence that the family, yes, this family, what about it? All belong to the same species. No, they don't belong to the same species. They don't belong. That's not an evidence to show that they belong to the same species. Number 2, they're saying that they are equal distributed in all continents. Who told you that? They are not equally distributed. Are you trying to mean that what is here and what is in South America and what is in Africa, they are equal? No. Wrong. They're saying become extinct. They're still existing. Arose from a common ancestor. Simple, because at first they were together, yes, but they went to different environment due to the separation of the continent. And then they developed into different species. That's why we say that they don't belong to the same species. Therefore, D is the most correct answer. 1.6. Yeah, these ones, they take some time, but when you reach there, we're going to be faster. The diagram below shows the DNA profile of a girl that her mother and 4 males. Are about the males. Okay, maybe they are claiming. Which male is a girl's biological father? I have to look for all the bands which correspond to the mother, because the mother is known. This goes with the mother. Done. This goes with the mother. This goes with the mother. Now, the remaining bands from the girl must be matching with the guy who is claiming. Okay, let's go. This one matches. Cool. Yes. This one matches. Is there any? Yeah, because this one is matching. So is there any other for the girl? No. So it means that this is number 3 is the biological father. All of them must be ticked for the case of the girl, but they must be the remaining one which did not match with the mother must match with the father. Hence, number 3 is the most correct answer, which is C. Okay, the following are the characteristics of group of animals. Yes, animals. If you're saying animals, Just a moment, guys. Okay. Just a moment. Okay. Animals. They're saying that which characteristics of group of animal able? Which are the characteristics of group of animals able to interbreed, occupy the same habitat, produce infertile, belong to the same species? Which combination correctly represents a population? Oh, which combination represents a population? Yeah, it means that you can't know which group represents a population. Just a moment, guys. Let me increase here so that I can see clearly. Yes. Yes. Which population does the population is being shown by what? Is being shown by the able to interbreed, and they must produce a fertile offspring, occupy the same habitat. Yes, they must be able to interbreed, but interbreeding they must produce the fertile offspring. And the only word which can help us to bring about to produce a fertile offspring is one word. Which word is that? Same species. They belong to the same species. So the answer is B. Same habitat, same species. Yeah. So we go to the answer becomes 1, 2, 4. 1, 2, 4. Where is 1, 2, 4? Is there any 1, 2, 4? 1. 1, 2, 4. Yeah, it's B. Yes, it's B. So yeah, it gives me the correct answer. Let's go 1.8. Which diagram? Let me see. Which diagram? Not which diagram. During Anaphase 2 of meiosis, the two chromatids of the chromosomes are pulled apart, each moving towards the opposite poles at a rate of 1 micrometer per second. This question, okay. I see it as a new content to students. Anyway, but it's just an application question. Okay, they're saying that, yes, two chromatids of the chromosomes are pulled apart, each moving towards opposite poles at a rate, this is the key point, at a rate of 1 micrometer per second. Okay, each second, 1 second, it means a distance moved is 1 micrometer. Let me say 1 like that. The distance in micrometers between the chromatids after 20 seconds. Obviously, if it's 1 second, 1 micrometer, after 20 seconds, they'll have moved 20 micrometers. So that is a direct question. However much is just an application question. So the answer becomes B. Yes. So the answer there becomes B. Yes, let's go to the next question. The next question, they're saying that which one of the following indicates the type of variation for I gave you examples yesterday. Anything which does not change since you were born is continuous variation. Anything which changes as you are growing up, it's continuous variation. For example, here they give you height. Height changes, therefore is continuous. The way you were born and the way how you are now is not the same way. Height is continuous. So is where there is this continuous is I think I have the answer now. Because the rest are wrong for the height. And it can't be correct for the next one. Okay, skin color. I don't think most of us we are born yellow bones. Check us now. Hey, we are original indigenous. Yeah. Yes, color changes, skin color changes. You check the kind of people. Some people, I want the black one. I want the light skinned one. I don't know what you're going to use for them for. But it changes among the individual. All right, this is continuous. Check. Earlobes. The way those earlobes are since we are born, unless you get an accident and then they change. But if not, it's the same thing. So, correct. So, let's not waste time there. Answer is B. Let's go to terminologies. I know sometimes the terminologies, they make these students a little bit of a problem with it, but I don't think these terminologies were that hard. Ne? Let's go. Let's go. The first terminology, they are saying the type of inheritance which produce intermediate phenotype. Intermediate phenotype. Don't forget that we said it in the what? In the distinction material book, our distinction material book. Ne? Yeah, for those people who were not there. But okay, there we go. Intermediate. How do you call this? Incomplete. Incomplete dominance. Intermediate. Yes, we talked about it. So the answer here is incomplete. Complete dominancy. Yes, done. The point at which adjacent chromatids overlap during crossing over. Chiasma. Chiasma. Guys, spelling is mandatory. The process by which information is copied from DNA to RNA. Transcription. Told you. Transcription. Let's go to the next question. What are they saying? They're saying that sugar found in DNA. Sugar. Thymine. They're saying sugar, not base. Gone. Thymine. That's a base. They're saying sugar found in DNA. Ribose is found in RNA. So for the DNA, it's going to be deoxy. That's why it's saying D. DNA, deoxyribose nucleic acid. So the sugar found there is deoxyribose. The bond that occur between nitrogenous bases in DNA molecule. The bond that occur between nitrogenous bases in DNA molecule. So how do you call that bond? That bond is weak hydrogen bond. Yes, you must say weak hydrogen bonds. Most of the people, they wrote peptide bond. Peptide bond. In some cultures, they say that sometimes you can laugh because of sadness. Not because you are happy, but you are sad. The same way someone can cry because of happiness. When I laugh at because someone is because of the sadness. So this is weak hydrogen bond, not peptide bond. Yes, because I highlighted it that these are the mistakes students do. Yes, okay, let's go. Manipulation of organisms' gene to obtain the desirable characteristic. How do you call that? Manipulation of a gene to get a desirable characteristic. Whenever you talk about manipulating in science, whenever you say manipulating, what did I say? I said that is engineering. Manipulating. But you are manipulating genes. So, what are you manipulating? Genes. Therefore, genetic. Simple. Genetic engineering. Guys, I told you don't need to cram. You don't need to cram. You just understand life science. Tell the people to join the channel. If you want a distinction, Thundereduc. Yeah. Use the distinction material. There is no way how you can fail a paper. Never. Yes. Okay. Unless you don't want to. All right. Oh, unless you found me. Oh, you found the channel late. But if you want a distinction, tell the whole world. Thundereduc. Okay. They're saying that representation showing the arrangement of diploid set of chromosomes. Hey, representation. They are looking for how do we call that representation? Which shows the diploid set of chromosomes, meaning that chromosome number 1, chromosome number 2, chromosome number, but they are saying, see, diploid. Like that, we call it a karyotype. Karyotype. Done. Terminologies out of 7. Okay, source of variation. How do you call random? We talked about this yesterday. Random mating, random fertilization. Source of variation for the species. I told you that a man produces over 500 million sperms. That is a rich man. That's why they tell you that you are 1 in a million. It means that your sperm was 1 in a million sperms which were released by your father and 2 there, and then only 1 you fertilize the ovum. So random fertilization is also random mating. You don't just find people come and say that, okay, now I want this one. You just go and then marry. You should just say, I want the small one, I want the big one, I want the light skin, I want the dark skin. You see, you're not doing it randomly. So you are minimizing variation. So variation, it must be random mating. When you are chasing this one and then another one comes into your eyes, you just go with that random mating, like a chicken. Yes. So this is both A and B. The answer is not A and B. The answer is both A and B. I explained it yesterday. Both A and B. Organisms that possess 2 factors which separate so that each gamete contain only one of these factors. Law of dominance. There is no domination here. Law of independent assortment. Organisms possess 2 factors that which separate that each gamete. Law of independent assortment. This is done in dihybrid cross. We call this law of segregation. Law of segregation, not principle of independence. So the answer here is none. Because this is a law of segregation by Mendel. You remember that guy Mendel discovered the law of segregation in he was there in 1865. Yes, Mendel, Georg Mendel, that priest. Yes, Georg Mendel. Yes. His life science is nice. So what you should do is be nice to it. Once you are nice to it, the subject is gonna be nice to you. If you are not nice to it, forget about. Okay, guys, just 1 second so that yes, Mr. Ramaphosa has given back electricity to us, so now we are happy. Yes, it's back. Yeah. All right. So if you see some disconnection a bit, just know that you're just switching the system. But we are continuing. Reproductive isolation. Reproductive isolation. Is my mouse. Okay. Reproductive isolating mechanism in species. You separate organisms through reproductive isolation, breeding in different seasons of the year. Correct. Infertile offspring. This is not correct. This is not correct, Baba. Look, same. This word, these people are confusing us, ne? Breeding in the same season of, no. It's supposed to be breeding at different seasons of the year. Let me show you what these people are trying to trick you up, ne? Let me just show you using the distinction material so that you see exactly how these people are trying to this is a real fake. They are faking us, so that we are off. We are sober. All right. Let me show you what they are showing you. Breeding, yeah, is here. You see, breeding at different times, not breeding at the same. So that answer is wrong. So this answer is wrong. You see, when you're saying the same, the answer is supposed to be breeding. This distinction material, there is no way you can go astray. Yes, so this is wrong. Infertile offspring. Correct. Check here in the book. Check here. Infertile of the way it is without even changing anything. Yes, infertile offspring. So you see, we say that mechanism of reproductive isolation. Now there are some people saying that I wish I knew this booklet. Sorry. Ne? Yeah. But you can refer to someone else so that you can get rewards. All right. So the answer here is B only. Yeah. Let's go to the next question. What does it say? They're saying that this is a phase. Ne? What I've seen in this paper, ne, as I'm going, ne, I don't know that much what is down, but what I've seen it here is they are using small words to take you off. if you don't read these questions very well, they're just using very small words just to take you off. All right, let's go. So we have to be keen, guys, when we are trying to answer. Yeah. So even me have to read too much so that I don't go astray. I might go astray, give you a wrong answer. No, man. All right, let's go. They're saying that the diagram below represent a cell in early stage. You see these people. The word early. That word, there is early and late. Late stage, mid stage, early stage. So now they're saying early stage. Okay, so if you look at this, which stage is this? This is not interphase. This is early prophase. Chromosomes which are homologous, they align together. Homologous. Yes, crossing over is not yet there, but they have aligned together. And you see that these dots shows that the nuclear membrane is starting to disappear. The nucleolus is already disappeared. You see? These people. So this is prophase. Prophase. Which prophase? Prophase 1. If you don't say prophase 1 is 0. Number of chromatids. So how many chromosomes do you have? 1, 2, 3, 4, 5, 6. So you have 6 chromosomes, but each chromosome has 2 chromatids. So 6 times 2, which is 12. So the answer here is going to be 12. Number of homologous pairs. They are saying number of homologous pairs. They are not saying number of chromosomes. Pairs. So it means that 1 pair, 2 pairs, 3 pairs. 3 pairs. Someone is going to write 6. Ask you, do you have eyes? Yeah. Do you see 6 there? No. I see 3. Why did you write 6? I don't know. Okay, let's go to the next question. Identify what is R. Yeah. R is nuclear membrane. Nuclear membrane. What is S? S is cell membrane. Membrane. Yeah, you know, I told you last time that you guys, you confuse nuclear membrane and cell membrane. T. What is T? What is now T? T is nucleolus. Nucleoplasm, not nucleolus. Nucleoplasm. So, the nuclear, okay, maybe they will slash it with the nucleus. Nucleus, yes. But based on this is nucleoplasm or nucleus. So if you give nucleoplasm, yeah, they might give you. If you give nucleus, they might give you. Based on what they have shown us. Ne? Yeah, don't say that. No, me, I wrote nucleus. Yeah, you might be right. Ne? Yeah, correct. All right, let's go. 2 organelles in animal where meiosis takes place. They're asking your parts. The most important structure on you. Which structure is that? Testes. Testes. That's 1. Oh, if you wrote it like this is also correct. Because this is singular, this is plural. Okay? I'm not saying that you write 2. No, just write 1. Hey, students. That's number 1. Number 2 is ovary. So they give you a tick here, they give you a tick there. Simple. They're saying that, you know, they write these funny words of these plants. Okay. Who knows this? I don't know. Let me just quote. The plant carries edible fruit. Scientists have been studying the inheritance of these 2 genes. One for the stem texture, and then another one is for the fruit shape. Okay, sharp. Let's go. They're saying stem can be okay, smooth. Smooth is represented by a capital letter. And then prinkly. Prinkly means that they have those yes, small letter. And then the shape round is capital letter, elongated is small letter. The type of cross, dihybrid. Dihybrid. Dihybrid cross. They're saying state the dominant characteristic in the stem texture. Texture is here. Ne? Smooth. Answer here is smooth. Smooth. Boom. Done. This goal. Recessive. Recessive for what? Fruit shape. Fruit shape. Elongated. Elongated. Let's go. 1.5.3. Give there. The genotype. The genotype. It means that you are supposed to write in terms of gene. Prinkly stem, and elongated. Prinkly is small error, so I can't be with the heterozygous. Small n, and then elongated. Elongated is small r. Listen carefully. If you wrote NNRR is wrong, ask me what? Why? Because of this space. I explained it yesterday. Yes, a space must not be there because this is a genotype. These are not sperms. It's a genotype. So it's supposed to be 1. Yes. They're saying that phenotype of plant with capital N is what? Because the small n would be masked. Capital N is smooth. And what? Capital R is what? Round, smooth and round. Sharp. If you have the space there, is cool, but the genotype is supposed to be no space. Yeah, let me know if you have questions. If you have questions, if you have challenges, yes, I'll smash them. Yeah, I'll smash them. Yes. Okay, let's go. Let's continue to the next question. Which question? All right, they're saying that the table below shows the information selected on hominid fossils. Okay. All right, we are hearing. What are they saying? What are they telling us? They're saying that P. What is P now? P. Oh, the common name. Australopithecus sediba carabo. We say this is a transitional fossil. Missing link between Homo habilis and the. Okay. Who discovered Dihybrid? I don't know why they like carabo too much. They brought it in 2017. Yeah. It was the second or third objective. Yeah. Is read the bagger. Lee. Beggar. Yeah. Like that. This is Sediba, Carabo. Yeah, you can and you can find that someone called Carabo is feeding this Carabo. All right. They're saying that Taum child. Oh, Taum, if you're coming from Northwest. Yes. How do we call? Remember, I told you don't go the paper without knowing the 3 Australopithecus africanus species. I said Taum child or Taum child, little foot, Mrs. Place. What are they looking for here? They're saying this is Australopithecus africanus. Cool. Just saying A africanus, they would have given a tick. Yes, because there is no Adipithecus here. So A africanus. But they have already given you the spelling here, so there is no stress there. They're saying that who discovered? Who discovered the Taum child? Raymond Dat. Yeah, it's Raymond Dat. Raymond. I don't know whether you call it Raymond or Raymond. Raymond. Dat. Raymond Dat. Check that booklet, the distinction material. Check this distinction material. I organized it nicely for you guys. And even I created a video. Check. Australopithecus, Carabo, read the bagger, Malapa. And then they're looking for the 3 Australopithecus africanus species. Check bone. Raymond Dat. Taum. Sharp. Let's go. They're saying name the fossil. Name the fossil. P. We have said it. P is Carabo. Then they're saying identify species R. We said Australopithecus africanus. Let's go. They're saying that give the name of the scientist who discovered Q. We say that read the bagger. Yes. Well, you can also give that one brief Ilof. Yes, Ilof. Where is he? Yeah, this one. So it depends on whom did you give. Yes. Then they're saying S, we say that it's Raymond Dat. Now, by this time, you have 50 marks out of 150. This one is enough for you to shift from a failure meaning that you're above 30 now, 30%. However, it's not enough for my students like you. Like you. Guys, don't have too much pressure. Like, now I'm failing. That's life. Still, doesn't mean that if you fail this, then life has stopped. No. All right, let's continue. It just make you to prepare, and I pray for you guys so that you pass this, so that you get enough chelete. All right, let's go. They're saying that X, what is X? That is amino acid. I told you guys that you must always label these things before you start. Amino acid. Simple. Let's go. X, Y. What is Y? Y is anticodon. Z, messenger RNA. Let's go. X, amino acid. Z, messenger RNA. Give the nitrogenous base of sequence, nitrogenous base sequence of DNA base triplet. Complementary to complementary DNA base triplet, complementary to mid codon of Z. Oh, they're saying DNA, meaning that we have to convert it backward. Let's say this is 3, this is 3, this is 3. So we are looking for this. So, but we are converting it backward. You have A, U, G. So we're going to be, say A goes with T, U goes with A, G goes with C. So this is the answer. That's what they are looking for, because they're saying mid in the middle one and for DNA. Then they're saying that give the nitrogenous base of Y. Where is Y now? Oh, the anticodon. So meaning that they want you to change this into transfer A, meaning that A goes with the, remember A transfer is an RNA. RNA doesn't have T. Therefore, we're gonna say U. U goes with A, and then G goes with the C. So the answer is this for this question and this for that question. Yes. Describe the process shown in the diagram above. Hey, what is this? This is translation. I told you guys. How many marks are this? Guys, don't tell me that you disappointed me. Unless you are not trusting me. Don't tell me that you disappointed me. There is someone who told me in the comment, I was so sad, and but I didn't stop. He told me that you lost me in the first 5 minutes. I knew that this one is looking for attention. Yeah, because I talk too much. Okay, I talk too much, but I wanted you to understand the concept. Yes. Anyway, what do we mark? We can have different ways how we can mark. But this one you are describing, name and describe. The process is called translation. That's one mark for the word naming. And then according to the messenger RNA, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acid are joined together by the peptide bond to form a required protein. Simple. That's how I have been describing. But still in my book, the distinction material, I showed you exactly what you are supposed to do. Now the mouse is jamming. Okay, doesn't want me to this mouse doesn't want me to give you okay, let me use this. Okay, this mouse doesn't want me to give you answers, ne? Sorry. Yes, born it's there. Each transfer A is a marking point. Here is a marking point. This is a marking point. Each transfer A carries a specific amino acid. We give you a tick there. When anticodon on the messenger matches the codon on transfer A, we give you a tick. Specific amino acid, we give you a tick. matches the anticodon. Yes. Then you're saying that this brings the required amino acid. Yes. Where does it go? To the ribosome. The amino acids are joined together or attached by peptide bond. Guys, we call them peptide bonds. We don't call them polypeptide bond. We call them peptide bond. I was very clear when I was explaining this. And then to form a required protein. That simple, short as that. But you can find a person writing a full page. So you're okay. That's short as that. That's what we wanted. Let's go. So this description thing, I'm not going to waste too much time because it is open thing, and you can find them in any book. Yes, you can find them in any book. Yes. Let's go to the next question. What are they saying? Just a moment. Yes. What are they saying? They're saying that the table below shows the codons. I explained this. Guys, take note of this because I explained it and I said this is one of the confusing subtopics. The table below shows the codons of codons that code for some amino acids. Okay, sharp. A mutation caused by DNA triplet to change this. Okay, so it means that we changed this to this. But they're saying this is DNA. But what is here is messenger. Meaning that to read this, I have to change it first. So I'm going to say that, okay, this was C. C goes with G. T goes with A. And then G goes with C. But change it to C goes with G. T goes with A. And A goes with U. Because this is messenger RNA. Wabona? Yes. Now I come. I go to, you go to what? I go to this table. I look for it. GSC. 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 GSC is aspartic acid. What about this? Does it correspond to a different amino acid or the same amino acid? Let's go. And then you're saying that GAU, aspartic acid again. Wabona? Ntuli. I can call you Ntuli. Yes? Isn't it? I'm just saying Ntuli because Ntuli called me and said that, thank you very much, sir, for teaching me. Yeah, I think I managed to pass. I pray for you guys, so that you pass. Sometimes when you're marking you're marking your students, and then you get something, you think of something, you end up giving a wrong one correct. You never know. Make sure that you pray until the results come back. Yes. Anyway, so since they code for the same amino acid, yes, so it means that the protein will not change. But these are 4 marks. Where am I going to get the 4 marks? You're gonna say that this change of this, yes, this changed into this. You writing it GAC changed to GAU. Those are marks. Those are 2 marks. This changed to this. Why add? Why this one is a mark and this is a mark? Is because you changed it. You see, you changed this. That's 1 mark, and you changed this. Those are 2 marks. And then this code for the same. This code is for aspartic acid as well as this code for aspartic acid. Meaning that these two, they code for the same amino acid. Is another tick. Those are 3 marks. Then the 3rd mark, the 4th mark will say that this will not change the protein formed, or it will not change the functioning of the protein formed. Simple. Why? Because it's like calling you with the first name, calling you with the second name. You are the same person. Yes. Or sleeping on your bed when you are straight and sleeping on your bed when you have carved somewhere, or does not mean that you're a different person. It's the same person. So they're just saying that, yes, however much the mutation occurred, but the mutation did not change the amino acid. Simple. This question, they have brought it back again. Tabulate 2 difference. I emphasized this question of mutation, and I told you that I know what students find when it comes to this question of mutation. Anyway, this question. Tabulate two differences between DNA nucleotide and RNA. I explained it before. Yes, those people who were on my channel yesterday. They are they are not asking you the differences between DNA and RNA. No. They are saying DNA nucleotide and RNA nucleotide. Clear? So there are two differences. The first one, DNA nucleotide, has thymine. And the RNA nucleotide has uracil. The second one, DNA is made up of, nucleotide is made up of deoxyribose sugar. And RNA nucleotide is made up of ribose sugar. There is no any other difference. They are looking for the nucleotide. They are not looking for DNA. They are not looking for they are not looking for just those nucleotide for the RNA and for DNA. That's all. So if you wrote a lot of things there, but there are 5 marks. Where is the fourth mark? Fourth mark is for the table. Just drawing a table and say DNA nucleotide, nucleotide. Even if you didn't write anything here, you draw a table, a complete table, you will get a mark. At least you are not getting 0. Yeah, at least. Yes. Let's go. Down syndrome. I talked about it also. Down syndrome. Okay, let's go to Down syndrome. What are they saying about Down syndrome? Okay, let's go. Down syndrome is the result of individual having extra chromosome on chromosome number 21. Okay. The genetic variation that can be caused by Down syndrome are, okay, trisomy 21, all somatic cells are individual, 3 copies of, okay, chromosome number, all. This word is a key point. And then they're saying that mosaic means something which is moving. Yeah, okay. Individuals have only some cells, not all. So this is a key point versus this extra chromosome, which is caused by abnormal process of cell division after. This is after. It's caused by after fertilization, but what occurs during gamete formation. This is during gamete formation. This is after fertilization. You see? Yeah. That's the major difference. The type of mutation lead to the, trisomy 21. The type of mutation that leads. You can say chromosomal aberration. Those who wrote chromosomal aberration, correct? And then chromosomal mutation. Because these are chromosomes. Chromosomal mutation. Yes. Or aberration, correct. Abnormal process during gamete production that lead to 3 copies of abnormal process that lead, name the abnormal process that lead to the 3 copies of Nondisjunction. The type of cell division that occurs after fertilization, it is mitosis. They're saying describe how the process mentioned above, which above B here, Nondisjunction lead to trisomy 21 is 5 marks. This is a general question. Yes, go back to my distinction material so that we don't waste too much time there. Yes, I talked about it, but sometimes people they have challenge. But before I left meiosis, I said that please don't go without knowing this process, because this process they will ask you. Here is a question. Nondisjunction, failure of the chromosome to separate rings. They are looking for this. You see, this page. Each nucleus has it contains 23 chromosomes after meiosis, but nondisjunction when occurs, okay, one nucleus will contain 22, 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 , 22 of the resulting gamete will fuse with the normal gamete. But they said, yes, with the normal gamete. They said, yes, they said what will happen. Yes, so the normal gamete is 23. Then the resulting, the 20, if the 24 fuses with 23, it's going to be 47. The 22 fuses with the 23, it's going to be 45. Yes. So in this case, they are looking for 23 because they're saying trisomy 21. It means that the 21, it has an extra chromosome, not short of a chromosome. Yes, let me explain to you. Yes, and then they are saying that, let me show you what they are trying to say here. Yes. And remember, during Nondisjunction, this is a cell, under metaphase. Yes. So this is what happens. So during Nondisjunction, the whole set is going to go to one side. So meaning that I'm going to have 2. So the normal one is going to be 2 cells. I'm going to have 2 here and 2 here. But during Nondisjunction, the whole of it is pulled to one side. And then this one, yes, it will go to one side. And then this one, another one will go to one side. Meaning that here, yes, here I will have 1, 2, 1, 2 plus this one, 3. And then this side, I'm going to have only 1. So this one has extra chromosome. 2, 3 instead of 2. And then here it has short. So they are looking for this. Because they are saying, yes, if it occurs on chromosome number 21, it becomes trisomy 21. Tri means 3. 1, 2, 3. You understand? So now, when this one fertilizes a normal ovum, then it's going to have, now the cell is supposed to have 23 chromosome. But this one is not going to have 23 chromosome. It's going to have 24. So if this fertilizes a normal ovum, which is 23, it's going to result in 47 instead of 46. So that's what they are saying here. So go to the distinction material. Yes, get there. So that's what they are trying to explain, or they're asking you here in this question. Let's go to the next question so that, yes, this describe 2 differences between trisomy 21 and the mosaic Down syndrome. Yeah, I think I talked about it. Trisomy 21, all cell, check, all somatic cells in the individual have 3 copies of trisomy, of chromosome 21, you see, all. But here, yes, make sure you draw a table. I think they don't want a table. They're not saying tabulate. While here in mosaic, the individual has only some cells with extra chromosome 21. But here all the somatic cells. Then number 2, the process, when does it occur? It occurs during gamete formation. While here it occurs during or after fertilization. Those are the 2 differences they are looking for. It's just as something from there which you can use to get the answer. All right, I see ourselves trying to finish soon. Yeah, so we are going to the question number 3. Question number 3. How is the paper? Let me know. How is the paper? Let's go to the next question. Yeah, I think the paper was simple. The paper was simple for those who read. Yeah, let me say like that. And I think I should come back and I teach you how to read. It does not stop only here, even when you go to the university. Good. All right. The diagram below shows the inheritance of type. Oh, they like this funny name. Anyway, I'm not going to remove my teeth because of these names. Okay. Is a rare disease which lead to this destruction of the neuron. It is inherited as a autosomal. Okay, autosomal means that it occurs in the body cells, meaning that it's non-sex linked. It's occurring in these other cells, these other chromosomes, not on chromosome number 23. Yes. Okay, TT. So meaning that you must not use X and Y. This is not sex linked. It's autosomal disorder. Anyway, Let's go. Describe what is meant by is a disorder which is caused by a gene carried on non-sex chromosomes. Yes, non-sex chromosomes, meaning that from chromosome pair number 1 to chromosome pair number 22, it must not be sex. Yes. How many sons do individual 1 and 2 have? 1 and 2. Yes, sons. Son is a box. It's 1, is 2. But this son is not belonging to these two. You understand? This guy, yes, just married the daughter of these two guys, meaning that the answer is 1. You understand? You see? Yes, this is just an intruder who came to marry the daughter. Yeah, the beautiful daughter of 1 and 2. Yes. So the answer here is 1. Using the individual 3, 4, 5, explain why it can be concluded that this disorder is controlled by a recessive allele. 1 is 3, 4, 7. 3, 4, 7. Okay, let me clear up here so that at least, yes. Okay, let's see. 3. Okay, this is the affected individual, né? And these two people who are, if they produce a person who is affected, it means that these two people were heterozygous. Meaning that it is capital T, small t, capital T, small t. You see? Since they carried that what? Since they carried that gene, this gene, recessive allele. And then this recessive allele and this recessive allele gave rise to this. Meaning that, what is the question? What are they saying? Which question is this? 2.5.3. They're saying that it means that individual 3 and individual 4, they are both normal individuals. I see. That's the first mark. But they gave rise to individual 7 who is affected with the disease. You have to tell us the disease, né? A female who is affected with the disease. Yes, that's the second mark. This means that individual 3 and 4, they were heterozygous. Meaning that they are heterozygous because they are both normal, but they gave someone who is sick. Meaning that they were heterozygous, they were carriers. That's another mark. And then they pass their recessive allele. 1 recessive allele from 3 and 1 recessive allele from 4 to individual 7. That's another mark. You're able to get all the 5 marks. 1 recessive allele from 3, 1 mark. 1 recessive allele from 4, another mark. Yes, to form this one who is affected with the disease. So, yeah, it is an application question, whereby you have to apply and explain exactly what happened to inform individual 7. Then they are saying that individual 1 and 2 can produce children with 3 possible genotypes. Okay, list those 3. Individual 1 and 2 can produce. Yes, individual 1. 1 and 2 is the same thing. 3 and 4, they are both heterozygous. Okay, they can produce. If they are both heterozygous, it means that, look, it means that this is capital T, small t, heterozygous, capital T, small t. So it means that when they produce, they're going to produce capital T, capital T. This is what they are trying to show you. Capital T, small t. This is capital T, small t. And then small t, small t. You check these two. They are both heterozygous. This is homozygous and this is the homozygous. Let's go. The question. They're saying that list all the possible genotypes that have 25% chance of offspring. Then this is 50 and this is 25. This is 25. Meaning that I'm going to come here and say capital T, small t, small t. A comma. That's all. That's what they're looking for. They give you ticks. In human, hemophilia is caused by a recessive allele of X. Of X. Anyway, it's caused by recessive allele of X. Let's go. Of X. They're saying that in humans, hemophilia is caused by recessive of X. Okay. This is carried on X chromosome, meaning that this is sex-linked. The moment you say that that's sex-linked, hemophilia, yes, we know is a sex-linked disorder. A woman who is heterozygous for hemophilia marries a man who is heterozygous for hemophilia. That's a woman. It means that it's like this, capital H, small h. Yes, marries a man who is hemophilic. Yes, meaning that the man is definitely is like that. Is use the genetic cross to show the percentage chance of couple having having a daughter who is homozygous for normal. Homozygous for normal and is a daughter, meaning that this person is gonna be like this. There's no way. Check, the father is not normal. Then it means that the answer is gonna be 0. 0. Yes, but they are saying use the genetic cross to show the percentage chance. The percentage chance is 0% because there is no way. But they are saying use the genetic cross to show. So you're saying 0% is 1 mark, but you have to show. Don't forget to show all the steps in a genetic cross. Let me see, is it P1? Let me see. A man who is, they didn't talk about anything. So I'm going to come here and say P1, F1. P1, parent, phenotype. You go to the question, you'll get the phenotypes there. I told you that this one is simple, and a man who is heterozygous, a woman who is heterozygous, meaning that this is a normal woman. Yes, marries a man who is hemophilic and a man who is affected. Then you go to genotype. Genotype, I'm just rushing so that a lady was like this because they say that heterozygous, marrying a man who is like this. Yalla, let's go. They're saying like that. Don't forget that these are gametes. I always tell you that they are not at the beach, so they must be circled. If you didn't circle, it's still fine. Don't stress. Give you a tick. These are gametes. Wabona. Yeah, but how do you call this process? Meiosis. You saying P1, F1 will give you a tick. So that's the second tick. 0% is a tick. But it's a second tick. This is a third tick because we got it. So you have 4 marks. Mahala. The question is out of how many? 7. So we are searching for the 3. This is the next tick. It's the next tick. Then after that, you can draw your Punnett square here, or you can just cross. When you cross, you'll find out that this, look critically. This goes with this. So it's going to be X, capital H, X, small h. Then this goes with this, capital H, small, capital H, yes. And then this with this, small h. This with this, small h, and then y. Is there any probability of having a child who is homozygous normal and is a female? No. There is no way. Therefore, this is offsprings. F1 offsprings. And then you remember you have to cross, show cross or the Punnett square. So, and this process is called fertilization. So we give you another mark for this. But what I know that the marks are more than what we stipulate. But you saying that therefore the percentage chance is 0%. This is going to be a compulsory mark because the question is emphasizing that percentage chance. They want a percentage chance. Do you understand? Yes. So it's 0% having a child who is normal, homozygous, and is a female. Yeah. Let's go to the next question. I think I'm clear. Yes. Let's go to the next question. These papers are simple if you read. If you don't read, they become sour. But I don't expect you to. Let's go to the next question. The next question. Question number 3. It means that now you have 100 marks. 100 marks. See now, you are approaching slowly a distinction. You're approaching a distinction. I know some of you just made small mistakes, which can be acceptable, but there are some mistakes which cannot be accepted. So yeah, anyway, let's go. Yeah, let's go. This is year 2023. Yeah, I know some of you gonna be successful. I pray for you so that you become successful. Anyway, learners conducted an investigation to determine the blood group was the most common in the community. I talked about a pie chart yesterday. Yeah, they conducted information about, okay, 200. It's a big number. It means that the sample size. Take note of that. In each of the 3 types of blood group. Okay, yalla, let's go. They're saying that in their community, they did not include the first donor in the investigation. Why? Comparison of results. Okay. Yeah. The pie chart below shows, all right, let's go. The percentage of each blood group in the sample. I know you read all these questions, you know them. So I'm not going through too much. State the aim of the investigation. Ooh. The aim must start with the word to and is always put in the first statement or second statement. That's why always I tell my students, if you can't find it, then you have to figure it out. Learners conducted an investigation to, that is the aim. From to, the investigation, the aim is to determine which blood group was most common in their community. Yes, in the learners community. Conduct an investigation. Yes. So the aim is done. Answer the following question. State 3 planning. We always ask you this, but you go back and read the question and see the possibilities for you. What are you going to plan? What are you going to do? So, but I need these people who are going to be giving me blood. So you have to organize them. Yes, get those 200 people. Yes, you are not just going to just get them. Yes, you have to get permission because this is a community thing. Get permission where you're going to do this, or the clinic, or try to go inform them so that they know and they accept. You are not going to force them. Get permission so that they can do that. You are not going to just collect the blood just like that. Get the necessary equipment, planning before you do the activity. Yeah, that's what they are looking at. Yes, something like that. State one way in which learners ensured. Yesterday I told you the word language and I emphasized it. If they ask you, don't ask you, how did this person ensure, how did the person do increase reliability, or how do you ensure reliability? Now they say that, how did the learner ensured? That is a past tense. Your answer must be in a past tense. The learner ensured reliability by using two. Remember, reliability is to increase the sample size by, using a big sample size, by using 200 people. 200 people. You see? So, this learner used the 200 people. Sharp. So, that is done. Give the reason why give one reason why they did not include the, yes, we said comparing the result. Calculate the percentage, the number of participants that had blood group B. Show your working. Blood group B. They use 200, but only 15% blood group B. So you just come here and say 15 out of 100 times 200. So the answer you get is the answer, around 30%. Done. They give you your 3 marks. Give you a mark, give you a mark, give you a tick. Done. They're saying that name the blood group that has only recessive allele. Only recessive allele, blood group O. Remember, it is recessive allele. Remember? Yes, I said it yesterday. Results of codominance is AB. I said it yesterday. Remember, it's A and B. Give the possible genotype of the blood group represented by 25%. 25%. Where's 25%? Is A. But they're saying the possible genotypes. If you remember, A is either homozygous. Go to the distinction material. You will see what I was saying yesterday. A, heterozygous. I explained this yesterday. I explained it very well. Very well. Yes. I hope you guys did not disappoint me. Ne? I don't want the marks to come back and then I'm disappointed. Say, oh, why did I do this for my learners, these learners of mine? Ne? No. I don't want that. Ne? I want everyone to pass. Ne? Yeah. I want everybody to pass. Anyway, let's go. Question 3.2. We are almost done. We are almost done. Anyway, question 3.2. These are dogs, man. Do you like dogs? Let me know the question in the comment below. Do you like dogs? Do you have a dog at home? Sometimes they call you a dog. And a dog. Okay. Dogs have been selected and bred over many years to produce approximately 340 different dogs breeds. They have been bred for certain characteristics desirable to humans. Saying that analysis of 786 okay, gray wolves, only direct ancestor, present day dogs bred. Okay, all the dogs belong to, I'm just reading so that I get the idea, ne? But you know, you can read it for yourself. Okay, I've got the idea, ne? Very fast. They're saying that name the process whereby the different breeds of dogs were produced. Artificial selection. Yes, these are the things we are talking about here. Where am I? Yes, artificial selection. Is it? Yes. Where is it? Okay, this is meiosis, ne? Yeah, artificial selection is they are looking for you. These dogs, even this question has these dogs we are talking about. Exactly the same dogs. Don't disappoint me. Don't disappoint me. Don't disappoint me, guys. Dogs, wolves. They're there. Born. Check. They are dogs, the wolves. That's what are the wolves we are talking about. Okay, they're saying that. Name the artist. What did we say? Is what? Artificial selection. Human. They select desirable traits. You see? That's what they said here. Desirable. They're desirable to humans. Yeah. Yes. Distinction material. Get it. Yeah. Get it. I'm talking about those who, yeah, your sisters, your brothers. Yes. Anyway, let's go. And we're saying that name the process whereby different breed of dogs were produced. Artificial selection. Describe how human carried out the process named above. Meaning that they got the characteristics they wanted. They let that individual which has that characteristic to mate with the other characteristics they want, and then the other one which doesn't have the characteristics were not allowed to give to have sex, meaning that to pass their genes to the next generation. So after many generations, they had to be with that desirable characteristic. That's what they are saying. So if you look at this example, check, it was about milk. Choose or select the cow in your herd, or now they're talking about the dogs to produce which has that characteristic. Yes. Then they are saying that let only those dogs to reproduce, the one which have the specific characteristics, then select the offspring that produce that characteristic most. Yes, let those only those offspring reproduce, allow them to have sex and have offspring like that. Keep repeating this process of selection and then the breeding until you achieve your goal. That's it, to get the desirable dog you want. Explain how it can be confirmed that all the dogs breeds belong to the same species. Same species. It means that they can interbreed to produce a fertile offspring. If they are not producing fertile offspring, then they don't belong to the same species. Yes. They must produce to produce a fertile offspring. Hence, this answer becomes right. Yes. Explain how present-day dogs may have been disadvantaged. Yes. In the relation to their common ancestor. This common ancestor, wolf, Baba, it could resist diseases. Now, you have to take it to how do they call it, the veterinary doctor. They were staying in the wild. So these dogs are susceptible to diseases. Less chance of these dogs being adapted to the environment. Yes, these dogs, the one which are new, yes, they have less chance. This one, it could adapt, Baba. Yes, meaning that some genes are being lost as you are doing this selection. Yes, so the dogs are being disadvantaged. Yes, let's go to the last question. Describe the process of speciation by geographical. Geographical barrier. Guys, I told you don't go to the paper. Speciation is here. This is what they're looking exactly. This formation of new species. Exactly. There is a population of a particular species. A population of a particular species becomes split into two by geographical barrier. As a result of, you don't talk about geographical barrier. Okay, you can say river, mountain, or whatever, but geographical barrier. As a result of two parts of the population cannot interbreed. There is no gene flow, yes, between the two. The natural selection will occur independently, yes, due to different environmental conditions. As a result, the population became genotypical. You start with genotypically, then phenotypically different over time. Even if you remove you bring these two populations, you mix them together, meaning that you remove the barrier and you let them mix and they interbreed. They cannot produce a fertile offspring. They cannot produce a fertile offspring. Therefore, one or both population have become new species. This is exactly without changing any word or what. That's what you're supposed to write. That's what you are supposed to write. Someone is going to tell me that what if I used a scenario? That's not KO because they didn't give you a scenario. KO is when the examiner sees, just give you a tick, not arguing with him or herself. Yeah, should I give no? Something like that. Don't like it. Just when the examiner sees your paper, is marking it as you are. You know, when you're marking someone, you see that someone is getting all the ticks. Yeah. You feel good, Baba. You feel good. You feel good. The same way you feel good when your marks are coming back and you have distinction. How do you feel? You feel like you are not fitting on the when marks are nice. All right, let's go, man. You feel good the same way I'm feeling good when I'm teaching you. Yeah, I feel good, ne? I feel good. All right, let's go. They're saying that. Name the type of diagram represented above. Okay, the diagram will represent the evolution of the family Hominidae. Okay, sharp. They're saying that. Name the diagram. I showed you these things. Phylogenetic tree, and I showed you the questions you're supposed to answer. And I don't think that they skipped those questions here. Let's go. Name the diagram shown above. Phylogenetic tree. Done. Give the letter of organisms, which is the common ancestor for all the hominids, P, because all of them, they come from here, is P. They're saying that, what is common, okay? Share most recent common ancestor with the gorilla. Gorilla is here. Gorilla is there, but they share a common ancestor. What shares the common ancestor is here, meaning that is this. But what shared is R, because gorilla and R, they share a common ancestor. So the answer here is going to be R. Give two organisms that have S as an ancestor. Two organisms that have S. Where is S now? S is this. Is this point. Stop it. Stop it when technology. Okay, this is S. So meaning that you're going to say this and this. Those are the two individuals. Let's go. Name two organisms that have S as a common ancestor. We have said it. pedal. These ones, gorillas, you know them. Is chimpanzee quadrupedal? I think so sometimes, but these ones, they are quadrupedals. Okay. They're saying that describe 3 anatomical features of the skeleton of a quadrupedal organisms. Skeleton, when you talk about a skeleton, you're gonna talk about the spine, the cranium, because that's a skeleton, the spine, the cranium, and then the pelvic girdle. Yes, talked about. I showed you here. When talking about. Yeah, for the case of the skeleton, here, see, it's a skeleton they're talking about. They're saying about quadrupedal organisms. Foramen magnum is in forward position. Yes, it's a skeleton. Yeah, that. Yes. We talk about the spine is C-shaped, and then long and narrow pelvis. That's what they are looking for. Simple. As simple as eating ice cream. No bones. Yeah, let's go. Second last. Yeah. Fossil evidence is used to support out of Africa hypothesis. State out of Africa hypothesis. Modern humans originated from Africa and migrated the rest of the world. I told you when I was concluding, don't go to the paper without knowing this. Modern humans originated from Africa and migrated the rest of the world. And I showed you this. Take note of this. Unless you do not want to trust me. The modern humans originated from Africa and migrated the rest of the world. That's what they want. The word modern must be there. Modern. Don't just say humans migrated the rest from Africa. And Homo habilis, Homo naledi, Homo neanderthalensis, all those are humans. But the modern ones, Homo sapiens. Yes, they're looking for. Okay, let's go. Last question. Describe how the fossil evidence is used to support out of Africa hypothesis. How is this fossil evidence? How is this fossil evidence used to show out of Africa hypothesis, guys? How is it used? Describe how this fossil evidence is used to support out of Africa hypothesis. You guys, I told you this and then you didn't write. Okay. You talk about all the Adipithecus, Australopithecus, Homo habilis. You say that only, the word only must be there. Only. Adipithecus, fossils of Adipithecus, fossils of Australopithecus. You can talk about the examples. Fossils of Homo habilis. Only found in Africa. Then when you go to Homo erectus, you talk about the oldest fossil of Homo erectus, distinction material, and the Homo sapien. Homo erectus and Homo sapiens. Here you talk about oldest fossil of Homo erectus and Homo sapiens were found in Africa. Don't say that only found in Africa. Nope. You have to say oldest. The key point there is oldest. That's why I put it in bold italic and I was supposed to underline it so that your eyes can open when you are reading it. I think, guys, the paper was easy. Yeah. Don't forget to subscribe, like, and share. It has been M. Saidi, has been taking you all over this paper. I think, yeah, the paper is a little bit easy. It was a little bit easy. Okay, they had some challenging questions, but it was not that bad. Ne? It was not that bad. Yes. Thank you very much for being with us. Yeah. Thank you very much, guys. Ne? It has been a wonderful paper. Don't you think so? Let me know in the comment below to know how was this paper to you, based on you. How was the paper? Yeah, let me know in the question in the comment. Thank you for being with me from the beginning until guys, ne? Yeah, I just love you so much. Stay blessed, ne? Stay blessed, ne? Yeah. Maybe I just a small note that even physical science, yeah, even other papers we will be doing discussing them immediately after you have done them. Thank you very much, ne? See you. Is M side as usual? Yeah. You know the protocol. If it's your first time to visit our channel, don't forget to do that. To hit that subscription bell so that we can enjoy the channel. All right. Yeah. These are the topics which you are going to be dealing with on the paper we're gonna write. Number one is DNA code of life. So we're gonna see what exactly you're supposed to focus on when it comes to DNA code of life. Yes. Number two, we're gonna look at meiosis, exactly the same thing as we said. So if you look at DNA code of life, it has 18%. This is 18%, which is 27 marks. These 27 marks are easy to get. So we shall see how we're gonna get these 27 marks. Meiosis, there is some people they call it a devil, but it's not a devil if you really attend our classes. So it is 21 marks. So having these two topics is enough for you to be skipping out of out of what? I think you'll be able to skip out of um the low mark which you call the 30%. But I always tell my students, please don't be fooled by any system. I'm saying it right, I think. By any system, 30% is not taking you anywhere. So make sure that you focus for something above 50%. That's why I'm saying that please read more, focus on more on this. You'll be able to get more than 50%. Genetics, yeah, we look at where to get free marks in genetics, and then that is 32%, which is 48 marks. So if you have genetics, as genetics is enough for you to get out of the level one, based on the government level one, which is 30%. 30%, we only need 45. So however, to me, it's not a pass. You are still failing, but we want to achieve so that you push these marks more than what? More than 50%. Then evolution is divided into two. That is evolution by natural selection and human evolution. We're going to see where to focus on when it comes to human evolution and when it comes to evolution by natural selection. Guys, are you ready? Let me know if you have any challenge. Junior is saying, hi, it is not. It Hi, is it on my side? How about it? I don't know what you're saying, but let me know if you have any challenge so that I'm able to sort you out. Yeah, by the way, it is a live class. However much today I'm not putting my face on. Yeah, but if you want, if there is any need, if I want to emphasize something, then automatically I'll put that face on. Guys, DNA code of life. I'm not going to be teaching. I'm gonna highlight where you're supposed and then remind you. That's it. And then I show you that this Zwan is saying challenge on human evolution. Okay, I'm gonna reach there. Challenge on human evolution. Don't stress. I'm gonna reach there. They're saying that how did I get distinction? How do I get distinction material? You order from us. All right, let's start. DNA. Here on this diagram, what you need to know is what called a chromatin network. Yes, chromatin network, then the nuclear membrane. Please make sure that you know that. Then here, when it comes here, describe the structure of DNA, 6 marks. Yes, describe the structure of DNA. This is the reason why we have all this. Ne? Yes. So here, these are nucleic acid, whereby you have messenger RNA, transfer RNA, ribosomal RNA, in actual sense RNA and DNA. So all of these are made up of nucleotides. So the building blocks are called nucleotide. The building blocks of DNA, if they ask you that, you say it is a nucleotide. Sometimes we call it a monomer. Please don't forget about that. Ne? Done. Building blocks are made up of a phosphate, sugar, nitrogenous base. If they ask you about DNA, then make sure that you know that when it comes to sugar, don't say sugar, say deoxyribose sugar. And then when it is RNA, be specific. Don't say that sugar. No, say that ribose sugar. I think I'm clear there. You need to know the symbol that a circle means that, this pentagonal means that, and then that square means that. Is it clear? Yes. Then we go to types of DNA. Here the question comes like this. Let me tell you how the question comes here. The question comes here, give 2 places where you can find DNA in the plant cell. So you'll find out that people won't talk about the mitochondrial DNA, or they won't talk about the nuclear DNA. In the plant cell, if they bring about plant cell, you mention all of them. Nuclear DNA, mitochondrial DNA, chloroplastic DNA. But if they say the place, then you talk about nucleus, mitochondria, and chloroplast. I'm done. Let's go. When it comes here, what you just need to know 2 things. Who discovered the structure of DNA using X-ray diffraction pictures? Only this now. Then that's when you talk about Maurice Wilkin and Rosalind Franklin. If they ask you who discovered the structure of DNA, just like that, you talk about James Watson and Francis Crick. The last question is, who discovered the structure of DNA using the X-ray diffraction pictures? Sorry, using who discovered that DNA is double helix with the complementary base pairs? That's when you talk about James Watson and Francis Crick again. So I think, guys, you understand that very well. Let's go to the next sub question. This one can come as 6 marks. Describe the structure of DNA. I don't need to memorize what is here. Yes. However much you need to know the marking points, what you are looking for. Is it clear? What you are looking for. So, what you're looking for here is one thing. Come back to the structure of DNA. Yes. Go to the structure of DNA or draw it. Okay, I'll come back here. Draw the structure of DNA and then start to describe it. DNA is double helix with the complementary base pairs. It's made up of nucleotide. Each nucleotide is made up of each nucleotide is made up of phosphate, sugar, nitrogenous base. Then you have the nitrogenous bases are adenine, thymine, guanine, cytosine. Adenine is complementary to thymine, and guanine is, so you explain what is the structure of DNA. If they ask you, describe yourself, you tell us you are big, you are small, you are dark skin, light skin. What you see, that's what they want. Please, you talk about anything you know about DNA. And these questions, make sure that you just give everything you know. Don't just list and then say, maybe this is wrong, this is right. No, you just. Someone is saying, I speak too fast. No, the reason why I'm speaking too fast is I'm assuming that you guys, you know this DNA. I want to put more emphasis on these other topics like evolution and the genetics. However, if you see that I'm too fast, let me know. If okay, let me just reduce the speed. I'm sorry. I'm sorry. You call yourself who? Cupcake. Cupcaker. All right. And the B-boy. Right. Hmm. You're saying so describe that structure. I think I'm done there. What is the function of DNA? Basically, it carries genes for inheritance. Yes, and also makes it provide information for formation of a protein. Is it clear? Yes. All right. Know the functions of DNA. Now, when it comes to base calculations, guys, I think you know the base calculation. We have like 2 ways or 3 ways in which we can bring this question. If adenine is 20, I'm just giving what is the percentage of thymine? Or what is the ratio of thymine to cytosine? Let me just do like that. I'm looking for the ratio of this to this. So if adenine is 20, adenine goes with thymine, cytosine goes with guanine. Whereby if adenine is 20, obviously this is 20. So it makes me to have 40. So 40, remember it's percentage. So minus 40, so I'm gonna get 60, and then 60 is for these two, meaning that if I have 60 divided by 2, I'm gonna have 30 here, I'm gonna have 30 here. So since I've got these two, then automatically I will come here and say this is thymine is 20, and then cytosine is 30. Then I divide by 10 so that I make them to be simple, so that it gets into 2 by 3. So that becomes the ratio of thymine to cytosine. I'm just doing this so that you are able to remember what you did before. All right, we go to the next question. When it comes to DNA replication, please don't go without knowing these questions. What is DNA replication? Process where DNA makes the exact copy of itself. When does it occur? It occurs during interphase or before the cell divides. Where does it occur? It occurs in the nucleus. Why does it occur? This is the importance. Why is it happening? This is the what? The importance. Palisa, you're saying I should start again. Palisa, I can't start again. I've just started it. Just continue. You replay the video at the end. All right, thank you so much. They're saying that double the chromosome number. It doubles the chromosome. It helps to double the what? The chromosome number. So meaning that the results in this information. So basically, why do we double the chromosome number? So that when it undergoes meiosis, we are able to to have each cell is supposed to have same number of what? Chromosome. Sir, do the calculation. Again. Renato. Yeah. You are seeing the calculation for the first. Oh my God. Hi. Unless just to give you a different example. So you have to know these questions. You understand? You have to know these questions. You can't go without knowing. Some of the questions they bring in the DNA describe the structure of DNA, DNA replication, transcription, translation. Those are the long questions they can bring in the DNA. All right. Guys, please take note of the information you want me to talk about maybe at the end. Ne? Yes. All right. Then, so when they say, how does it occur? Let me tell you something, guys. When they say, how does it occur? Yes, I'll do the genetics. Don't stress about it. How does it occur? You describe the process of DNA replication. But sometimes we can bring something. Let me see if I did prepare something like this. For example, like this. This question is saying that the diagram below represents the process that occurs during protein synthesis. Identify X, identify Z. But at here they say that name and describe the process. Sometimes they don't say name the process. They just say describe the process. So meaning that when you name the process, we give you marks. And then when you describe it, we give you marks. Even if they have not said that name, as long as they have not identified that process, make sure that you identify it and then you describe it. Is it clear? So it's the same thing here. If they have not given you the process of DNA replication, but they've brought a diagram and that diagram is showing that DNA replication is taking place. Is it clear? Is the one which takes place, is one which you're gonna describe. So you have to first identify it and then you're able to describe it. Therefore, you'll be getting those marks. Sir, will you do genetics? Definitely. I will do so. So that's why I'm rushing for this other content so that you are able to do genetics. All right, I'm done with that. Then we are going to protein synthesis. Before I go to protein synthesis, they can ask you a question that give the difference between the nucleotide of DNA and the nucleotide of RNA. In this case, they are looking at the nucleotide of which the phosphate is the same, but the sugar and the nitrogenous bases are not the same. So here you say deoxyribose sugar for DNA and the ribose sugar for RNA, and then here you say thymine for DNA and you say uracil for RNA. That is they are not talking about the difference between DNA and RNA. They are talking about the nucleotide of DNA and the nucleotide of RNA. I think, guys, I'm clear. Let me know if the sound is 100% right. Yes, because we had some technical issues. Ne? Yeah, so if we are 100% right, then let's continue. All right. Here is protein synthesis. What do you need to know about protein synthesis? You need to know how to label what is happening in transcription and what is happening in translation. Yes. Transcription means what? Formation of messenger. They asked that question some time back and people were failing it. Translation means what? Formation of a polypeptide chain. Please don't confuse polypeptide chain with the DNA, with the protein. A polypeptide chain means less than 50 amino acid. If it is above 50 amino acid, we call it a protein. Is it clear? Yes. So now let's label. Let's label. Let's label. What is W? W is what? Let's label together. However much you are not writing it, but before I say it, you are able to give me the answer. Ne? Like say it in your heart, or you can say it loudly where you are sitting, so that when I say the answer, at least you get it right. If you get it wrong, then it means that you need to know it better. All right, let's continue. What is W? W, DNA molecule. Yes. What is Y? What is Y? Y is amino acid. What is Z? Z is the ribosome. And then what is X? X is the messenger RNA. Good. What is this? Okay, now since this one is not labeled here, now you're gonna tell me from the comment section that what is this. I'm not going to say it. I will first see the answer there, then I will continue. What is this? What is O and what is P? Ne? So we have O and then we have P. We have three bases on DNA. We call them code. We have three bases on messenger. We call them codon. We have three bases here. We call them anticodon. Someone is saying polypeptide chain is O, and then is saying peptide bond, and then okay. I've got you. Thank you so much. All right. Guys, O, thank you so much. O is polypeptide chain. Let me show you what is a polypeptide bond. Let me zoom this out. Let me show you what is a this is the polypeptide. This is a peptide bond. We don't have polypeptide bond. No, we have a peptide bond. You see these lines? These are the polypeptide bonds. This circle is an amino acid. So the whole structure is polypeptide chain. Is it clear? I think I'm clear there. Thank you. Thank you so much, guys, who gave me the answers. Yeah, at least it shows that you are learning something. All right. I think I'm done. So if they ask you to describe the process that takes place in the nucleus, then automatically you describe transcription in terms of synthesis. So this is the nuclear membrane, while these small holes are the nuclear pores. Yes. Can we go? Yes. So you go to transcription, DNA double helix unwind. DNA double helix unwind. The double helix DNA unwind is the same thing. Weak hydrogen bonds break to form two separate. The word separate must be there. To form two, don't say to form separate strands. How many are they? To form 2 separate strands. Then we are saying that each strand is acting as a template when it is about DNA replication. But when it is about transcription, you say one strand is acting as a template using free floating DNA nucleotide if you're talking about DNA replication. But if you're talking about transcription, you are forming a messenger. Therefore, we use using free messenger RNA nucleotide. Is it clear? Yes. From the nucleoplasm. A messenger RNA is which is complementary to DNA is formed. Is it clear? Whereby adenine goes pairs. Uh, 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 um, let me be clear here. It's better to say that adenine is complementary to, use the word complementary to. Why? Because here we talked about complementary. Adenine is complementary to uracil, and guanine or cytosine is complementary to guanine. Now, messenger RNA with coded information. This is a marking point. Whatever you see in red is a marking point. Coded information, yes, for protein synthesis, and then now it moves out of the nucleus through the nuclear pore to the ribosome in the cytoplasm. So basically, guys, you know this. So I don't need to repeat this over and over and over and over again. But I'll show you where you get sucked by this topic. I'm coming there. Sir, did he do meiosis? Not yet. Not yet. I'm going to go there. Yes. 20 minutes ago. What does that? Oh, all right, guys. Hey, guys, just start. Just as long as you are here, let's study together. All right. Each here you talk about. Now you see this. That is a polypeptide chain. You see this A. That is a peptide bond. You see this. That is amino acid. You see this. That is transfer RNA with the amino acid. Transfer RNA. You see this. That is anticodon. You see this. That is amino acid. So we say that when the transfer RNA anticodon matches with the codon of messenger, this transfer RNA will bring this required amino acid to the ribosome because this structure is the ribosome. Is it clear? Yes. Now, once it brings it here, then the amino acids are joined together by the peptide bond to form a required protein. That's it. So, guys, I think you got it right. So that is it. So each transfer RNA carries amino acid. When the amino anticodon of the transfer RNA matches the codon of the messenger, the transfer RNA brings the required amino acid to the ribosome. Then we are saying that now amino acids are attached by peptide bond to form a required protein. Simple. Guys, don't make a mistake to go when you don't know how to label this. You can't. You can't. Mutations on DNA. Yeah, we're gonna talk about it. But before we talk about it, answer for me this. Let's answer them together. All right. I'm not. Answer for me what is X and answer for me what is Y. I want to know what is X and what is Y. So as you're answering, I want to give you something here. Uh-huh. Okay, we are starting. What is X, guys? Hey, I'm not seeing your answers. What is X? Okay, I'm not going to answer X. What is X and what is Z? What is X and what is Y? So Z is messenger RNA. Uh-huh. Then give the nitrogenous base sequence of DNA triplet complementary to middle codon. So now when we look at middle codon, now I've robbed off. I don't see the answers. Where are you? Okay, X is the amino acid. Don't be scared to give me the answer. And then Y is the anticodons. You understand? Because they're looking at these three bases. So they are anticodon. Now you know the answer you are trying to give me. All right. So this is amino acid is anticodon. They're saying that give the nitrogenous base sequence of DNA based triplet complementary to the middle codon on the Z. On the Z, where is Z? Yes. So meaning the middle codon, meaning that they're looking for this, of which they're saying that the DNA trip, this is a messenger, so it's gonna be A goes with the T, and then U goes with the A, and then C. So that's how it's gonna be. Is it clear? Yeah. Then they're saying that describe name DNA based trip. Okay, we're done with that. Describe, name and describe the process that occurs in the diagram above. So here we are talking about translation. So you describe translation. I think I'm done with that. Let's go to the most difficult part. Imitation. Imitation. All right. We have two kinds of mutation. There is a mutation which results in a change and a mutation which doesn't result in a change. And how do we ask them in the question? Then there is another question which we ask about this mutation. Guys, DNA profiling, please don't go without knowing it because I have not gone through it. I expected it to be, you know it. All right. What will happen if a mutation changes the base triplet 1, yes, from GT, from ATG to ATA? Guys, look critically. You see, even I've reduced the voice so that you can understand it better. Ne? Look critically. I repeat. All right. Thank you so much for those who participated. Now, we are saying that uh, when they give you this information, first check what is here and what is here. Oh, I forgot to talk about the other thing. Where is it? Yeah, because it's going to help me to answer that. Codon, these are three bases found on the DNA. Code, these are three bases found on DNA. Codon, these are three bases found on messenger RNA. Anticodons, these are three bases found on the transfer RNA. So, how many amino acids? Yes, are in 20 anticodon. Remember, each anticodon, look at it here. Each anticodon, because we say that Y is anticodon, né? Each anticodon codes for one amino acid. If they have 20 amino acids, automatically there will be 20 anticodons. But if I have 20 amino acids, how many bases will be there? It will be 20 times 3 because each anticodon has 3 bases. So if I have 20 amino acids, how many anticodons do I have? It will be 20. But how many bases will I have? It will be 20 times 3. Why 20? Because each anticodon codes for the same amino acid. One anticodon is equal to 1 amino acid. 20 amino acids are equal to 20 anticodons. But each anticodon is equal to 3 bases. So if I have 20 amino acids, it means that I have 20 anticodons. Each anticodon is 3, so times 3, which gives me 60 bases. Is it clear? All right. Sometimes we bring these questions where they ask you this and you go off. Now, since I know that, now we come back here. I said that when they give you this table, first identify what is here and what is here. What is here because of the T, it means that this is DNA. However much, they have mentioned it here. So you have DNA, but here you have codons, meaning that you can't read from here and you bring it here. What you need to do is you have to convert this and then you read it there. So if you do that, yes, if you do that, then it means that now this you will be able to convert. For example, what they give us is ATG. So ATG. Sometimes when you go direct, you'll find the answer there, but that answer will be wrong. You understand? You have to convert it into ATG is gonna be A goes with U, and then T goes with A, G goes with a C. So you look for this here. UAC is tyrosine. Oh, even I pointed it direct, is tyrosine. Now you come here. You look at this, the one which has changed to ATA. You convert it into UAU. UAU is what? UAU against tyrosine. So meaning that this, thank you. This is tyrosine, and also this is tyrosine. So it means that there will be no change in the protein, however much there is a change in the sequence of DNA. So how do you write an exam? You tell us that this changed to this and this changed to this, of which these two they code for the same amino acid, which is tyrosine. You have to mention this and you have to mention this. This changed to this and this this changed to this. You understand? And this codes for tyrosine, and this codes for what? For tyrosine too, which does not change the protein. Why? Because it is the same amino acid. What about when there is a change? What about when there is a change? Okay, right. What about when I have A, and this TTA changed to TTT, of which TTA, this one is gonna be AAU, it's gonna be U, it's gonna be AAA. Yes, there is no mutation, there is no change. Boy B, there was a mutation, but this mutation had no effect because the mutation is not the change in the protein. No, the mutation is the change in the base sequence. It's called gene mutation. So if the base sequence have changed, then automatically this guy confusing me. Who is confusing me? Okay, automatically there was a mutation, because a mutation means change in the sequence of DNA. bases, or the change of the structure of chromosome, if it is chromosomal mutation. But if it is change in the sequence or the bases, then we call it gene mutation. So if this mutation, if the change leads to the same amino acid, therefore they won't be effect on the protein. But if the change in the base sequence leads to a different amino acid, then there will be a change in the protein. Is it clear? I told you this is you call it a devil. I don't know why you call it a devil. Yeah. So now, base triplet number 4 changed this one. Changed from this, and then it went, meaning that this A was replaced by T. Cool. So when I change this to this, why do I need to change it? Because I can't read this direct here. Why? Because this is codon. So I have to change this DNA into anticodon. Sorry, into codon. So now AAU is what? AAU is valine, isn't it? And then now TTT, when I convert it into messenger, is going to be AAA, which is AAA. AAA is proline. You see that a different amino acid is formed. So to get those 3 marks, you have to tell us that, yes, this changed to this. This changed to this, of which this code is for valine and this code is for proline. Therefore, different amino acids will be coded for, and then a different protein will be formed. So in other words, let me give it a summary. When you convert this and you see that it's the same amino acid, then you tell us those amino acids, ne? Then you tell us that there is no effect on the protein. But if you convert them and you see that the first one codes for one amino acid and the second one codes for the second amino acid or another amino acid, then it means that there will be an effect on the protein. Is it clear? So please don't go to the paper without knowing this. However, there is sometimes this question they ask you. They say that what will happen if the base sequence of DNA is changed? What will happen to the protein if the base sequence of DNA is changed? So that question is open. So you tell us that change in the DNA sequence will result in a change in the sequence of messenger RNA, which will result in a different sequence of transfer RNA bringing amino acids, isn't it? Then which results a different protein. Or if they ask you from messenger RNA, then you start from messenger. A change in the sequence of bases of messenger will result in a different sequence of transfer RNA bringing amino acid, and then it will result in a different protein. Yeah. I think I'm done with that. Please don't go without knowing that. Lastly, when they bring here, they say it's DNA, and here they put anticodon. What do I do? You change this DNA into messenger, and then you change the messenger into transfer RNA, because you don't have anticodons. If this is anticodon, because you don't have anticodon on messenger, so it means that you convert it 2 times. Yes, you convert it 2 times so that you're able to read what is here in the table. Please assure me that we have the 27 marks now. And you know our method, that by the time we are done, we will be done with the reading only to practice, and you pass the paper by smashing it. Meiosis. you have any challenge, let me know. Meiosis, you know the functions, why is meiosis needed? Basically, meiosis is needed for almost 4 reasons. Even if you know 2, is better. Number 1, to produce haploid gametes. Number 2, genetic variation. It's better to say genetic variation. Don't say variation. Because variation can be genetical or it can be mm, mm, environmental. When you go, my girls, when you go to to ShopRite and then you buy gentle, you could eat gentle magic, you change your skin to be lighter, ne? That is a variation before and after. You understand? But that's not genetical because the kid you produce will be like your original color. You understand? So that's why we hear when you say variation, it's better to say genetic variation. All right. It maintains the chromosome number. Those are functions of meiosis. The first one, haploid gametes. The second one, maintaining the chromosome number from generation to generation. The third one, you can talk about you said formation of haploid gametes, ne? Yeah. Genetic variation, haploid gametes, and then maintain the chromosome number from generation to generation. All right, let's continue. Where does meiosis occur? In animals. In animals, ovary to produce ovum. That's female. Testis, testis. This is one, this is many. So testis are in male to produce spermatozoa. Cool. In plants, ovary produce ovules, anthers to produce pollen grains. Think I'm clear there. You confuse yourself by unreplicated chromosomes and chromatids. Chromatids, we only call them chromatids if the chromosome, these are linked together. If they are separated, they are not chromatids. We call them unreplicated chromosome before DNA replication. Unreplicated chromosome. You see? Unreplicated chromosome. But once DNA replication occurs, they become like this. That's why you call them replicated chromosomes. Replicated chromosomes. Well, here we call them unreplicated chromosomes. You think I'm clear there? Huh? We continue to the next question, or karyotype. When you talk about a karyo means nucleus. Which type of nucleus do you have? Karyotype. So that's the meaning. So then when they ask you to describe the structure of a karyotype, it can come in 6 marks. Describe the structure of a karyotype, 6 marks. This one means that you tell us the arrangement of chromosomes. Karyotype, you are telling us arrangement of the chromosomes. How many are they? What are they in a cell? So you arrange the chromosome from pair number 1 to pair number 22. We call them autosomes. 23rd pair, we call gonosome. 23rd pair has X and Y. If it's X and X is a male, is a female. If X and Y is a male, and Y is shorter than X, or X is longer than Y. So you just describe what is happening there. Is it clear? But when they ask you how many chromosomes do you see there, then here you have to look at it. Don't rush, because rushing will never bring anything good. Sometimes we make this one 2 or 3, like that, and then ask you how many autosomes are there. You know that autosomes is from 1 until 22, isn't it? Then we ask you how many autosomes are there. But there is another one here, because when you know that it's 21 pairs, therefore is 40, 42. Did I say 21 pairs? It's 22 pairs. It's 44. Then I say, yeah, I know it. Can't there is something there? They have put another one there. Or this one is not there. You see? So please count them. Count them. And sometimes they bring it when it is not about a human karyotype. It would be something else. So please, please, please follow based on what is given. All right. Karyotype. When it comes to these phases, in prophase, you need to know what is crossing over. Please, don't go to the paper without knowing crossing over, unless you are burying yourself when you are still alive. The word homologous chromosomes aligning together is a key point. Is it clear? Then the word chromatids of these homologous chromosomes. One chromatid from one homologous chromosome, or each homologous chromosome overlap, is a marking point. But where do they overlap from? At a point called chiasma. Is it clear? Is a marking point. At a point called chiasma. So we give you another mark there. Then the chromosomes will have two new combinations, meaning that each from these homologous what? Chromosome, meaning that this one will have one part of this and this one will have one part of this. That is the function of crossing over. Basically, these chromosomes are not genetically identical. No. They are this, they call them homologous. They are the same, but they are not identical. So does it mean that because you are Zulu and your father is a Zulu or your mother is a Zulu, does it make you to be identical? No. So because they are the same family doesn't mean make them to be like identical. They have the same location of genes, but they don't have the same genes. Like here, you see, here it is capital letters, while here is small letters, but it's the same type of gene. That's the meaning. So when the crossing over takes place on top and they ask you to draw, when crossing over asks you like on top, so the variation must be on top. When it's starting from here to there, it must also show that. So don't just draw because you want to draw or you want to finish. You will get a 0 with big eyes. I'm telling you. So I think I'm done with that. Let me know if you have a challenge, né? Yeah, because I'm here to smash the challenges. Guys, what happens in metaphase 1? Metaphase 1, homologous chromosome line up at the equator randomly in 2 rows. Randomly in 2 rows. I think I'm clear there. I don't want to emphasize. It must be checked randomly. We don't know whether this chromosome is going to be here or is going to be there. Or this one is going to be this side. And the moment they swap, those are different chromosomes. Is it clear? Yes. So they align along the equator in homologous pairs. Is it clear? And then in anaphase, the homologous chromosome, they are pulled to opposite poles, meaning that they separate. Yes? Pulled to opposite poles, meaning that they're what? They are separated. So don't go to the paper without knowing. Process of crossing over, metaphase, anaphase. You can fail to know others, but you can't fail to know those ones. Cytokinesis. Cytokinesis division. Cyto means cytoplasm kinesis. Basically using this so-called energy. So the division of the nucleus, we call it cytokinesis. Hey, did I say nucleus? Division of the cytoplasm. So please, we talked about this, but you need to tell me before I go there. What is non-disjunction? What is non-disjunction? Failure of the chromosomes to separate during metaphase is what called non-disjunction. And this non-disjunction is going to lead to Down's syndrome. Down's syndrome. So we call it non-disjunction, and then it's going to lead to Down's syndrome. I think I'm clear there. When they ask you the difference between meiosis 1 and meiosis 2, you don't need to suffer. Just go to meiosis each phase, then you tell us. In prophase 1, we have crossing over. In prophase 2, we have no crossing over. In metaphase 1, homologous chromosomes align along the equator. In meiosis 2, individual chromosomes align along the equator. You see? In meiosis 1, under anaphase, chromosomes, homologous chromosome moved opposite poles. Under the other one, we say chromatids moved opposite poles. Is it clear? In meiosis 1, you result in two daughter cells. In meiosis 2, you result in 4 daughter cells. So you see we have a lot. You can go phase by phase, phase by phase. Someone is asking me, wait, isn't it anaphase 1? What is it? What is anaphase 1? What do you mean by anaphase 1? I don't know what you are saying. Anaphase 1. Random, you mean non-disjunction. Non-disjunction can occur. Difference. Sorry, sir. What are the different? Okay, I'm going to talk about it. Non-disjunction occur in anaphase 1 and anaphase 2. In anaphase 1 and anaphase 2. Yes. And sometimes they bring this cell and then they ask you that you explain what is happening based on this anaphase 1. Is it for anaphase 1 or it is for anaphase 2? In the difference between meiosis, what are some of the similarities? Similarities between meiosis and mitosis, both they require the nuclear, meaning that there is nuclear karyokinesis, there is cytokinesis. You see? Yes. Both involves the same phases. Interphase, prophase, metaphase, anaphase, telophase. Both there is involvement of DNA replication. So that's it. And then the difference, actually, meiosis 2 looks like mitosis. You understand? So basically, in meiosis, there is variation. In mitosis, there is no variation. In meiosis, you result in 2 daughter cells. In mitosis, you result in 2 daughter cells. In meiosis, you result in 4 daughter cells. In meiosis, you talk about having half the chromosome number. In mitosis, you don't have the chromosome, meaning that the chromosome number are what? Are maintained. So, guys, you need to know this. There is no crossing over, there is crossing over. It is easy. Guys, can I tell you something? Before we go to genetics. You need to read loudly if it is possible. When you are reading, make sure that you read loudly if it is possible. Make sure you read loudly. Yeah. So we are going to genetics, né? But before we go to genetics, let's just do a few questions here. Just 1, 2, and then yeah. All right. Shabalala, what are you saying? All right. The diagram below represents a chromosome of human somatic cell of two individuals. I found out that people confuse what is called orosomes and gonosomes and somatic cells. Guys, when you talk about orosomes, orosomes, these someone say restart the live. Okay, guys, can you hear me? Let me know if you can hear me. Ne? Because I think there is some technical issues. It was, I think, some technical issue with the internet, but I think now they have resolved it. Ne? Thank you so much for making me know that now you can hear me, and thank you so much, guys. Those who told me that uh, uh, you can't hear me. Ne? Yeah, thank you so much. All right, now let's continue back. Let's go back to our discussion. Let's go back to our discussion. So I was saying that. Now I'm lost. I don't know where I stopped with you. Okay, let me just do like this so that maybe we are able to be at the same page. All right, I'm saying that here is a question. Ne? These diagrams, yeah, I was saying that some people they confuse what you call the somatic cell, the gonosomes, and the orosomes. Orosomes they go with the somatic cell. These somatic cells are cells, are body cells. You understand? They are body cells, meaning that these cells they have the chromosomes inside. So when they say somatic cells, these are body cells. Good. Name the specific type of chromosomes numbered 1 to 22. You see, how do you call this up to there? So we call them orosomes. Each of the pair shown in the is a homologous pair of chromosomes. State the origin of the chromosomes in the homologous pair during the zygote. Obviously, one will come from the mother and one will come from the father. What are stem cells? We will talk about them, né? Because that is genetics, and genetics is our next topic. We are just finalizing with the meiosis, and then they are saying that uh, what is it? State the origin. One chromosome comes from the father, and one chromosome comes from the mother. Now here is another question. List 3 characteristics chromosomes in a homologous pair have in common. If these are homologous chromosomes, what do they have in pair? We saw it when we were discussing about, yes, one from the father, one from the mother. We saw it. We say that first of all, they have the same size. Ne? They have the same shape, same size. If you look at them, same size. Ne? They have the same shape. Ne? They have the same locus or loci for the genes. They have the same size, same height, same shape, yes, and then same location of, not same location of chromosomes. No, not the same location of chromosomes, same location of genes on the chromosome. You understand? Because it's already a chromosome. Thank you so much. Thank you. Thank you. All right. Explain one observable reason. Explain one observable reason why the two individuals are not identical twins. So what do you say when it comes to that? So the observable reason you can give here is they're not identical. Why? Because this is a female, this is a male. So these are two different chromosomes. Sorry, individuals. You cannot have identical twins who are different sex. You understand? You cannot have identical chromosomes which are different sex. So it cannot happen that way. So this one, how many marks? Different sex, whereby individual one is a female, is a male, while individual two is a female, because of this same size of shape, same size of chromosome, and then here different sizes. Yes, thank you so much. We got the next question. Do you have any question? Okay, that is it. Let's go to genetics. There are some things I need to emphasize here. Ne? Terminologies, you need to do that. When it comes to the format of writing a genetic cross, you not write phenotype, genotype, meiosis, fertilization. They need to be in the corresponding position. Is it clear? Oh, before I go there, just need to tell you this before I go to genetics. Guys, when it comes to meiosis, when they say that, how many chromosomes do you see? How many chromosomes are there in that cell? And this cell is showing you this. Let me just show you before I go back. You see these chromosomes, and then they ask you, how many chromosomes are there in that cell? Don't say 46 chromosomes, because this is not human cell. If they don't say human cell, please just say 1, 2, 3, 4. Four chromosomes. How many will there be at the end of meiosis? Because these are in pairs, therefore there will be 2 in each. How many chromosomes will there be in the same cell of human under the same phase? That's when, because this is meiosis 1, meaning that metaphase 1, yes, then you talk about 46 chromosomes, because they mentioned about human. But if they don't talk about human cell, please do not do that. Do not do that. They can ask you also, how many chromatids are found in this cell? So each chromosome has 2 chromatids. So it's 1, 2, 3, 4, 5, 6, 7, 8. So they're going to be 8 chromatids. Simple. Let's go back. I think that's what I wanted to talk about. Yes. So when it comes to this genetic cross, I say that please make sure that you write phenotype, genotype, meiosis, fertilization. You know why? Because we give a mark here, we combine this and this, we give you a mark, and then we combine this and this, we give a mark. So it is P1, F1, we give a mark. Meiosis and fertilization, we give another mark. So you go to the paper we have when you have 2 marks. I wonder those people who get 0. Even when you can't do this, no. If you see genetic cross, yes, P1, F1. If that is P1, F1. If it is second generation, then you say P2, F2. Is it clear? So phenotype, you get it from the question. They will say that Karabo who is anemic, married Joseph who is normal. So you say that Karabo anemic, Joseph normal. Crossed, you see, you understand. Then genotype, genotype, that's where the problem comes in. You need to find out whether it is homozygous or is heterozygous. If it is homozygous, then is it homozygous dominant or is it homozygous recessive? Someone saying that, am I still talking? Yes. Yes. Do you have any challenge there? So I think I'm clear there. So that's it. Now, when it comes to this, when you're marking, yes, we shall mark, we shall give you a tick here, we shall give you a tick here. We shall give you a ticket. Remember, this is from the question. So you have 3 marks, mahala. You understand? You have 3 marks, mahala. So now, since you have 3 marks, mahala, so now you have to find this one correct from the question, whether it is dominant or recessive. Once you get that, now, for example, let me show you another mistake students do here. You have capital T, small t, capital T, small t, for example. Then meiosis takes place. This one must be separated. This is capital T, this is small t. This is capital T, this is small t. Guys, please, 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 I beg you, don't write this one here and this one there. We shall call this one. How are we going to call it? We're going to call it a genotype. So there must be space in between. That's why I prefer to tell my kids that, you know what you guys do? What you should do? Number one, just capital T, put a comma, small t. Even if they're together like this, at least we shall know that there is a comma there. Or don't make them to be like the beach. You know, at the beach, what they do, they people are naked. They only have, they are covering a specific part. So don't leave them like that. Just dress them up. Put them on in circles. Dress them up. Ne? Yes. Once you dress them up, even if they are too close, still we shall give you a mark because you have circled them, you have separated them. I think I'm clear there. Then, right fertilization, now start crossing. If you don't cross like this, use a Punnett square. You understand? So the Punnett square, bring this here, and then you say capital T, small t. Then you bring this here, capital T, small t. So we shall give you a mark there, we shall give you a mark there, we shall give you a mark there. Then you cross capital T, capital T, capital T, small t, capital T, small t, small t, small t. We give you another mark there. Are you seeing? Then you write the genotype, the ratio, the phenotype, the ratio, depending on what they have asked you. But I prefer write everything because whether you have written it or not, we won't cross you. But if you have not written it and they wanted it, you lose a mark. So just write everything you know about it. That is the first point. So the second point, write everything. Why am I saying that? When you say P1, don't wait to write F1. Start with the P1, F1, because sometimes you write P1 and you forget F1. Please, 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 please. Yes. All right. Another thing which you do, you guys, when you are doing this genetic cross and makes you lose the marks is, now you might see your teacher is a little bit sympathetic to you, but when it comes to the marking, they will not be sympathetic to you because they won't know you. And no one will be willing to lose, let me say, the job they have signed for. So, meaning that please do things which are right. So, for example, you write here capital T, small t, capital T, small t, and then you start crossing. You know what you cross? Let me just write it here. Capital T, small t, capital T, small t. Then you cross like this. Capital T, small t. You cross like this. Capital T, small t. You cross. You cross like this. See? Capital T, small t. Cross. There's a way how you cross. You cross like this. There's a way how you cross. You cross like this. Then you say capital small t, small t. So we ask you, what is this? What is that thing? Where is this? Is it this or this? Where is this one? So I prefer, if you don't know, it's better use a Punnett square method. So it's better that way. Is it clear? Yes. Lastly, about this. When they say that P1, F1 is a compulsory mark, I'm just giving an example. And the meiosis and fertilization is another compulsory mark. 2 compulsory mark. And then they've given you 6 marks. In most cases, the genetic cross is 6 marks. So what they do is They first subtract the compulsory mark and they give you any other 4. If you don't get this right and you don't get this right, you get 6 here, they will give you 4 out of 6 because you have failed to score a compulsory mark. So that's why I'm saying that write everything, tau fail. Write everything, fail. Yeah. Then once you do that, you'll be able to succeed. All right. Let's continue. Dominances. When you talk about complete dominancy, incomplete dominancy, codominancy, here we are talking about Mendel's law of dominance. In Mendel's law of dominance, when you are talking about Mendel's law of dominance, it means that one allele is completely dominant over the other in a heterozygous state. When you have a capital letter, small letter cannot speak. You understand? If there our president Ramaphosa is the president, if it is if he's available, you cannot the vice president. Natural selection is very challenging. Prepare more questions. All right, Karabo Karo. All right. The vice president cannot speak if the president is available. That's the meaning. So when a dominant allele is present, a recessive allele cannot what? Cannot do anything. Is it clear? Yes. Sorry. So this is not my concern. My concern is about the pedigrees. I know these ones you can kill them, but when it comes to pedigrees, sometimes you mess up. And you know yourself. All right. We will sort it out. So someone asked me about that question. So now look, when this is complete dominance. You have a red, you cross to the white, you produce the red. You produce one of the colors. Then it is complete dominance. If they bring such a question, please know. All right. Thank you so much. I hope now you can hear me. Today I think I've been with the problem with the network. Right? Network has been a very big problem. All right. I hope now we are together. Can we continue? I hope you are hearing me now properly. All right. And we continue to the next question. We continue to the next question. Then when you cross red with white and you produce an intermediate, that is incomplete dominance. One of the allele, neither of the allele is completely dominant over the other. And in this case, we use capital letters. Capital R for red, capital W for white. When you cross the two, you produce capital R, capital W, which means pink. That is incomplete dominancy. When you produce a mixture, there is no difference between incomplete dominancy and codominancy when it goes to the crossing. It is the same. Is it clear? It is the same. Still, it's going to be capital R, capital, for example, W, and then you produce capital R, capital W. So it's the same style. But the only thing which helps us to know is the phenotype. When you produce a mixture of the two colors, then it becomes what? It becomes codominance, which means codominance means together. The two colors are existing at the same time. Are we together? Rivonia, are we together? All right. Rivonia, you let me know, eh? Yeah. If you have a challenge. Sex determination. Here, when you talk about sex determination, you just describe basically a karyotype. Nothing else. 22 pairs of chromosome in female autosome. From 1 to 22 is autosome. The 23rd pair is gonosome. When X matches with when X of the female of the ovum fertilized fertilizes or fertilized by the X of the sperm, you produce X and X, which is a female. When the X of the ovum is fertilized by Y of the sperm, it becomes X and Y, which is a male. We have 50% chance of having kids who are female and 50% chance of kids who are male. So that is sex determination. They can ask you to describe it in words or describe it in terms of what? In terms of genes or genetic cross. Yeah. Aha. Pedigree is starting now. Pedigree is starting. Call your sisters, your brothers, your uncles, your what, whoever is having a challenge here. Please take note. If you have not taken a drink, take it. Relax. No one must disturb you. We start the pedigree. Okay. Sex determination. Sex determination means sex-linked genes. These are genes which are carried on X chromosome or sex chromosome. You understand? Meaning that you only find it in that sex. So if it is the disorder, then it means that these are disorder caused by genes which are carried on X chromosomes. So examples is hemophilia, color blindness. Night blindness is also an example, but is not examinable. You understand? Yes, but I don't think if they ask you and you write it, they will cross it. Anyway, it's better to write what is being asked. All right. Should I start with the degree first? Okay, let me just highlight this. Hey, I want answers now. How many genes control blood group? Write for me how many genes control blood group. Ibafetu, write for me how many genes control blood group. And then, yeah, then I continue. So, yes, how many genes control blood group? Let me know. As I'm preparing for you here, questions. Yes, this question will work. Okay. I don't see the answer. 3. Okay, thank you so much. Any other answer or the same answer? Yeah. Sorry, 1. Hey, you are giving answer and then you are crossing. Hey, people are saying, yeah. Hey, people, you must tell me. Ne? Okay, 3, 1. All right. How many genes control blood group? How many genes control blood group? Hey, why writing? Guys, it is 1 gene. 1 gene control blood group. Thank you so much, guys. Which gene is that? Gene for blood group. Hey, do you have any other gene? It's gene for blood group. Ne? How many alleles? There are 3 alleles. That's why it's called multiple allele. 3 alleles. Allele for A, allele for B, and allele for O. And when you are writing, don't write it like this. We will cross it. You'll get a 0. Don't write it like this. O. And then you say it like that. You'll get a 0. However much some books, they allow it in other books, not if you look at the internet and the other books, they allow it, but it's wrong. When it comes here, we will cross it. So, how many alleles? 3. How many genes? 1. How many genotypes? There are 6. 6 of them. And how many phenotypes? There are 4 of them. Is it clear? Yes. Then, is it possible to have, I want yes or no. Is it possible to have, I hope those people who have gone for drinking water, you are back because I want to start the pedigree. Is it possible to have blood group A, a man of blood group A, marries a lady of blood group B and they produce a kid of blood group O? Is it possible? Tell me answer, yes or no. A man of blood group A, someone who is blood group A, marries someone who is blood group B, and then they produce a kid of blood group O. Is it possible? Is it possible? The answer, yes or no. Just want to see. Yes or no. Yes or no. Yes or no. Yes or no. Yes. Tell me what you know so that yes, yes or no. Yes or no. Yes or no. All right. Yes or no. Yes or no. Thank you. Thank you. Thank you. All right, guys. The answer is yes. It is possible. It is it is possible. Why? Look here. Look here on our distinction material. If you have blood group A and you are heterozygous, and blood group B, you are heterozygous, you can produce blood group O, whereby this small allele combined with this small allele, which gives me blood group O. You see? So it is possible for a person who is blood group and a person of blood group B to produce a kid of blood group O. That's why we say that it is not conclusive. You understand? It is not conclusive to use blood group to identify the paternity or the maternity of an individual. It is not conclusive because many people have the same type of blood. Is it clear? Yes, it is not conclusive. You will find out that your neighbor have blood group A and your what you have blood group A. So does it mean that your neighbor is your father or is your mother? No. What if you resemble the neighbor? I don't know. It's just you are resembling. Hey, they have never told you that when you resemble someone somewhere far. What if that person was your neighbor? Does it mean that something happened? Hey, guys, I'm not in there. I'm just saying, eh? Yeah. All right. Hey. Uh-huh. Hey. All right, let's start. We are saying that if they ask you that it is, you just tell them that it is not what? It is not conclusive to use blood group as a way of identifying the paternity of an individual. So these are the questions we talked about them. Yes, cool. Okay, before I go there, let me ask you. Okay, it's fine. I'll ask you the same time. So when it comes to dihybrid, guys, don't stress too much. Ne? When they ask you dihybrid, how do they ask you? Identify the alleles. No one is going to ask you to draw the whole thing. No. Identify the alleles, identify the genotypes, identify the phenotypes, complete the table, and then identify the gametes. So they give you the gametes to identify the genotype, or they give you the genotype to identify the gametes. Is it clear? For example, something like this. If I have T, small t, capital R, small r, and ask you what are the gametes there. Obviously, I'm gonna say this is gonna go with this, so I'm gonna say capital T, capital T, capital R. That's the first gamete. Now this one with this, gonna be capital T, small r. You circle them, ne? Then this with this is gonna be small t, capital R. Then this with this, small t, small r. You understand? Don't forget that Mendel said that the presence of one allele cannot influence the presence of another allele in a different location. That's why we start with a capital letter. But if they are different alleles, then automatically you keep the order. This is capital T, small r. But this one is small t, capital R. Because T is different from R. You understand? Yes. T is T, depending on which allele is it determining. Which character is it determining? And then R is not T. Therefore, that's why we keep the same this order of starting with the T. But if it was capital R small r. You can't write it like this, capital small r, capital R. No, because it is the same kind of gene. Therefore, you have to say capital R, small r. Is it clear? Yes. So, no one is gonna ask you about those what? Those those. So if they are like this, if it is, they can ask you the gametes from there, they can ask you the genotype there, they can ask you the percentage. If they ask you the percentage of those which are one seed, and then wrinkled, in this case, you have to add everything here. And then when you add everything, you get a total, for example, 300. I'm not saying that it's 300. Ne? So you say that 100 and then divide by 300 times 100 so that you're able to get the percentage. You see? Then you come here 209 300 times 100. So you get the percentage. So you understand? So if they ask you the percentage, please don't confuse the ratio with the probability. If they want a ratio, it must be a ratio. They want probability, yes, you can say the probability, or the percentage. Yes, you can do the what? The percentage. Let's go to the pedigree, guys. Oh my God. My students, listen carefully. We have 4 different pedigrees. Finally, 4 different pedigrees. The first pedigree is autosomal. Let me make it clear for you. The first pedigree is autosomal character, meaning that is carried on autosome chromosome, meaning that non-sex linked. I think now when I say it like that, it's clear. The second one is sex linked. You need to know that. The second category, so this is 1, this is 2. The second category is recessive allele or dominant allele. So is it caused by dominant allele or is it caused by recessive allele? Is it clear? So now here, these two, this is what it comes. If they say some or same, married, carabo, this is a boy, this is a girl or a woman, whatever. However much this is a boy, you see is a boy, and this is a girl. And they didn't say that this is caused by sex linked. Please don't use X and Y. Don't. Use the letters they have given you. For example, in this case, first identify if it is caused by dominant allele or is caused by recessive allele. If it is caused by dominant allele, I will say that capital D, whoever has capital D or capital D, small d, is caused by dominant allele. Meaning that whatever I have, if I have capital D, it will be that individual who is what? Shaded. Is a male, you see, but they have 2 letters because it is not sex linked. The female who, okay, the one which will be normal will have 2 recessive allele because the moment you have a dominant allele, that individual is sick. Is it clear? Now, if it is caused by a recessive allele, the one which is shaded, ne? Sorry. Someone is asking me, do you teach physical science? Yes. Yes, I teach physical science. All right. Now, the one which is shaded means if it is caused by recessive allele. If it's like this or it's like this, it means that these individuals are normal. Do you understand? Here it's caused by dominant allele. So these individuals are sick. So the one which is dominant, the one which are sick will be normal for recessive. And the one which are normal will be sick for dominant. So this one will be normal. And then this one will be affected. It is caused by recessive allele. All these ones will be affected. And then this one will be normal. It's caused by dominant allele. Hey guys, do you understand it? Before I go to the real example. So this is non-sex linked. If it's non-sex linked, whenever you say capital letter, that individual is sick or is affected if it is dominant allele, caused by dominant allele. If it is a recessive allele, the one which is shaded, the one which is capital letter, it is normal. The sick one or affected one will be recessive allele. When it comes to sex linked, dominant allele caused by dominant allele, the female will be that female will be sick. And that male will be sick. It's caused by dominant allele. And then this one is dominant allele because I have a capital letter somewhere. And then this one will be sick. Will be sick. You see, when it's caused by recessive allele, it is only one option for the person who is sick, meaning that the chances are fewer for this individual to be sick if it's caused by recessive allele. Let me go to the examples. I think I show you, you'll understand it better. Okay, here is the saying that this disorder, whatever, I don't care, yes, is caused by an allele. You see, this disorder damages the arteries supplying blood to the brain. The pedigree diagram shows the inheritance of this disorder in the family. Did they mention that this is sex linked? No, they didn't. Since they didn't, and is caused by a dominant allele, so what does it mean? It means that whoever is affected has a capital letter R. That's the meaning. Whoever is affected has a capital letter what? R. You see? This is not sex linked capital letter. You see? You see? This individual is sick. You see? These individuals are what? Sick. And the one who is normal, you see, it has a small letter. Why? Because it is caused by a dominant allele. Are we getting there now? Yes. Meaning that this individual is affected and is a male. Because it's not is caused by a recessive, is caused by a dominant allele and non-sex linked, I can't use X and Y. Therefore, this one is normal, therefore is obvious. So it's gonna be small r, small r. The same way we said it here, the normal is this recessive allele. You see? And then this one will either be capital R, or capital R, small r. Let's find out which one is A. They produced a person who is small r is not affected. Automatically, this one will be this. One R came from him, one R came from her, so that they're able to produce this kid. Therefore, this individual is capital R, small r, so that they're able to produce small r. It is caused by a dominant allele. So, shaded are dominant. Yes. In this case, because they say it is caused by a dominant allele, the shaded one, they are dominant. Is it clear, guys? The shaded one is caused by a dominant allele. Uh-oh. You guys? Okay, let me just finish up this, and then I have to break for some 10 minutes. Because I am a Muslim, I have to go for prayer. And I didn't see it, that it's already past time for my prayer, which we call Magharibi. All right. So we are saying that how many generations are represented in the diagram? So you count 1, 2, 3. Is it clear? Yes. So you count 1, 2, 3. Is it clear? Yes. So since you count them, so there are 3 generations. So it's cool. So boy tomorrow, obviously I'll pray for you. Then you're saying that yes, then we are saying that give the letter of unaffected male. Unaffected male. So the unaffected male are those ones which is a box, you see, and is shaded. Give the letters of unaffected male, isn't it? Unaffected male is only H. Yes. So the answer is H. Because this disorder is caused by a dominant allele. So definitely it's only H. Give the genotype of A. I think we talked about it. Yes. Talked about it because this A produced a baby who is not affected. And remember, this disorder is caused by a recessive allele. So it's caused by a dominant allele. So if you are not affected, it means that you have small letter R, small letter R. Yes. Then sorry to disturb you, sir. Promote the name. What name is that now? Jumero. I don't understand. Okay. All right. You mean my name? My name is M. Saidi. Just call me M. Saidi. All right. Musa Saidi. All right. They're saying that individual B, G, individual. So we say the genotype of individual A. We are done with that. Letters of individual not biological relative to individual A and B. When you come back here, individual A and B, the one which are not kids. Okay. This one, these two, they produced that and they produced that. You see? So this one just married this guy, and this guy just married this lady. So the answer is C and F. If this was like this, then automatically C would have been a child. But it's showing the marriage sign. Showing the what? The marriage, that this one they got married. So this A produced this, and this one produced that. I think I'm clear. Yes, someone is saying, yes, how do we find is a sex linked? We're gonna come there. I have an example for that. So now that is the first scenario for dominant allele. So if it is a recessive allele, now this one is for a recessive allele. You see it? It is a recessive allele. If it is not a recessive allele, they will mention it still. So if you see it here, you see that you have a capital letter, have a small letter, but they didn't want to mention it there, but here they say that why is it a recessive allele? Is it clear? Yes. So you guys, let me leave you with this question. Then I go for prayer. Yeah. Let me see if you can see it. You can flip your phone so that you're able to see it. I hope now you can see it. Yeah, 5 minutes. 10 minutes. 10 minutes. Yeah, you guys, please do for me those questions. Then I come back and then we smash it. But there is a key point there, Nana, who asked me. Please, this shows that it's a sex linked. So meaning that, and sex linked, not just sex linked caused by a dominant allele. You understand? So we have seen the non-sex linked caused by a dominant allele. Now we want to see sex linked caused by a dominant allele. You understand? So please do that for me. Yes. I'll be back in very few minutes. Back. Someone is saying, okay. Okay, any chicken see. Okay. Someone is saying, in the examination guideline, they didn't ask the difference between meiosis 1 and meiosis 2. Do we still need to? Yes, please. Can I tell you something? What else they didn't ask you and they asked it? Karyotype. Go check it. It's not there. They didn't talk about karyotype, but they ask it. Why? I don't know. So you need to know it. All right, guys. Let's start. We saw that are we there, guys? Are we there? All right. They're saying that this disorder is a sex-linked genetic disorder. It is caused by a dominant allele. This one, meaning that whenever you see it is not shaded, it's shaded, it must have that. If it is a female, it must have at least one G. Female, meaning that it's going to have either that or it's going to have that, depending on the offsprings they have produced. And if it is a male, it's going to have capital G, then it's going to have a small, it's going to have a Y. So that's what it means. So this disorder is a sex-linked genetic disorder. It is caused by a dominant allele, capital G. All right. So guys, I think now you see that when they say that, Rivonia, what do you want from me? Smash, you want to smash? All right, let's smash it. All right, we're saying that this disorder, this disorder, they have said that is caused by sex linked. They will always say that in the question that it is caused by sex linked. If they don't say it's caused by sex linked, please do not use capital letter. Sorry, do not use X and Y. So that one is done. So number 2, we have seen that whenever it is sex linked, whenever it's dominant allele, make sure that you use a capital letter. Whoever is shaded has a capital letter attached. Whoever is shaded has a capital letter is what? So if it is a male, obviously, then it's going to be just a Y next to it. If it is a female, we're going to have to look at the offsprings and we see whether this offsprings is the one who is affected or is not affected. Then we'll be able to know whether is it homozygous or it is heterozygous. Is it clear? Yes. So now let's find out on what they are saying. So it's better to write these genotypes and then you do the calculations you are doing. All right. They're saying that the diagram shows the inheritance in this family. Name the type of diagram shown above. Obviously, it's going to be, I don't know why I like obviously. It was a student who said it. And I said, stop using obvious. Now it is on me. Ish. It's not obvious. Yeah, so which type of diagram is it? It is phylogenetic tree. Is it clear? Yes. How many females are in this family? How many females? Okay. Let's find out how many females. Remember, female is a circle. Sometimes we bring how do they call it? Sometimes we bring the key, sometimes we don't bring a key. So whenever you see a circle, it is a what? It is a female. So it is 1, it is 2, it is 3, it is 4, it is 5, it is 6. There are 6 females. Is it clear? Yes. Mm-hmm. How many? How many females? How many males in F1 generation? Listen, guys. Here they're saying F1 generation. They are not asking in the first generation. No. F1 generation is different from the first generation. You are in F1 generation, but your parents are in first generation. I don't know whether I'm clear there. You, the kids, are in F1, the first filial generation. So, this is the first generation. This is the first filial generation. So, this is F1 generation. And these are the parents, they're in the. That's why you say that those are, daddy, those are your generation. Meaning that when and your daddy, you are in different generations. Is it clear? Yes. So they're saying how many in the F1 generation. Then we come back here and we look for that F1 generation. We have 1 and we have 2. So there are 2 males in that regard. They're saying that Herpes genotype. They want the Herpes genotype. What is Herpes? Genotype. Obviously, this Herpes is affected and is a female. So since it's affected, now we know, at least we know where we are starting from. First of all, is affected. First of all, is a female. Is a female. And is affected. And is caused by a dominant allele. You see, it's caused by a dominant allele. Meaning that this individual has a capital letter somewhere. But now we need to find out whether this capital letter, what are the kids of these two? We have a kid who is not affected at all. And this kid is a female, meaning that is having this. Remember, the one which has small letter are the one which are not affected. Therefore, how did she get this? One got from the father and one got from the mother, meaning that the mother is like that, is heterozygous for that condition. Yeah, is it clear, guys? Okay. Then they're saying that Anju and Pilusa have 4 children. Give the phenotype of their sons. Give the phenotype of their sons. They're saying Anju, and Pilusa have 4 children. So this is a male. Obviously, then we say that since it's a male, you write it there. Is capital, is a male who is affected, meaning that capital G. And then this is the female, which is capital, but this individual is normal, meaning that is having that. So meaning that they're saying the genotype of their sons is only talking about the phenotype of the sons. It's only talking about the sons. If the mother is normal, definitely all the sons will be normal. If one affected male, Okay, yeah, thank you. Yeah, so meaning that, here it means that they are looking for the males. Remember, males are like this. So this got from the father from here, meaning that they're gonna get from the mother. You see, so the genotype, the phenotype of the male is gonna be an affected male, or unaffected male. So that will be the genotype. Give the genotype of their sons. Yes, it's unaffected males. Then they're saying that explain your answer. So it means that you explain them that since Anju is unaffected female and Pilusa is affected male, therefore the males will inherit one recessive allele from Anju and a Y chromosome from, yes, resulting them to be male, and the recessive allele will be for this. You see? Yes. So that is it. You see that if you practice these questions, it is easy. It is easy. You are able to get the answers right. I wish you can pass and you have a beautiful life. Keep reading, you will pass. I know that. All right. So, guys, this is the first question. The second question on sex, on dominant allele. So we say that if it is dominant allele, please, the one which is affected is the one which is having a capital letter. If it is a dominant allele and is not sex linked, it means that still the one who is affected is dominant, is capital letter. But even if they talked about the male, please do not use X and Y. You only use X and Y if they say that it is sexy linked. Then we go to this question. What about when it is non-sex linked recessive? It's caused by a recessive allele. Now, the one which is shaded will be recessive allele, will be small letters. So they are saying that it means that this one is affected. This one is affected. This one is not affected. So it means that this one is not affected. This one is affected. But now, what is the genotype for this? Because they produced this kid who is affected automatically, then it means that this kid is going to be small t, kid number 5, because kid number 5 is affected, isn't it? So it means I got one recessive allele from there and one recessive allele from there, from the father, and one recessive allele from the mother. So when it is like that, then what does it mean? It means that this is it's normal, meaning that they have the one which is making them to be unaffected. Therefore, it's capital letter T and capital letter T. So automatically, these individuals are heterozygous, so that they're able to produce this individual who is homozygous recessive. Then they're saying that describe what is meant by autosomal disorder. It means that is a disorder whose genes are carried on autosomes. You understand? Yes, from pair number 1 to pair number 22. Yes, chromosomes. How many sons did the individual 1 and 2 have? Individual 1 and 2 have, they had one son. Why? Because this one is not a son. This is just someone who came to marry a beautiful girl. Number 4. Yes. Then they're saying that using individual 3, 4, and 7, explain why is it can be concluded that this disease is controlled by a recessive allele. Individual 3, 4, and 7. Yes, it can be concluded by that is because individual 3 and individual 4, they are normal. You understand? And they produced someone who is affected, meaning that they are heterozygous. This one is heterozygous, and also this one is heterozygous. So the recessive alleles combined and then produced individual who is affected. Meaning that, how do we say? You say that individual 3 and 4 are both normal. You understand? So they passed a recessive allele. Individual 3 passed, or individual 7 is affected with that disease. You understand? So those are 2 marks. So individual 7 obtain one recessive allele or small t from individual 4, and one recessive, say another mark, and one recessive allele from individual 3. You understand? Making the individual 7 to be affected. Simple. Will we get a mark if we say 3 and 4 are what? I don't see the. Okay, let me use this one. Will we get a mark if we say individual 3 and 4 are carriers? Individual 3 and 4 are carriers. Okay, let me check. Will you get a mark if they say individual 3 and 4 are carriers? You might, but it's better to say they are heterozygous. Yeah, it's better to say heterozygous. They are heterozygous. Uh-huh. So, guys, another example is a recessive allele but dominant, sorry, recessive allele but sexy linked. So, guys, we said that it can be dominant allele, non-sex linked, recessive allele, non-sex linked. Dominant allele, sex linked, dominant recessive allele, sex linked. You understand? So, you need to read. First of all, guys, make sure that you identify whether it's a recessive allele or it's a dominant allele. That's number one. Then identify whether it's sex linked or non-sex linked. Once you know that, you'll be able to answer questions which are concerning with this. Thank you so much. I think I'm winding up. I'm winding up with the genetics. So there are so many questions here. You can practice and practice and practice. Ne? Yes. What are stem cells? These are cells which have ability, uniqueness. They have ability, unique ability to develop and form other specialized cells. You understand? Have unique ability to form other specialized cells. Sources of stem cells, bone marrow. Yes, you need to know this one. At least you know two. Uses, they are basically used to replace the damaged cells or for, yes, damaged cells. So you have to know that. Cloning, guys, please. What you need to know about the. Did I talk about it? Did I remove it? Yes, it is here. Talk about it. Yes. About the genetic engineering. Whenever you talk about genetic engineering, guys, it must include the word manipulation. You manipulate. When you say engineering, you are manipulating the genes. Is it clear? Yes. So when you're engineering a building, you are manipulating the plan on the paper into a full structure. So, advantages: expensive, interfering with the nature, unforeseen long-term effects. Those ones, guys, ne? Yeah, you can just read them. Then now this one, cloning. When you have sheep A and sheep B, but you want to produce sheep 3, you understand? So what do? First of all, since I want sheep 3, this sheep 3 should resemble like sheep 1, then automatically what I need to do is I should use the genes of sheep 1. Yes, isn't it? So what do? I get a somatic cell, somatic cell, and then this somatic cell I remove the nucleus and I keep the nucleus. I throw this away. Then I go to the ovum of this sheep. Why do I use the ovum of this sheep? I remove the ovum, I remove the nucleus, I throw it away. I remain with the nucleus, sorry, the ovum structure, the outer structure, the membrane. Then after that, I combine the two with mild electric shock. And then I form a structure which looks like a zygote. Why do we say it looks like a zygote? Because we inserted this nucleus into the ovum, meaning the casing outside looks like the ovum. And because this cell is a somatic cell, therefore it's diploid. So it's like this thing is diploid and the casing is ovum. It's like when fertilization takes place, that's exactly how the ovum looks like. And then now I make this one to multiply by mitosis, and then forms a ball of cells, and then now I can separate those cells, and then I put them into different wombs to form embryos, and then it's able to produce this. How is the question being set here? The question is set in this regard, or in this way. The first question is this: why did we use the nucleus of this ship, not this ship? It's because we wanted the characteristics of ship one, isn't it? Which method do we use to combine the two? We use the mild electric shock. Why did we keep the shell of ship number two? Because we obtained an ovum. Therefore, the resultant structure should look like an ovum. Huh. Which ship one or two will the offspring look like? It will look like ship one. Give a reason for your answer, because we use the nucleus from ship one. You see? So you explain based on what is being. Then I can ask you other questions about biotechnology. Simple. Hmm. Welcome to evolution. Yes, I know that, guys. Evolution is a little bit simple. Why? Because you just recently did this. Someone saying finally. Yes, finally, we are there. We are winning. So, evolution here, I'm just going to, you need to know what is called a hypothesis. You need to know what is called a theory. A hypothesis is just a tentative um, um, statement which can be proven correct or wrong. It must be tested. Statement, a phenomena which is an inquiry which must be tested. Then a theory is an explanation of a phenomena which is accepted to be true and or is supported by facts, experiments, laws. It must is an explanation of something which has happened. Is it clear? Yes. So theory, I'm done. Evidences of this, yes, variation. We talked about the causes of. Did we talk about them? Yes, we talked about them. Cause of variation: random arrangement of chromosome during metaphase, random fertilization, crossing over, and then mutation. But if they say meiosis, the cause of variation based on meiosis, then you talk about random arrangement of chromosome during metaphase and crossing over. Sometimes mutation is also there, but you write it at the end. So variation is the difference among organisms of the same species. Difference among organisms of the same species. That's why I always ask people, are you sure your father is your father? Don't ask that to your mom. Ne? Are you sure your father is your father? Yes, your father could be your father, but because of variation, because of crossing over which took place during the formation of these gametes, that's why you don't resemble him or you don't resemble your sisters. The only problem is that one I asked him before. What if you don't resemble him but you resemble your neighbor? All right. I'm just asking. Okay, they're saying that yes, it could be your father, but because of the variation, the genetic variation which occurred crossing over, then it made you to be different. Yes. So what's a species? You need to know that definition is a group of organisms consist of similar individuals capable of interbreeding. Capable of interbreeding. You don't stop there. You say produce fertile offspring. You guys, please help me. Don't use viable anymore because we have removed it. To produce fertile offspring. A population is a group of organisms of the same species living together in a particular, yes, area. You understand? At the same time, that specific time, it should be living together. And they be able to interbreed. So that one, you know. Aha, here is some small confusion. Types of variation: continuous variation and discontinuous variation. Anything which we are born with and it is exactly the same way you are born with, then it follows under what you call discontinuous variation. Your blood type is discontinuous variation. If you are born blood group A, you are blood group A, you will die blood group A. You won't change. If you are born blood group B, you will die blood group B and your blood group B. You won't change. Is it clear? So whatever doesn't change from the time you are born until you die, then that is discontinuous variation. But the one which changes, for example, the size, the height, everything, we call it continuous variation. Yes, imagine, yes, the size of your head now and the size of your head before. It's not the same. The shoe size, it's not the same. So those are continuous variation. So meaning that we have a range of characters. While here we don't have intermediates. Here we have intermediates. We have that dark skinned, light skinned, too light skinned, too dark skinned. So we'll find out the one which are in the moderate will be having high numbers. You understand? Yes. So here is blood group. Tongue rolling is also another example of discontinuous. If you know how to roll the tongue, unfortunately, I didn't put on the camera. So theories, Lamarck's theory, Darwin's theory. I can't repeat this because you know it from your, yes. But there is only few questions I'm going to ask you. Please, when they bring a question, make sure that you apply the way it's supposed to be applied. Yes. When they bring a question about snakes, use snakes. Don't use giraffes. You'll find people using giraffes when they're talking about snakes. Why is the Lamarck's theory rejected? Lamarck's theory is rejected. That's what I'm going to focus on. Lamarck's theory is rejected because there is not enough evidence to show that the acquired characteristics are inherited. Don't just say that there is not enough evidence to support the theory. There is not enough evidence to show that acquired characteristics are inherited. Is it clear? So if they have given you an example, use that example. There is not enough evidence that the acquired characteristic of long necks are inherited. Do you see? The acquired characteristic of big shoe, big leg, big what are inherited. Number two, organisms don't evolve because they want to evolve. These giraffes, they didn't become longer because they wanted to become longer. They evolved because of natural selection. Is it clear? So please, it's rejected because there is not enough evidence to show that the acquired characteristic of long necks you apply based on the question. Unless the question is just flat, then that's when you say there's not enough evidence to show that acquired characteristic is. So, but if not, make sure that you give it right. So what is the difference between Darwin's theory and Lamarck's theory? Lamarckism, when you look at it, at first you say that there was no variation. That point must be there. There was no variation. Originally, all giraffe had short necks. All snakes had no legs. You understand? There is no variation. That's the meaning. Then now they started to use the necks frequently. They use those structures, whatever question they have given, frequently. And then as you use the structure, it becomes longer and bigger. You understand? And then they pass this gene to the next generation. So when you're explaining it, make sure that you use the law of use and the law of inheritance of acquired. No. And the law of inheritance of acquired characteristic. Is it clear? That sometimes it is the law of use or sometimes the law of disuse. If you want to remove that structure, then it's law of disuse. Those organisms did not use that structure frequently. If it is it is you want to develop the structure, then you tell us that this structure, those organisms use the structure frequently. Please, please, please know how to plan. So the difference, organisms here, uh, there was no variation, there is variation at the beginning. Here we say that organisms don't change because they want to change. Organisms change because they don't want change, because they want to change. Here in Darwinism, organisms change because of natural selection. So you have to know those things. Here, one individual in a population change. If you want to become light skinned, only you, not the whole population. But in Darwinism, if they say COVID is here, what is here, they say put on a mask. Everyone must put on a mask. You want it or not, you have to put on a mask. So, or the whole population will change. The whole population what? Change. But here, only individuals will change. So, guys, please, 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 you know how to know that. So natural selection, how it operates, please, you have to know organism produce a large number of offsprings. There is a great deal of variation among these organisms. Some have favorable characteristics and some do not have. And then when there is a change in environment, so you have to tell us which change is it. Some have favorable character. You have to tell us which favorable characteristics is it. Do you understand? How did the tiger came up with the stripes? There's a large number of tigers. Yes, there is those which okay, we say that tigers produce a large number of offspring, isn't it? Cool. Okay, some these tigers have, there is variation among these tigers, those with stripes and those without stripes. So you talked about the variation. So what are the stripes used for? Maybe to hide from the predators, or to hide so that the predators don't see them, whatever, depending on the scenario they have given you. So those with the stripes, they are able to capture, they were able to hide and capture the prey, so they survived. Those without stripes, they were being recognized by the prey, and then they couldn't get what to eat, and then they died. This is called natural selection. Those which had stripes, they reproduced in large numbers and they passed their genes to the next generation. Then after that, there was a very large number of individuals with the stripes. Like, you understand? Or after many generations, all tigers had stripes. So you explain based on the scenario. This one, punctuated equilibrium and gradualism. You need to know that in punctuated gradualism, there is check. It requires a lot of time. It requires a lot of time. Check the time it took from here up to here. While in punctuated equilibrium, check, it just changed from this. So it was here, it changed very fast. So it remained there without changing. So when you see it like this, then automatically that's punctuated equilibrium. It has periods of no change and long periods of rapid change. Periods of no change and long periods of short periods of rapid change. So you have to know that. So this punctuated equilibrium, gradualism, it requires a lot of time. It has intermediates. You see, here we now have no intermediates, which we call the punctuated equilibrium. Lastly, on this, but not the least, is artificial selection. Please don't confuse artificial selection with the genetic engineering. Artificial selection, we don't manipulate genes. We only give those ones which have desirable characteristics a chance to have sex so that they're able to reproduce. Yes, I hope, guys, we are together, and everything is good. You are hearing me properly. So we give a chance those which are able to produce. They are able to produce, so that they are able to they produce. Yeah. Uh-huh. So, and remember that it depends on the desire of the human. It depends on the desire, because what this farmer might need milk, the other one might need meat. So you give a chance to those which the farmer wants to develop. So please, 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 you have to know how that operates. What are the difference between natural selection and artificial selection? Natural selection, the nature is a selecting force, while humans here is a selecting force. You understand? Yes. Selection is done to be able to adapt in the environment. That's natural selection. While here we do it so that we satisfy human needs. That's what I told you, that depends on the desire of a human. And then this one occurs within species, while this one occurs in one or more species. You go bring this and then you interbreed with this, you see? Yes. So please, you have to know that. This question, to write exactly the way it is here, it depends on if the question is open or the question requires you to apply. Guys, you can't go to the paper without knowing these things. Don't make a mistake. Please. Do not make a mistake. You can't go to the paper without knowing this. There are so many people who are asking me, do I do physics? Yes, I do physics. I do physics. But I only do chemistry. I don't do physics. I don't do chemistry, physical science. Yeah, so I only do paper 2. Yeah, there is another one who does paper 1. Yes, in the studio. All right. So we are saying that a population of particular species, you mentioned the particular species. You understand? Ne? Becomes split into 2. Yes, by a geographical. You have to mention which. Sorry, you have to mention which geographical barrier is it. Let me show you this example. I think I have an example here. Yeah. How describe speciation of potos and lemurs. Or we can even bring these bobo plants or what? Bobo plants or yes, whatever. We can bring it depending on the desire of the examiner. Describe how they became new species. It means that there was a common ancestor somewhere, common ancestor. So they are separated by the sea due to continental drift. Yes. And then what happened to them? These two, they are not able to interbreed, meaning that there is no gene flow. They experience different environmental conditions from here and from here. So natural selection occurred differently. If it occurs differently, what is happening here is not what's happening here, and they are not interbreeding, then automatically what's going to happen is natural selection occurs independently, and then now they're going to become genotypically and phenotypically different. And then now, when you remove this barrier and you let them mix, they cannot interbreed. Don't stop there. They cannot interbreed to produce a fertile offspring. Therefore, now you tell us, therefore, this it formed new species of poto and lemurs. So you have to mention those species they have asked you to form. You understand? Then they form a new species of potos and lemurs. Yeah. You can't go to the paper without knowing these things. Please. Yeah, you can't. Okay. Then we go to the next question. So this is the different methods in which species are separated. You can be separated by different breeding at a different time of the year. You can courtship behavior. The way how you do courtship is not the same way how dogs do courtship. However, humans, you are different. You like different ways, styles, what? Humans, hi. All right, whatever. This is life science. So different courtship behavior. When the frog is doing, it's nice for them. It's not nice for you. So different courtship behavior. You want to go for a frog. You said humans. Then number 2, different adaptation of pollinatory agents. Sometimes you can even have a baby, and you produce an individual who is infertile. So it's a different way how you can separate yourself. Lastly is the present day evolution. Present day evolution works with natural selection. It doesn't work with speciation. You guys, you have a problem of identifying which one is speciation, which one is natural selection. It works with the natural selection, not speciation. Is it clear? So, meaning that here, before you apply, you'll find out that mosquitoes, DDT is just an insecticide. There is variation among these mosquitoes. So when you apply this DDT, some are resistant, some are non-resistant. So the DDT is gonna kill most of them which are non-resistant. You understand? So it's gonna remain with the resistant one. Meaning the resistant one, they're gonna survive and they pass their genes to the next generation. You understand? Then when you apply again the same dose, you'll find out that, oh, the mosquitoes are not dying. Why? Because now only resistant one are the one which survived. That's why when you go for medication, they give you different medication so that this one which is resistant to this one are killed, but the one which are resistant to this might not be resistant to another medication. And if they give you the medication, most especially antibiotics, make sure that you finish it, or otherwise you are creating monsters in your body. You're creating what? Monsters like this. If you're having HIV, make sure that you take the medication, or otherwise you are creating monsters in your body. And take it properly so that the resistance does not build. Because once you build this resistance, then you have to change the drug and you have to go for a stronger drug than before. I hope I'm clear. Hey, by the way, I'd forgotten to remind me. Don't forget to subscribe, like, and share. All right, guys, let's go to human evolution. Ish. Human evolution, guys. Are we there? Hey, someone was asking. I didn't see it. Yo, I feel like trying it is a lot. It's not a lot. Fumelel, it's not a lot. If you have been with together, it's like we are revising together. So now it's very easy for you to do. Just do some few questions. Don't worry. Keep on studying and practicing, sir. Make it. Yes, 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 yes. Thank you so much. Yeah, Mr. Goal. The goal, eh? Remember the goal. You have names, ne? Ah, yeah, 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 yeah. You are in Western Cape. You are listening to our teacher. Ah, yeah, guys. Thank you so much, guys, for being together. Okay, classification of humans. What you need to know here is where the family to which humans belong is Hominidae. Please make sure that you know that. And then genus Homo, we're gonna see how do we use it, when and how. Know how to write a scientific name, capital letter, and then small letter. That's it. We go to the next one. This one I'm not going to delay a lot. Why? Because here they're saying upright posture. When it comes to this, don't even stress about this. Just look at yourself. I have the eyes are in front. I have my opposable thumb. I have nails instead of claws. I have the free rotating arms. So you talk about yourself. About yourself, man, in the paper. I always tell people that it's on life science where you can copy an exam. Someone say, how? Someone is invigilating you and asking you characteristics humans share with other primates. Then you look at him that, oh, he has eyes in front, man. Yeah, you talk about things you see. And Max, on the paper, you copied from the invigilator. You guys, yeah. Life science is good. You talk about sexual dimorphism. You look at the female who is invigilating you. Yes, you can see this is the male, this is the female. You see the boobs there. Don't call them boobs. Yes. This is a female. This is a what? So you look at what you see. Yes. So don't be like raged with Hey. Let me go to another section. Then we go to this one. A difference between humans and other, in terms of cranial capacity. And they can ask you that how is cranium linked to intelligence? The higher the cranium, or the bigger the cranium is the brain box. The larger the brain cells, or the bigger the cranium, the more the brain cells, the higher the intelligence. So the cranium is big compared to the body size. That's why I say that cranium encloses. So human have a larger cranium, more than meaning that more brain cells compared to the body size. You understand? Yes. And then which suggests a higher intelligence. So guys, we have a higher intelligence because we have a high number of cells. Yes, compared to the other organisms. And this is linked to the body size. I think I'm clear. Someone is saying, sir, yes, sir, you will cry. All right, you'll try. All right. Non pomerero, ne? Mm. And then I'll look at this scalp. Eh? Guys, you got to say that if you don't know, touch yourself, feel yourself, feel yourself, feel yourself. Eh? Flat face. The other one is sloping. You just do the opposite. Flat face. Then you talk about foramen magnum in forward position. You will say forward position. You don't use any other word. Forward position to allow the spine to connect vertically. You talk about what you have. Wabona. You talk about what you have. Small canines. The other one has large canines. You understand? Large cranium. The other one has small cranium. So you look at this. You don't have broad ridges. It has broad ridges. If you are using protruding jaws, the other one is non-protruding jaws. Or you can say prognanthus, non-prognanthus. So it is easy. Mm. Then we are saying that you're saying that so these are the differences, guys. Please make sure that you know that, né? Yes. What is bipedalism? It's what I wanted. Bipedalism is walking on two hind limbs or walking on two legs. Cool. What are factors? The first one is position of foramen magnum. The second one is the pelvic girdle. The second one is the pelvic girdle. Yes, the pelvic girdle. Then the third one is the shape of the spine. Is it clear? Shape of the spine, pelvic girdle, and the position of foramen magnum. Foramen magnum is in the forward position. Allow the spine to connect vertically. Guys, it has to be that. Ne? Yes. It connects vertically. Pelvic girdle is short and wider to support the upper body weight. The spine is S shape. Allow flexibility. Look at your gogo, your mukulu. The spine is no longer S shape, it's bending like that. That's why walking is like that. When you tell him to stretch, you want to say, hey, you say that my son, man, I'm feeling pain. Why? Because the spine is not flexible. It's no longer S shape. And this is the problem tall people they find when they are growing up easily. Why? Because the spine can't support the upper weight. So what they do, it becomes a problem. Yes, meaning that they have those pains. So this is the difference between bipedalism and quadrupedalism. Foramen magnum forward position, foramen magnum backward position. S spine shape, C shape spine shape. Short and wider, long and narrow. So you have to know that. What are advantage of bipedalism? Don't still go to the book. Check yourself, the person who is invigilating you. When he's standing, he can see at the back and in front. You can see far. That's one example. Standing upright, you helps you to see the wide field of view. Is that difficult, guys? No, man. Isn't it? It's not. When he's walking, you see even he's checking the hands. Free hands so that you can carry other things. You see? Movement is made easier and energy efficient. What if you're walking on 4 limbs? Is that energy efficiency? No. The body is raised. So, guys, you explain based on what you see. You don't need to stress yourself with this so-called human evolution. You can't fail human evolution when you are a human. You can't. Human evolution. How? At least you fail the evolution by nice selection, you flies and stuff. It's not about you. You are not a fly. Cool. But human evolution, I engage. Never. You can't. You can't. Relato, you are starting me now, ne? Relato, you are starting me. I'm not the one who said those things. Anyway, they can give you such a question. Give the vis observable what you see. You understand? Hey, what you see. What you see, what do I see here? Now I don't know the blind ones. How do they answer this question? They say observable. Then they have to say what you hear. Hey, hey, questions are questions. And you find out that the person who is blind performs better than you. How? How? Anyway, that's what God has decided. But let us make sure that, sometimes they say that we are disabled. Ne? Sometimes they say that the person who is disabled is not disabled. The person who is disabled performs better than you, then it means that when you're disabled, it's not disabled. It's you. It's disabled. So please, when you read, you will, when you put something, you always get what you put in. Yes. Anyway, 4 characteristics. Now here they can specialize upper limb. Look at your arm and then talk about it. Explain how increase in the cranial volume related to intelligence. The bigger the cranium, the more brain cells, the higher the intelligence. That's it. We talked about it. What is bipedalism? We saw walking on 2 hind limbs. Explain each of the following structure. Okay, how contribute to bipedalism? Foramen and magnum. Forward position, allow the spine to connect vertically. Perivigato, short and wider to support the upper weight. Spine is shaped to allow flexibility. You see, you can do this. You can do it. Evidences. We have fossil evidence, genetic evidence, and cultural evidence. Guys, when they say evidences, you say fossil evidence. Genetic evidence, not in mitochondrial DNA. Mitochondrial DNA is an example of genetic evidence. Cultural evidence, use of tools is an example of a cultural evidence. I think I'm clear there. Yeah. See. All right. Fossil evidence. What is it? You need to know what is a transitional fossil. Unfortunately, I'm not live. I would have showed you using the skulls, but I'm not live. Ne? But I did a video on this. So transitional fossil, this and this. So this becomes a transitional fossil. It has characteristics of this recent ancestor and this next what generation. So it is in transit. It is moving. It's moving from here to there. Transitional fossil. It has both characteristics of the ancestor and. So when you're talking about transitional fossil, you have to compare. Actually, they are looking for comparison. This one, I'm not going to teach it. You have to know this table. But the only thing which I'm going to emphasize on is which one is a transitional fossil is Carabo. Which species first used the tools? It is Australopithecus. No. It's Homo habilis. Yes, is Homo habilis. First used tools. Yes, which you call the handyman. Homo habilis. And which species first walked on 2 limbs? It is Homo erectus, which means erect. Erect. Erect what? Standing upright. That's the meaning. Upright walking man. Erect. Homo erectus. You are walking when you have erected, meaning that you are walking upright. Think I'm clear. And don't have that in mind. Ne? I know you. You're saying that Name 3 Sauropithecus africanus species you know. Sauropithecus africanus species, Taom Chad, Mrs. Place, Littlefoot. Taom Chad, Mrs. Place, Littlefoot. That's it. Don't go to the paper without knowing at least Sauropithecus africanus species which we discovered in South Africa. Sauropithecus africanus species, all of them were discovered in South Africa. Wabon. Yes. Sauropithecus sediba, still South Africa. So when they ask you questions, you must know these questions. Simple. Name 2 species which were discovered in East Africa. Then talk about Homo habilis, which was discovered in Oliver George, which is Tanzania. You understand? Yes. So guys, please, you have to know that. So when they bring about the out of Africa hypothesis, you have to say that modern humans originated from Africa and migrated to the rest of the world. This word modern, it must be there because Homo erectus, Homo neanderthals, Homo naledi, all those are humans. But Homo sapiens are the modern humans. That's why you add the word modern humans originated from Africa and migrated to the rest of the world, during the Homo erectus period. Yeah, that's it. So when they ask you the evidences, you talk about fossil evidence, genetic evidence, cultural evidence. But how do you write it? This is where the problem is. Fossils of Adipithecus was only found. The word Adipithecus. Why? Because it is the oldest. Adipithecus, Australopithecus were only found in Africa. Then these ones were not only found in Africa. That's why we say that the oldest fossil of Homo erectus and Homo sapiens were found in Africa. The oldest were found in Africa. These ones, the oldest were found in Africa. But fossils of Adipithecus, Australopithecus, and Homo habilis were only, this word must be there, were only found in Africa. But when it comes to oldest, this word oldest, it must be there. When it comes to Homo erectus and Homo sapien, take a screenshot of that. Then we continue. Don't say, good day, sir. Is good night. Good day. Is it still a day? Good night. Hi. Okay, good day. How are you? Maybe you are in Dubai. All right. Anyway, all right. Let's continue. Wait, good night or good evening? I don't know. Hey, English, leave me alone. English is not my mother tongue. I know that's what you usually say. All right. Mitochondrial DNA, then you have to say that is inherited only from maternal line, and then analysis of these small mutations, yes, show that the oldest female, yes, ancestor were located in Africa, meaning that all these humans descended from that, which we call the mitochondrial Eve, meaning that Eve was from Africa. In other words, they are saying these people that Eve was a black person. Why? Because in Africa we find black people. But are they black? They're not black, man. They are dark-skinned people, not black. Anyway, you can call them the way you want. Yeah. So you are saying that they're like a pot. They're black. Anyway, so when they ask you colors, you say that's black, ne? You know this Mozas. This one. Others they call them colorless. So when you're colorless, they're skin, light skin. Me call them that. But anyway, if I'm black, I'm colored, guys. If you are black, you are colored. If you are white, you are colored. So everyone is colored. Yeah, they just want to make us divided. Aye, man, we are all of the same because we have the same type of blood, red. Anyway, I'm not talking about that. Let me just keep it quiet. Yeah. The oldest tools, you know them, they are called the other one tools. Ish. Guys, I think I'm supposed to be closing now. But anyway, let me just do this highlight very fast, so that you don't fail that phylogenetic tree. All right. We have different perceptions, Don't take me wrong, ne? I was just talking. And I'm not that kind of guy who appreciate everyone. Anyway, let's start. And then we continue. We get our distinction. We smash the paper on Monday. What is the name given to this diagram? This diagram is called a phylogenetic tree. How many genera are shown in this diagram? When they say genera, guys, look critically and look here. What's happening with my screen? Don't tell me that. So it means it happened there. All right. How many genera? So you look at the first one is Adipithecus. Remember, genus is the first name. The second one is Australopithecus. This one is Australopithecus, so I don't repeat. So you have two Paranthropus. Those are three. You see, Paranthropus, Australopithecus. I don't repeat. The fourth one is Homo. 1, 2, 3, 4. Do I have another one? No. I think there are 4. So there are 4 genus. That's how many genera. Is it clear? Yes. Yes. So how many genera? I think I've answered that. What is the common ancestor of Homo and the Homo sapien and the Homo erectus? You come here, Homo erectus is here, Homo sapien is here. When you join them, they join here, meaning that the common ancestor is Homo ergaster, based on this diagram. Is Homo ergaster. What is the common ancestor of all species? Automatically is Adipithecus remidus. Give 2 pairs of species which competed for resources, meaning that give 2 pairs of species which existed at the same time. So it means that you just have to draw, just choose. Maybe I'm going to choose this. Draw until it touches. So you see this, it's in the same line, even this. Yeah, they exit at the same time, but it could be half of the time of all the time. So, but they exit at the same time. So if they exit at the same time, it means that they competed for resources. Then they're saying that which species first use tools. In other words, it is Homo habilis. But based on this, you have to look at whether they gave you what to use, the scenario or not. Based on this scenario, the tools are here. They're saying that, yeah, the tools started here. So maybe it is Paranthropus Ethiopicus, the one which first used tools. Then they're saying that which species first walked on 2 legs. Here we talk about 2 legs, 2 legs, 2 legs. 2 legs is bipedalism. So you look for bipedalism. Bipedalism is this one. So it's around there. Which one is it? Yeah, Australopithecus and Amensis. You see? So that's how you identify. Then they're saying that and they're saying that which species first walked on. Okay, how many million years ago did Australopithecus africanus appeared? Australopithecus africanus. I look for Australopithecus africanus. I think it is here. Then I calculate. It appeared around 2.9 million years ago. And then when did the Homo habilis became extinct? When did the Homo habilis became extinct? Homo habilis, Homo ergaster, Homo neanderthal, Homo habilis is here. It got extinct around 1.6 million years ago. They are saying that how many million years did Australopithecus africanus existed? So it means that you have Australopithecus africanus where it started and where it ended, then you subtract. Yes, then you say million years. Here you don't say million years ago because they're saying yes. They're saying how does brain size show evolutionary relationship or trend? We say that the bigger the cranium, the larger the brain cells, or more brain cells, the higher the intelligence. Meaning that these organisms will use advanced tools. You understand? Yes. Remember, we said that these tools, the more you modify them, the more they become more. The more you advance the brain, becoming bigger, the tools become more sophisticated, meaning that these tools, they require thought. You require to think to make them. Give an example of a transitional fossil. We said it's carabo. How does dentition show the diet of the species? Large canines, meaning that they are feeding on rough food, and then the smaller teeth, they show that they are feeding on fructy food. Guys, I think that is it. We'll stop here. Thank you so much. Tomorrow, I think I'll make a cross night. It will be a cross night. Please, please, please make sure that you go through this video 3 times before you go for the paper. I repeat, make sure that you go through this video 3 times before you go to the paper. When you come out of the paper, call me or tell me here, what does it mean? With me saying go through it 3 times. You understand this when the paper has come. Is it clear? I wish you the best, and I know that you guys, you gonna make it. I want you to be responsible citizens. Please, please, please be responsible citizens. Now you can have a relax, watch more TV, then after that make your brain relax, and then you come back and redo, or you watch the video, or you revise. Yes, see you again when we come back for the last time. Relato, you are welcome. Hey guys, sleep well. I see you, Sandy Le. I see General. You have DJ AJ Junior. All right, Rivonia. I see you guys. It has been a nice lesson. I'm seeing Doomsay. I see you, big boy. Big boy, thank you for being with us from the beginning until the end. Rivonia, you also did that the same. Thank you so much. I see you and I salute you. I wish you the best, guys. Doomsay, please go back and then replay the video. I explained DNA. Thank you so much. See you again. It has been M. Saidi. As usual, don't forget to subscribe, like, and share of life, whereby this topic, what you're supposed to look at is DNA structure. Describe the structure of DNA. That's a question. Describe the structure of DNA. In this case, what are you supposed to look at? I'm going to tell you. Number 2, you know that DNA replication. Yes. Transcription. Translation. Yes. These ones, you know them. It's easy to get them. But now, the most important part here is the structure of DNA, because sometimes people neglect that. You know how to describe DNA replication. Structure of DNA, how do you describe? Just in case it has come. First of all, you know that DNA is made up of a simple structure called nucleotides. If they ask you what is the monomer of DNA, you have to know that monomer of DNA is a nucleotide. And these nucleotides, yes, they have different parts, whereby this is a phosphate, this is a sugar, and this is a nitrogenous base. This is a phosphate, this is a sugar, is a nitrogenous base. Remember, this side you also have the same kind of structure. Yes? If you have the same structure this side, it means that if they ask you to describe, you have to tell us that DNA is made up of a nucleotide. Yes? These nucleotides are made up of phosphate. Nucleotide is a mark. Nucleotide is made up of phosphate, sugar, that is deoxyribose sugar, and nitrogenous bases is also another mark. Then you mention the nitrogenous bases. What are those nitrogenous bases? Then you tell us is adenine, is thymine, is guanine, and then is cytosine. You mentioning this is another mark. Then you tell us that it is made up of 2 strands. Yes, these strands, you telling us strands is another mark. These strands are joined together by weak hydrogen bonds. These weak hydrogen bonds is also another mark, weak hydrogen bonds. And then it twists around to form a double helix. Double helix is also another mark. There is only one mark I've not spoken about of you mentioning the bases, but also saying that whereby adenine goes with the thymine, guanine goes with the cytosine is also another mark. You mentioning that adenine, thymine, guanine, cytosine is a tick. Adenine with thymine, guanine with cytosine is also another tick. So that is DNA. So all these, if you count the marks which are there are 8 marks, but we only need a few of them. And those few of them, what is it? We only need 6 of them. So don't go to the paper without knowing to describe the structure of DNA, because it belongs to 6 marks. DNA replication. Here you have to describe the structure of DNA. DNA replication, whereby you tell us that DNA double helix is unwinds, weak hydrogen bond, break, yes. Unzip and the breaking of weak hydrogen bond is 1 mark. We don't give you 2 marks there, is 1 mark. Then to form 2 separate, let me repeat it. The word separate must be there. To form 2 separate strands. Each strand is acting as a template. It's Zoleka, I think you are hearing me. Ne? Yeah, I'm just looking at the people who are live, so that if you have any challenge, let me know in the comment section. I will answer it immediately. All right. Saying 2 separate strands is another mark. The 2 strands, they will act as a template. They will act as template. And then using free floating DNA nucleotide. We use DNA nucleotide to form a complementary strand, whereby adenine goes with thymine, guanine goes with cytosine. I see. Then you form this DNA with 1 original strand and 1 new strand, which are genetically identical. That's how you can describe DNA replication. But we have some few questions. I'm gonna show you these other questions, because this is a 6-mark question, just in case. Remember, your paper, it has almost 45 marks full of this short question. In each section, we bring at least 1, of which this, at least 1 will come. All right. Then transcription. Transcription must also know it, that DNA double helix unwind, weak hydrogen bond, break to form 2 separate strands. Now, 1 strand acts as a template. You have to know that. 1 strand acts as a template to form the messenger RNA using free floating messenger RNA nucleotide. In this case, we are using free floating messenger. For the case of DNA replication here, here we are talking about transcription. But when you're talking about DNA replication, 2 strands act as a template, while in transcription, 1 strand is acting as a template. Then whereby adenine goes with uracil, adenine goes with uracil, and then guanine goes with cytosine. Yes? Then this messenger RNA moves out of the nucleus via the nuclear pore to the ribosome. So that's, once you write it like that, obviously in these questions we always have questions or marks which are excess. So we are looking for only 5 to 6. Transcription is done. What about translation? In translation, depending on what you have memorized, but I'm going to show you the marking point in each. If you skip, you have lost a mark. According to messenger RNA codons. Codon is a mark. The transfer RNA with the complementary anticodon. Anticodon is a mark. Complementary anticodon is also another mark. Brings the required amino acid. It brings the required amino acid to the ribosome. The required amino acid is also another mark to the ribosome. Ribosome is another mark to form. The amino acids are joined together. The amino acids are joined together by peptide bond to form a required protein. That's it. Nothing else. According to the messenger RNA codons, the transfer RNA with the complementary anticodons bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. DNA code of life is done in terms of the 6 marks. So it means that you have at least one question which is most likely to come. Now listen, I'm gonna show you now these other questions which might come, or which you're gonna find in your paper. Listen. Before I go there, students make a mistake and they call peptide bond, they call them polypeptide. This is dumb. This is wrong. Polypeptide bond, we don't have this. We call it peptide bonds. We don't have polypeptide bond. Please don't call them polypeptide bond. We call them peptide bond. And then I'm going to show you also another mistake where students make. They call a polypeptide chain polypeptide chain a protein. It is only a polypeptide chain if it is less than 50 amino acids. 50 and above amino acids, we call them protein. It's only a protein when it's 50 amino acid and above. I hope you are getting it right. Okay. What we are saying here.
Recording in progress.
Sorry about that. All right. We are saying that. Look at the questions where other questions. DNA replication. Why does it occur? To increase the chromosome number. Why does it occur? To increase the chromosome number. Where does it occur? It occurs in the nucleus. When does it occur? It occurs during interphase. I think. How does it occur? Is the process which you have described. So DNA replication is done. Who discovered the structure of DNA? James Watson and Francis Crick. James Watson, Francis Crick. Another question. What is the difference between DNA nucleotide and RNA nucleotide? What is the difference between DNA nucleotide and RNA nucleotide? They are not asking what is the difference between DNA and RNA. They are looking for only the nucleotide of DNA and the nucleotide of RNA. So in this case, you talk about DNA has deoxyribose sugar, RNA has ribose sugar. DNA has DNA nucleotide has thymine, RNA has uracil. We don't have any other. That's it. Someone is asking me that I should repeat the structure of DNA. Describe the structure of DNA. Yes, I'm going to go to even genetics. I'm going to go even to evolution. I'm going to show you the whole paper, what you expect, and within these 2 hours, you'll be able to write the paper. You write the paper within 1 hour. So the discussion we're going to do within these 2 hours, you'll be able to write this paper within 1 hour. Meaning that nothing we're going to miss. Okay. So now as I'm winding up, What is the difference between the difference between DNA and RNA? Those ones you know. But there is one question they can ask you. What is the difference between transcription and transcription and DNA replication? Transcription, here you form one strand will act as a template, while DNA replication, the two strands will act as a template. I think. In transcription, yes, we use uracil, adenine, thymine, guanine, uracil. While in translation we use adenine, in transcription we use adenine, guanine, cytosine, uracil for the new strand which is formed, meaning that we form a messenger RNA. While on DNA replication we form DNA. Then in transcription, yes, you'll find out that in transcription and DNA replication, we have also some few similarities, whereby DNA double helix unwind, also in transcription, DNA double helix unwind. Weak hydrogen bond break, also decide weak hydrogen bond break. If just in case they are looking for the similarities there, you are able to answer that. Now, as I'm winding up this DNA, there is a question which is always a challenge to students. What is that question? That question is about the genes. The gene mutation. Gene mutation. Now, let me give you an example. If you have A, G, C, and this is codes for leucine, and you have A, C, G here, and this codes for tryptophan, yes, and then you have A, C, G, and this also codes for leucine. Then they ask you, how will the protein be affected if this is changed to this? Because you form a different amino acid, the protein functioning or the functioning of the protein will change. So it means that you have to tell us that when AGC is changed to ACG, tryptophan will be added to the protein instead of leucine or the polypeptide chain. This leads to a different protein or the different functioning of a protein. You mentioning this and mentioning this is a tick, and also mentioning different protein is also another tick, because this question is always 3 marks. What about if this with this G changes this and this changes to this, even if it is 1, but it is the same amino acid? This means that the functioning of the protein will not change. However much the base sequence has changed, but what is formed codes for the same amino acid. Therefore, it means that you will find out that it is exactly the same protein. The functioning will not change. It's like someone saying that, for example, let me just use an example here. Someone can call you Sibusiso. Someone, you will say yes, sir. Or can call you Ntuli. Still you say yes, sir. I think. Meaning that is the same kind of person. But if this, because this is leucine and this is leucine. What if someone calls Sibusiso, and then the next time the next caller calls you Dobe? Obviously, Sibusiso will not reply. No, Sibusiso will not reply. It's Dube who will say that, yes, sir. Because now it is different protein, the functioning will change. But it's the same protein, the function will remain the same. So, I repeat, I've concluded with DNA. We will only look at it when you're talking about the questions. So here you are saying that DNA structure, replication, transcription, translation. Please don't go without knowing it. And this gene mutation of DNA. When you talk about DNA profiling, that one is a little bit easy. Please just read the advantages, disadvantages, and sometimes the trick which is there is they tell you that other than that what is mentioned in the question, give another, and you find the student giving the same answer in that question. So it means that you lose a mark. Let's check and see here. I'm not going to go through objectives, but I'll go through the terminologies because I love the terminologies. Here is the question of DNA. Let me just reduce it a bit. Now, fast. Now, this is just a simple question. What is X? In most cases, people say that X is a cell membrane. X is not a cell membrane. X is a nuclear membrane because the cell membrane is out. Nuclear membrane, this is a nucleus, but they can ask you the process. What is this? This is translation. Once you see something like this, it is transferrin A, this is a amino acid. But if they put dots there, this is not a protein, it's a polypeptide chain, because I said that polypeptide chain is less than 50 amino acid, above 50 amino acid, it is a protein. Since this is just 1, 2, 3, 4, 5, only 5 amino acid, therefore that is a polypeptide chain. And then what is this? This is messenger RNA, and then which organelle is this? Is a ribosome. Now, question can come. How many bases, how many nucleotides, how many codes code for amino acid? So, let's answer that. How many bases code for an amino acid? Amino acid, remember, an amino acid, yes, an amino acid, yes, it's always having 3 bases because of this, 1, 2, 3. So, if how many bases? Is 1, 2, 3. But they're saying that if there are 3 amino acids, how many bases are there? So, it's going to be 1, 2, 3 times 3, which gives you 9. If I have 9 bases, how many codons are there? Remember, each 1, 2, 3 is one code, is one codon. So how many codons are in 9 bases? So I will say that divide by 3. Therefore, how many codons? Codons will be also 3. I think you have to know when to divide and when to multiply. Yeah. Lastly, on this, they can also give you a question that they can ask you a question that, look, they can ask you a question that 20% is adenine. What is the percentage of guanine? So you have to know this. If 20%, you have to know that A goes with T, C goes with G. C goes with what? C goes with G. So if A is 20, this one is also what? 20. So 20 plus 20 is equal to 40. So now, because this percentage is out of 100, therefore I'm going to say 100 minus 40. So it gives me 60. I think if it is 60, what is really happening there? 60 is going to be for these two. So divide by 2. So it's going to be 30 is going to be 30. So it means that the percentage of guanine is going to be 30%. Don't go to the paper without knowing how to calculate the base. Yeah. I'm using these as examples. So we are saying that which organelle is X? We have seen organelle X is nucleus. We have seen it. Let me do like this. Is nucleus. And then which molecule is Z? Which molecule is Z? Where is Z? Where Z is messenger RNA. Don't just say RNA. It's messenger RNA. State 2 locations in the locations of DNA in the cell other than the nucleus in the cell. So it means that you have to talk about number 1, mitochondria. mitochondria and chloroplast. Yes, mitochondria and chloroplast. Describe process W. W is we don't have any other process occurring in the nucleus except transcription. So you have to describe transcription. I've exhausted most of the questions you're gonna find in DNA code of life. It's only one last question. Let me yes, this is the question, how to interpret this. And however, I've tried to explain something here. The trick behind here is one. How? The trick which is here, please check and see what is there. If they say this is DNA and they gave you DNA at the beginning, then you don't need to change. If they say they give you DNA and what is here is transfer RNA, you have to change it to messenger RNA and you have to change it to transfer RNA. Are you seeing? So, in this case, they are saying that name the DNA-based triplet code for serine. Now this is transfer RNA. Serine is here. So I'm going to say is UCG. This is transfer RNA. Convert it to messenger, which is going to be, yes, AGC. Then convert it to DNA because they want DNA. How many times do you convert? It depends on what they have given you. So it's going to be, yes, it's going to be A, yes, G, and then C. So that's going to be, are they saying serine? Yes, that's what is going to be the answer. First two amino acids coded for Z in the diagram. So they are saying they want to identify. Now, this is transfer RNA, but Z, what is Z? Z is messenger. Therefore, you have to just change this into transfer RNA, one conversion, because it is messenger to transfer RNA, and then you come to the table, you find it. What is the change in sequence of nitrogenous bases? I've talked about it here, that these changes. Gene mutation, sudden change in the sequence of DNA. So that's what you call gene or the nitrogenous bases. When they change the sequence, then automatically you're going to have a different protein structure if it codes for a different amino acid. So what is a change in a sequence of nitrogenous base in DNA code? Gene mutation or mutation. They're saying that code CUU changes to C. Now they're saying codons. It means that they are not saying anticodons, codons. So it means that I have to convert this into transfer RNA. Sibosiso, are you hearing? All right. We are saying that change this is going to be GAA. And then this is going to be GGA. So GAA is going to be what? So now you saying this is going to be leucine. Like the same example I gave. CCU is going to be GGA. GGA is going to be what? Proline. Proline leucine. Now you writing to us that this is going to code for leucine, this is code for proline. Those are 2 marks. Leucine, a mark. Proline, a mark. I think. Now you tell us because of a different amino acid is coded for, it will result in a different protein. But if it was coding for the same amino acid, then the protein would have remained the same. So it would have said that this code is for the same amino acid, for example, leucine, and also this code is for the same amino acid, which is leucine. This will not change the functioning or the structure of the protein. But because now it is changing, this is leucine and then this is proline, you're mentioning leucine a mark, proline a mark, this will result in a different protein or functioning of the protein. All right, let's go to meiosis. Yes, meiosis, unless you have any question. Someone is asking me, where do you get my booklet? There is a link in the description below. Ne? Yes, that link, it will take you to the website. You go to grade 7 to 12. Then it's going to bring you another page which has past paper 2022. It's going to bring you notes. You click on notes. Then it's going to give you the different subjects for grade 12 and grade 11 and stuff. You click grade 12 and then it's going to give you the subjects now. Now the subject, when you click life science, is going to show you the different topics. Then you download the topic per topic. Yes. That's how you can get that booklet. Let's look at meiosis. Meiosis. What do you expect to find in meiosis in your exam? Meiosis. What do you expect to? I'm now talking about the 6-mark question. I showed you the 6-mark question for DNA. So now I'm showing you the 6-mark question for meiosis. You cannot miss. You will come back and then comment that, sir, thank you for giving us the scope. Yeah. If you follow what I'm telling you, I guarantee you a distinction. Share the video to others so that the others can also. Someone is saying that it's not clear. The clear is as a result of your the internet and your internet connection. You can increase the maybe from 128 to maybe 480, something like that. All right, let's continue. Meiosis, number 1, you need to know how to describe the karyotype. Karyotype. I think you know the karyotype. If you don't know, I'll show you how to describe it. You need to know how to describe crossing over. You need to know how to describe Nondisjunction. Ne? Now, this one is a star, is a drawing. Phase before, phase after, phase at this side. So you have to know that. How do you describe a karyotype? Let me see if I can find a question here which has a karyotype. And then I'm gonna describe for you so that when I'm describing, you see ne? You see what I'm trying to describe. Sorry for, yeah. Here is an example of a karyotype. Describe the karyotype of individual 2. Ne? But when the question comes, the karyotype might not be there. You have to be knowing it by head. For example, the karyotype is individuals are made up of 46 chromosomes or 23 pairs of chromosomes. Yes? From you mentioning that 46, ne? 46 is a mark. Ne? 40, sorry? You know, technology. What's happening? Okay, let me check and see. 46 or 23 pairs. Don't say 23, because if you just say 23 without writing the word pair is wrong. It's made up of 23 pairs of chromosome. From pair number 1, yes, to pair number 22 is called orosome. You see now you're describing a karyotype. It's called what? Orosome. Yes. The 23rd pair is gonosome or sex chromosome. Yes. The 23rd pair is made up of X and Y. If X and X, they're together, the individual is a female. All those are marks. If X and Y come together, the individual is a male. You see, you are describing the karyotype. You have already exited the 6 marks. Each chromosome pair, they have different sizes. So that's how you can describe the karyotype. It's made up of 23 pairs. Pair number 1 to pair number 22 is called orosome. The 23rd pair is called a gonosome. These pairs, yes, each pair of chromosome have different size. The 23rd pair, if X is made up of X and Y, if X and X meet, is the individual is a female, is X and Y meet, the individual is a male. That's how you describe. Now, what about if they bring a karyotype like this? Yes, like this, and then ask you which one is a male, which one is a female. Check. This is short, this is, two of them are long. Therefore, this one is a male, this one is a female. Give a reason for your answer. The chromosomes here, they are not of the same size. I see here, same size and shape. While here, they are same size and shape. So it means that this is X and X, while this is X and Y. Remember, we said is longer than Y. That's why you find that to happen like that. Now, someone is asking me, all right, I think it's sorted. Now, we are saying that, let's go to another question of karyotype here, just in case, because I was describing the 6-mark question, but there can bring some few questions here. What are somatic cells? Somatic cells, these are body cells. These are just body cells. Name the specific type of chromosome number 1 to I said it is the orosome. Each pair shown in the homologous chromosome, each pair states the origin of each pair of chromosome. Definitely, one came from the nucleus of the ovum, another one came from the nucleus of the sperm. Meaning that one chromosome. For example, this chromosome came from the mother, and this chromosome came from the father. So came from the nucleus of the ovum, this one came from the nucleus of the sperm, and then fertilization took place, and then a zygote was formed. That's what they are looking for. They are saying that state 3 characteristics of chromosomes which are homologous. Homologous chromosomes, these are chromosomes with the same shape, size, same location of genes. Not same genes, no. Same location of genes. If they are same genes, then it would have looked alike. But the chromosome where they are located, the locus of these genes in these chromosomes are the same. I think now I'm clear on that. So please, you must not miss out any question. Explain one observable reason why these two individuals are not identical twins. Why are these ones not identical twins using this karyotype? These are not identical twins. Why? Because this is a male and this is a female. Obviously, you know that if it is a male, what happens? If it is a male, the sperm is gonna be Y from the father. Ne? And if it is a female, the sperm is gonna be, if it is a female, the sperm is gonna be X. You understand? We only have one ovum. So I cannot use one sperm and then I form X and Y. Never. Now, why are they not identical? Because of the different sex. Observable, the 23rd pair, this individual one has X and Y, while individual 2 has X and X. So, identical twins are as a result of one sperm fuses with the ovum and then the ovum splits into 2. Non-identical twins are as a result of 2 sperms fusing with the 2 ova. So, meaning that you have different individuals. That's why is the non-identical, you can have a boy and a girl, but the identical twins is either boys or is girls, because we used one sperm. If the one sperm of X fuses with the ovum, then it's gonna be identical twins girls. If one sperm of Y fuses, it's gonna become identical twins male. So that's exactly happening. Yes, I think, yes, I'm done with the karyotype. What about crossing over? Crossing over. Describe the process of crossing over. I think I organized for you a question which you're gonna see in crossing over. Oh, I think it's not there. It's here. Crossing over. Yalla, it's here. When you talk about crossing over, you talk about two homologous chromosomes. These chromosomes, they come together. I think these chromosomes, they come together. They cross over at a point called chiasma. They must be homologous. The word homologous chromosomes align together. That's the key point. Homologous chromosome. They come together and the chromatids of the homologous chromosome cross over at a point called chiasma. I think at a point called chiasma. You understand? And then these chromosomes, these chromatids, they exchange the genetic material. When they exchange the genetic material, yes, they split and then they form genetically different chromosomes. That's why I told you these are genetically different chromosomes. But these homologous chromosomes, they are not the same. They don't have the same chromosome number. They have the same location of the genes in their chromosomes. That is the meaning. All right. Now, what is P? What is this P? We have talked about, we said that is easy. Some people, they call this centromere. That is not a centromere. A centromere is Q. Please don't confuse centromere and chiasma. C, chias, this pen, chiasma. If swan is called chiasma, if the men we call them chiasmara. This is a centromere. And then this is a chromatid. It's not a chromosome, it's a chromatid. What about this? These are homologous chromosomes. Sometimes they can ask you, how do we call the homologous chromosomes under crossing over? It's called a bivalent. It's called a what? A bivalent. Chromosomes under, homologous chromosomes under crossing over. We call it a bivalent. Yes. I've not checked if, because the schedule changed, if just in case that electricity goes at 4 o'clock, I will come back at 6 o'clock live. But if it doesn't go, we are still continuing until a distinction is scored. Yes. All right, let's continue. Now, we are done with the crossing over. Let's talk about nondisjunction. What is nondisjunction? Are you there, guys? Nondisjunction. If you have any challenge, if I'm going to another point, please let me know. I'll read your comment there and then I'll come back to you. All right. Nondisjunction. What is nondisjunction? Before I go to nondisjunction, where does crossover take place? It takes place during prophase 1. Don't just say prophase. We will get it wrong. Prophase 1. Phase 1. What are some of the causes of mutations? No. What are some of the causes of variation? We'll talk about them. Causes of variation. I'll see how do people lose marks here. You think that you have written the paper, can't the paper has written you? I'll show you. There is no way the paper is going to write you tomorrow. You're going to have to write it. All right, let's continue. God willing. We are saying that nondisjunction occurs during anaphase. Nondisjunction occurs during anaphase. It can be anaphase 1 or it can be anaphase 2. Anaphase 1 or it can be anaphase 2. So what is this non-disjunction? Check here. I'm gonna show you how. Someone is asking me that, can I please repeat bivalent or bivalent? Bivalent is when homologous chromosomes are under crossing over. Chromosomes under crossing over is called bivalent or bivalent. Yes, that's what I said. All right, now come back here. You have chromosomes in metaphase. Sometimes students fail to understand the chromosomes under metaphase. The chromosomes must be in the equator. Equator is an imaginary line. An imaginary line. We just imagine a line which is happening. Yes? It's called what? Equator. But we said that is the center of the cell. So these chromosomes must be at the center of the cell. This thing must be on the sides. Unfortunately, I have not used the camera. I would have showed you. You'd have seen me when I'm trying to say sides. So that, but you have to know that, make sure that the centrioles are inside, so that you, sometimes we twist it so that we confuse you, so that you go out, so that we know that you if you just cram, then gone. So they must be on sides. So they can ask you, draw a cell after, cell before, or crossing non-disjunction. How is this non-disjunction? How this cell gonna lead to non-disjunction? Now, if these spindles contract and they pull this chromosome, now this whole set is pulled to this side, and then this one comes this side, this one comes this side. You result in a cell whereby this cell has 1 plus this, and then this one, because the whole thing went to the because this one went this side and the whole thing of this went to this side. Let me show you. Let me do like this. Let me do like this. Let me do like this. Let me do like that, so that, yeah. So it means that this part came to this side. This one came this side, while this one came to this side. It means that now this cell is going to have 1, 2, 3 chromosomes. What about this side? It's going to have only 1. I think. Now this one is 3 instead of 2, and then this one is 1 instead of 2. I think. So because it has 1, therefore it's going to call it monosome. Well, because this one has 3 instead of 2, it's going to call it trisome. I think. How does this lead to Down syndrome? How does this lead to Downey's syndrome? I think. So it means that now, if this fertilizes the ovum, then it's gonna lead to an individual less of 1 chromosome. If this fertilizes the ovum, it's gonna lead to an individual with an extra chromosome, and this is called Down syndrome. But it's not called Down syndrome except it has occurred on chromosome number 21. So which chromosome number is chromosome number 21? Sometimes when we bring these karyotypes, okay, I think I used it. No. Karyotype. What is karyotype? Sometimes, okay, I wanted to show you. Sometimes we bring it and then we confuse you on the karyotype, ne? And then we ask you, how did yes, check here, ne? On this chromosome number, they put another one there, and then ask you, is this chromosome individual fine? How many chromosomes? Yeah, how many chromosomes are there? Because you know that this an individual is supposed to have 23 pairs. When I just say 23 pairs or 46 chromosomes, wrong. Check before you conclude anything. Please check the chromosome number 21. I'm done with that. Let's go to this. They're saying that a drawing, we have shown it, but a drawing, it must have a title. Ne? Must have a title. When they ask you to shade, for example, if the chromosome is like this, and this one is shaded, ne? Please, when you draw, still draw it like that and is shaded. If you shade it down, shade it down. If you shade it up, shade it up. After that, label. Have some 2 labels. I want to give you one thing students always mess up with is Metaphase. Listen carefully. Don't go away. Anaphase. Yes, then telophase. I'm going to say this Metaphase 2, Anaphase 2, telophase 2. When these chromosomes, let me show you here down. Let me show you here down. When these chromosomes, look, I have it here. When they're under Metaphase, we call them chromosome. When they're under, they're being pulled now. They're being pulled to opposite poles, like this. These are not chromosomes. These are now chromatids. But the moment they reach the poles, we don't call them chromatids. We call them single-stranded chromosomes. Unreplicated chromosomes or single-stranded chromosomes. We only call them chromatids if they are at under anaphase. They are being pulled. Here, they are chromosome. These are replicated chromosomes or double-stranded chromosomes. These ones are called chromatids. The moment they reach the pole in telophase, it becomes what? It becomes unreplicated chromosome. Don't call them chromatid. Yes. Let's go back to this last question of meiosis. Then we conclude it. Yes. Okay. Now, they are saying that drawing variation. What is variation? Variation is the difference among organisms of the same species. Variation is the difference among organisms of the same species. Don't compare mangoes and oranges. Don't compare dogs and pigs. That's not variation. Variation must be comparing dogs. Variation is the difference among organisms of the same species. It must be the same species. Yes. Now, what are some of the causes of variation? If you're comparing humans, you must compare humans. So what are some of the causes of variation? The causes of variation is, now, the major mistake students do in this exam, if they ask you 3 causes of variation, the first thing the student is going to say that the first cause of variation or genetic variation is going to say that meiosis. Yeah, we shall say correct. And then the next point is going to say crossing over. We're going to say thank you. Then it's going to say random arrangement of chromosomes during metaphase. We're gonna say yes, but no. Why are we crossing it when it's correct? It's because you mentioned here meiosis, and meiosis involves crossing over and random arrangement of chromosomes. So if they ask you this question, please remove meiosis. Don't think about meiosis. Then you talk about crossing over, random arrangement of chromosomes. There are 4. We will not put you this. We will give you a tick here and we'll give you a tick here. So you see, you have got 2 marks. Then plus you can talk about mutation, random fertilization, unless they have said that give 3 causes of variation during meiosis or during the process of meiosis. Then now you talk about crossing over, talk about random arrangement of chromosome, and then you can talk about mutation. Then you don't talk about random fertilization, don't talk about random fertilization or random mating. You don't talk about it. Yes. Yeah, I think that is the meiosis. Can we check and see? There is one question you usually lose marks. How many number of chromosomes? That question is always killing students. Is always killing students. Is always killing students. All right. Let's go through this question paper. This one. Then they're saying that what is A? A is a cell membrane, not nuclear membrane. You can't see nuclear membrane when a phase is meiosis, is metaphase. Spindle fibers, then this is a chromosome. Yes, identify the phase of meiosis above. Because they are single chromosomes, therefore it's gonna become what? It's gonna become metaphase, because they are single metaphase 2. If they are double, then it becomes metaphase 1. Metaphase 1. But here, single metaphase 2. All right. Let's go to another question. They are saying that what is A? We have seen it. What is B? We have seen it. Give one observable reason for your answer. Chromosomes align along the equator singly, individually. That's, if you don't say singly, 0. Or individually, 0. It must have that word, individually or singly. Chromosomes align along the equator, 1 mark, singly. What about describe the role of B in the movement of these chromosomes during meiosis? B is spindle fibers. They pull the chromosome, the chromatids, in this case, the chromatids to opposite poles. They try to shorten. When they shorten, they pull the chromatids to opposite poles. Charlotte, are you still here? All right, let's continue. Draw it. This is the question I was talking about. You can't go without. Put the title. Good. After putting the title, what next? What next? What next? You talk about, you draw, because they're saying that draw a diagram of C, structure C. They're saying structure C, ne? As it would appear in the final phase of this meiotic division. They are saying only C. So if they're saying structure C, they're not saying draw the cell, how it will appear. Since they're saying structure C, then automatically you're going to draw this. Okay, let's see. Structure C. You have 2 options. Why 2 options? Then this chromosome, you see, it's 1. So you draw that chromosome with the centromere there. While you can draw this one also, draw the same thing with the centromere there. Are you seeing? But now this one is shaded. This one is not shaded. Why? Because it is colorless. See? Nothing shaded. This one is shaded. Therefore, you shade it up. I see? When you shade it up, what next? So it's gonna be like this. But now, where am I going to get the marks? Remember, they're saying final stage. Final stage has a nucleus. So this chromosome must be in the nucleus. Don't forget that at the final stage, you have the centrioles. You also put the centrioles in there. Centrioles are outside the nucleus. Then after that, please give a title and then you label cell membrane, nuclear membrane. Now, okay, you can talk about centromere, and then now this is not a chromatid. This is a chromosome. The chromosome I talked about, that these are called unreplicated chromosomes. These are unreplicated chromosomes. Yes. So I think you understand it very well. I think you really understand it very well. No, I think you got it right now. Ne? If you have not got it right, please let me know. Let me know. All right. Now, let's go to another sub-topic. Hey, there is one which stresses you. How many chromosomes will be there at the beginning of this cell? How many chromosomes are here? Oh, how many chromosomes are in this cell? So now, people always confuse. Please, if they don't mention this is a human cell, don't think about 46 chromosomes. Unless they have said this is a human cell. If they have not said this is a human cell, no, you are not supposed to draw both. You only have to draw one because they say draw a diagram. Label the diagram, not diagrams. Ne? Someone is asking, should we draw two? No, we only draw one. Yes. Or follow the instruction. What is it saying? But in most cases, they want one. But when you're marking, we draw the two and then we see which one to give you. Ne? Basically, depending on which one you have drawn on the memorandum, the answer is there, then we will do that. Yes. All right. Now, and when you are marking, we don't just mark. We just have to mark the title. We mark the title. We mark the shading. The shading is correct, maybe. Then we mark maybe 2 labels. Or the correct diagram, 1 label, and then the correct diagram, then here. All right. They're saying that how many chromosomes are found here in this cell? Someone is going to say 23. I ask you a question. 23 from where? This is not 23. I see 2 chromosomes. Why do you say 23? So it's going to be 2 chromosomes. People are always talking about 23, 23. Why? How many chromosomes would there be at the end of this cell in telophase 2? There will be 2 because this is meiosis 2. The meiosis 2 maintains the number of chromosomes. Another question. Sorry. Then they're asking you, how many chromosomes were there? How many chromosomes were there in the same phase of meiosis 1? There were 4 because meiosis 1 halves the chromosome number. So if meiosis 1 is 5, then meiosis 2 is gonna be. If meiosis 1 is 10, meiosis 1 is meiosis. If meiosis 1 is 10, meiosis 1 is gonna be, meiosis 2 is gonna be 5. So there is half the chromosome number. So please, you only if they say this is a human cell. Sometimes they ask you that, how many chromosomes will there be present in the same phase as a human cell? Now here, because it's meiosis 2, then you talk about 23. But if they don't talk about human, please and please, I beg you, don't talk about 23. All right. Then they are saying that, lastly, about meiosis, before I go to genetics. Ne? I show you the 6 marks which are going to come from genetics, and then I show you the simple questions, the way we are trying to define them. Share the video to others so that they don't miss out. Don't forget to subscribe, like, and also drop the question which you really want me to sort out. All right. Question. What is the importance of meiosis? What is meiosis? What is the importance of meiosis? The reason why we have meiosis is to have half the chromosome number. So that this doubling effect, because you remember sperm, let me show you. Sperm. The sperm, we'll call it sperm, ne? All right. The sperm, what happens? This technology, Ibo. Okay. I hope it's right now. The sperm will bring 23. And then the ovum will also bring 23. So when they fuse during my fertilization, you form 46. Yes? So meiosis must divide them again so that they form 23, 23. So that's why we are saying that it reduces the chromosome number, or it halves the chromosome number, or it solves the problem of the doubling effect of fertilization. Number one, that is it. Number two, the major importance of meiosis is to produce gametes. Produce gametes. Gametes, these are the sperms and ova or ovum. Number three, where does meiosis take place? Where does meiosis take place? Meiosis takes place in testes, in human, and then in ovary, that is female. Anthers in plants, that is male, and the ovary, that is female for the case of plants. You have to know those things. I think I've exhausted the questions of meiosis, unless someone is saying that load shedding is coming. Oh, sorry. And then okay, can I make it downloadable? I cannot make it downloadable until it is I'm offline. The moment I'm done, I'm completely done, then to be able to this one is asking me that how do I know the number I must count? The number is shown there. If look at this, these are 1, 2, 3. Let me check here. Okay, let me look for another question here. You will see the numbers. Sorry, when I'm looking for the question. Okay, that one. This one we are done with it. What about this? Let me check here. How do I know the number? How do I know the number? Oh, in this case we don't have, but this is the crossing over. This is the thing I was talking about, the process of crossing over. Centromere, chromosome, chiasma. This is a bivalent still. It's also a bivalent still. Also, this is a bivalent. But depends. You see now the crossing over here, there are two. So when they are drawing, for example, you have to draw them. In this case, you draw them up. You draw them up here. You draw them 2 times. So I think I've exhausted. But what you see, if the cell is showing you, chromosome 1, chromosome 2, chromosome 3 like this, and then they ask you how many chromosomes are there. These are the number of cells, number of chromosomes, 1, 2, 3. So tell us 3. How many will be there in meiosis 1 of the same phase? There will be, because meiosis 2 is single strand, chromosomes are 1, 1. Therefore, it's gonna be 6 in meiosis 2. Unless they say that how many will be there in the same phase of meiosis, but for human. I say that if they don't talk about human, forget about. You see now? Here is the question. Oh, where in the human female body would this type of cell division? This is where does meiosis take place in female? I've answered this, and I say that ovary. Give a letter name of the structure that attached to the spindle fiber, which is C, and then this case is the centromere. How many chromosomes will be found in each letter cell at the end of meiosis? Now come back here and see. The diagram represents the chromosome in the cell that is undergoing normal cell division. You see, it's undergoing normal cell division. So now they're asking, how many chromosomes will there be found in each daughter cell at the end of this cell division? How many chromosomes? How many chromosomes are you seeing here? This is chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5, chromosome 6. Because they are still under meiosis 1, so at the end of this cell division, it will be half the chromosome number, whereby 1, 2, 3, 4, 5, 6. So it means that the answer is going to be 3. I think Chantel, I think I've answered your answer correctly. Yes. Please share the video so that others can also benefit. Sometimes I do these lives And then after that, they look at it and they say, "You, I didn't see this live." So share it so that even other people can benefit from this live. All right. Let's go to our favorite topic called genetics. Called what? Genetics. Share the video so that everyone can get all the marks in genetics. Genetics. I'm not explaining too much of this, but I'm explaining only what you expect in exam. I'm not explaining the basics. 6-mark questions. 6-mark questions. 6-mark question. When you talk about 6-mark questions, yes, in genetics, what are you supposed to look at? Number 1, genetic cross. In this genetic cross, you need to know, when do I say sex-linked and when do I say non-sex-linked? Yes, sex-linked and non-sexy-linked. I think load shading is not there. I think so. On my side, ne? I think it's not there. Ne? I think they have changed the schedule. All right. Non-sexy linked. Sexy linked. Ne? All right. Number 3 is pedigree. You have to know the pedigree, but still here, you have to know whether it's sex or none. Another thing, disaster, blood group. And then lastly, cloning. Here we are going to talk about biotech. All these questions are 6 marks. 6 marks. I'm not saying this is what I'm going to talk about only. These are questions which come with the 6 marks. With the 6 marks. So if you mess them up, you lose. As I said that each section you're going to have one which has 5 marks, 6 marks, 7 marks. Latest. Oh, the worst is 6 marks. This, please don't lose them. Don't lose these marks. Distinction. Call your name Mr. and Mrs. Distinction. Tomorrow we have to crash the paper. All right, let's continue. Genetic cross. But before I start genetic cross, I've not talked about terminologies. I've not talked about them. But I want to know this. What is a locus? What is a locus? Or a position of a gene along a chromosome is called locus. Now, when do I use sex-linked and when don't I use sex-linked? I'm trying to go through this. After that, I go back. I'll show you. When you're doing the genetic cross, guys, even if you don't know anything, just write P1 space F1. We shall come and give you a tick. So you can't go to the paper without having this. So you have gone to the paper when you know. When you say genetic cross, before even thinking P1 F1, you get a tick. Okay, this is a phenotype. Phenotype is always given in the question. They will say that blue eyes, married, black eyes. You see, you come here and say blue eyes crossed with black eyes. That's a mark. Another mark you have got. After that, you say genotype. This is the only disaster which is there. We're going to look at it. And then now, what about here? Now we say phenotype. We are done. Genotype, we are done. Then now you go to meiosis. Meiosis will only have meiosis if you have the gametes. For example, if I have T, Okay, sorry. I think some people are not seeing there. Let me just draw it here. They are saying that, okay, P1, F1. I'm going to show you when do you write P2, ne? Because sometimes, yeah, but you saying P1, F1, mark. They say phenotype. Yeah, phenotype is always given in the question. As I told you that they can say big head crossed with small head, and then they produce a baby who is a big head. You see, I don't know. Are you a big head or a small head? I don't know. All right. Then they are saying that big head crossed with small head is given in the paper. So you don't need to stress. So you get a mark for this, you get a mark for this. Now after that you go to genotype. This is the only disaster I've talked about. We're going to look at it. You have to identify it. I'll show you. For example, T, small t, T, small t. Now you go to meiosis. Yes. They're saying meiosis. They're saying T, small t, T, small t. Which are these are gametes. You see? Now, gametes, in most cases, we love it when you circle them. You know why? Because sometimes people try to bring this close together. And once you bring them together like this, we cross. We don't give you a tick. So make sure that I prefer, always I tell my students, since you are my student now, circle them. Circle them. The moment you circle them, at least we shall know that, we shall know that nothing, even if they are too close, since you have circled them, still we will give you a mark. Now, then after that, you talk about fertilization, where you have the crosses. Now, you mentioning meiosis and fertilization is another mark. In most cases, this question is 6 marks. So you have got 1 mark, 2 marks, 3 marks. So you have got 50 out of how much? Out of 6. 50% you have got it. Before whether stress have started or not, whether you got these things correct or wrong, as long as you have that, you have the 3 marks. All right. Now, you continue. You cross this. Okay. I'm going to show you another way how you lose marks, né? And how to benefit get marks. This T with the T, and then this with this, T with the T, this with this, T with the T, then this with this, small t with the small t. Now, these are offsprings. Listen. In the question, sometimes we put what you call compulsory mark. We put what you call compulsory mark. When you're talking about compulsory mark, someone is asking me if I'm going to do cloning. I'm coming there to cloning. Compulsory mark, for example, if this is a compulsory mark, even if you get 1, 2, 3, so it's 1, 2, 3, 4, 5, 6. And if this question is 5 marks, and they say this is a compulsory mark, even if you say 1, 2, 3, 4, 5, 6, you got 6, but you lost the compulsory mark. We won't give you 6, no, we'll give you 4. We'll give you 4. But if you write this, the 3, the other 3, they are obvious whether the compulsory mark is there, whether the compulsory mark is not there. Those 3 are secured. You go to the paper when you have secured them. Then the other ones, the compulsory mark depends on the question they have asked you. In most cases, they ask you, show this. Then you do what? You show. That showing will lead to the compulsory mark. I hope I'm clear on that. Then they are saying, I'm going to fire you a few questions here. So the crossing, all of them, they do the same. Oh, I have not spoken about determination of sex. Sex. Don't forget to stay virgin. Determination of sex. Determination. Someone is asking. All right. Sex determination. Someone is talking about meiosis. Meiosis, we finished it. Sex determination. Okay. But you'll be able to watch it when the live is done. All right. We are saying that question can come, and this always kills the student. How many genes control blood group? How many genes control blood group? Let me know in the comments. Just now. Just type how many genes control blood group. I'm waiting before I continue. How many genes control blood group? Let me see. Who is the first one to answer? How many genes control blood group? I'm waiting. Define. Mutations. Yes, I'll talk about the mutations. How many genes control blood group? Yes, I'll cover every topic. Don't worry. I'll cover every topic. How many genes? Guys, type. I want to see the answer. How many genes control blood group? I'm not seeing the answer. 4. Someone saying 4. Thank you. How many genes control blood group? I'm still waiting for the answer. Type. Another one saying, I think 4. All right, thank you. How many genes control blood group? Tell me, Chantel. Yeah, okay, I think 4. Thank you. I want to know. How many genes control blood group? 3. How many genes? Okay, guys. Thank you very much. That's what I wanted to know. Yeah. Now, since we are looking for the distinction, they are not 4. It's only one gene. The gene, it is one gene polyallele. One gene. Which gene is that? Blood type. Blood group is only one gene. Are we together? How many alleles control blood group? There are 3. One gene. Remember, which gene is that? Let me show you here. Yeah, after I said one, after giving. Okay, okay, blood group. Blood group, the gene is called, it's like saying height, you're either tall or short. Is one gene which is height. You see? Height. You're either tall, height, you're either tall or short. You understand? Since it's one gene of height, but 2 alleles of tall and short. So blood group, the gene is blood group or type. Yes. How many alleles? There are 3. So say genes for blood group is 1. Alleles, there are 3. That is allele for A, allele for B, and allele for O. Sorry, allele for O. Like that. Are we together? Yes. Thank you for those people who tried to participate to give the answers. Since we are looking for the distinction, we can't go without knowing this. Gene, one gene. How many alleles? 3 alleles. 3 alleles. How many genotypes? Genotypes, there are 6. Yes? Genotypes, there are 6. You have this. Remember, A is either homozygous or heterozygous. That's 2. B is either homozygous or heterozygous. Then you have, that's 1, 2, 3, 4. Then you have A, B, and then you have that for O. That's it. So 3 genes, 1 gene, 3 alleles, 3 genotypes, and then phenotypes, there are 4, which is A, B, and O. So basically, that is it. Oh, AB, yes, AB is codominant. Yeah, O is the this is homozygous. Ne? This is the codominant or heterozygous. No, codominant. This is heterozygous, is homozygous, heterozygous, homozygous. So there are 6 of them. 1 gene, 3 alleles, 6 genotypes, 4 what? 4 phenotypes. Now, a tricky question can come. A man who is blood group A married a lady who is blood group B, and they produced a kid of blood group O. Show how this is possible. A man of blood group A married a woman of blood group B, and they produced a woman of blood group O. How is this possible? It means that once you find that question, for you to cross, it means that a man, because they are saying they produce a kid of blood group O. O is small I, small I. Before I go there, let me show you the mistake here you do. And we don't mark it. However much you love it, we don't mark it. Some people, they write blood group like this. A. What is this? This is not I. Don't write I like this. And you put a dot like that. I is like this. Don't write I like this. This is 1 in Roman figures. Check. You have sometimes, you have watches where it shows 1, 2, 3. Yes, they are like that. So this is not I. This is 1, this is 2, this is 3 in Roman numerals or Roman figures. So don't write A like this. We won't give you a mark. However much some computers, yes, they do. You are not a computer. Write the way
use
correctly.
My distinction
1
material. Yes. Those people
yes.
who use my distinction
so
material,
make
I
sure
know
that you
got
all these are simple things which might lead you to lose the distinction. For those people who have been using my book, Distinction Material, you know exactly what I spoke about it, and those people who have been watching the video. So it doesn't matter now. What matters here is for tomorrow's paper, for you to crush it. All right. Now, we say that how is a man of blood group A marrying a lady of blood group B, and they produce a kid of blood group O? 1. Know that blood group O, it has 2 eyes. Yes? Yes. If they have 2 eyes, how about this? Remember, A can be homozygous or it can be heterozygous. B can be homozygous and heterozygous. Now, if I have O, which is II, yes, it means that I used this and this. Because if I make this and this, there is no way I'm going to produce these 2. Even if I say this and this, there is no way. I'm going to only have 1 I, small I. Therefore, it's going to be heterozygous blood group A. So when you talk about the genotype, is going to be the phenotype. So I'm going to say P1, F1. Genotype, phenotype, phenotype. I'm going to say A crossed with B. And then I say genotype, I'm going to say A heterozygous crossing with B heterozygous. Then meiosis will take place. Some people, they then say A, I, B. This is B, ne? I. And then I circle them. Please don't put a cross here. Please don't. You'll get it 0. You'll get 0 there. Please don't put Don't put a cross there. Ne? Yes. Someone is asking, why didn't you find the channel at the beginning? That's the answer for God. All right. Now, let's continue. Let's continue, guys. But you can tell your grade 11 or your sisters, your brothers, subscribe the channel. Look for M-Side. Yeah. You always find the content there. All right. Now, look. Now, this is gametes. Gametes. So, if these are gametes, now what happens here? You can use a Punnett square method, or you can just cross. Whatever you want, still it is fine. For example, let me use the Punnett square because I've been using the cross. So you draw your Punnett square here, and then you say, okay, you have A, you have I here. You have B, you have I here, small I. And then now you cross, you're going to say this is A, B. Ne? This is going to be B. And then this is going to be A. And then this is going to be O. You see? You see? These are offsprings. Offsprings. You see? Now, this one produced a person who is blood group O. That's how it was possible. It can even come in the 20, in the 7-mark question. That's why I showed you those crosses. Those crosses. So please, blood group should not be a problem. Which blood group is now I'm done with the long question of blood group. Few questions now. Which blood group is a universal donor and which blood group is a universal recipient? Blood group A, no, blood group AB receives from every blood group. While blood group O donates. So blood group O is a universal donor. Some people, when they are blood group O, yeah, they think their blood is strong, they feel good. No, it's worse. Why? Because if you lose blood, if you need blood, we have to look for only one person with blood group O. But if your blood group AB, you are lucky. Anybody who comes, you can donate that blood to you. So blood group AB becomes a universal recipient. It receives from everybody. While blood group O is a universal donor, only donates to anyone. Someone is asking me that why these crossings are not allowed. The crossings are not allowed here because they are not crossing. They're already gametes. After that, you can draw your crosses here. Ne? So the crossing here, to cross here, no, it's not allowed there. It's not allowed. Crossing here is fine. If you cross them, it's fine. You can either use the Punnett square or you can just do the crossing. All right. Let's go to. We talked about the blood group. I'm coming back with the cloning, cloning. Or I'll give you one tip of an example of. Okay, let me give you the question because I just want to go just for 2 or 3 minutes. Yes, 3 minutes. Let me give you just one question for you to practice. Practice for me. Is 6 marks, ne? Yes, this one. It's a simple question. There is something behind it. Most especially on this question. I need to see so that you don't lose that mark. Please do that for me. Ne? Do that for me. Then after that, I'm gonna do it again for you. What's the time? Is 39. I'm giving you 4 minutes. 4 minutes to finish. Ne? I will be back. I will be back. I'm saying I will be back. I hope you are done. Hope you are done. Hope you are done. Okay. Oh, come on saying, yeah, it's done. All right. Anyway, ne? All right. Okay. That's nice. Okay, now let's go through it. They are saying, diagram below, okay, controlled by R and R. Check here. The first point is R and W. These are capital letters. The moment they are capital letters, it means that both of them are dominant. All right. Now they're saying red cross with white produce pink. You see, they have 3 phenotypes. The moment you have 3 phenotypes, it means that that is incomplete. Dominancy. It is incomplete dominancy. A. The question is here. State the type of dominancy shown in the Snapdragon plant. Here is the incomplete dominancy. Give a reason for your answer because a third phenotype is given or an intermediate is given. You see, a third phenotype is given or an intermediate is given. Now, if it is complete, if it was complete, ne? I would have been with red and white. I either produce red or white. Which one is dominant? Depending on which one is dominant. And then if I have codominance, red and white, I would have produced a mixture of red and white. But because this one is pink, a third phenotype, therefore, or an intermediate, therefore it is incomplete dominancy. They're saying Gardener crossed 2 pink flowered snapdragon plants using the genetic cross show the ratio of expressed phenotypes in the offsprings. Now, remember I told you that the first thing you need to do is to write P1 F1. But in this case, they're saying they crossed 2 pink. Pink is found on the second generation. Therefore, it won't be P1 F1. It will be P2 F2. In most cases, students lose that mark. Ne? If it is the second generation, then you say P2 F2. If the first generation, then you say P1 F1. Ne? Yes. So now, there you talk about phenotype, which is pink. I told you that this is obtained from the question. Check. They are saying, what are they saying? Check what are they saying. They're saying genetic cross showed a gardener crossed 2 pink flowered snapdragon plants is always obtained from there. Are you seeing? Then now, so it means that I've got a tick here. I've got a tick there, and also I've got a tick there. So now 2 marks. Now they're saying that pink, remember they say that white is capital W and red is capital W, capital R, white is capital W. Therefore pink is going to be capital W, capital R, capital W, whatever, capital W, capital R, or capital R, capital W. It's the same thing. So it's going to be capital R, capital W crossing with capital R, capital W. But this is the genotype. See, now you get another tick. Then they're saying that meiosis will take place, and then you have capital R, capital W, capital R, capital W. I said don't leave these so-called gametes to be as if they're at the beach. No beach, ne? At the beach, ne? You live naked, ne? You enjoy yourself. Ne? Don't leave them naked. Try to dress them up. Try to dress them what? try to dress them up at the beach. So try to dress them up so that they are not there at the waters there when they are enjoying their life. Try to dress them up so that you separate them. Even if they are too close to one another, you won't lose a mark for that. You won't. Then after that, you do the crossing, or you do a Punnett square. So cross with R, capital R, capital R, capital W, capital R, capital W. Because I started with R, I will start with R, capital W, and then capital W, whereby these are offsprings. Those are offsprings. Now, if those are offsprings, what is it? R, this is red, this is pink, and then this is white. Meaning that I will have one red, 1, 2 pink, and then 1 white. There is a mistake where the student do, whereby they ask you the ratio. In this case, you write 1 to 2 to 1. Some people, when they ask them the ratio, they write expression, and sometimes when they ask you the expression, they write the percentage. So when they ask you the ratio, please write the ratio. If they ask you how many were red, or what is the percentage of individual plants which were red, then you have to say it's 1 out of what is the total. The total is 4 times 100. Then you give us the answer. You give us the answer as 25%. Please don't forget that. All right. Let's continue. And we say our question is, I think I've exhausted that. Pedigree, let's just look at one question here which involves the pedigree, and then we can go to our there is one question here for blood group. There's one question here for blood group, but I think we exhausted the blood group. But you see that blood group also has 5 marks. Explain why paternity of the boy could not be established using the blood group. Why is paternity test is not able to be established, like to conclude that this is the father of the individual? It's because many people have the same type of blood group. So you can't say that, as I showed you, that is blood group A married blood group B, and then they produce blood group O, still they produce blood group AB, still they produce blood group B. So whom are you going to say that is not the father, or whom are you going to say that is the father? So it means that you end up losing the mark. All right. Why is it not conclusive? Because many people have the same type of blood group. But if they ask you, explain why paternity test for this situation, you will say that the mother is blood group O. You see, the mother is blood group O, while the boy is blood group B. So of which the boy can be B, heterozygous, or the boy can't be homozygous. Why? Because the mother is II, so gets 1 from the mother. So the genotype of the boy is heterozygous. Now this one can give the B, and also this one can give the B. So whom are you going to say is the father? Whom are you going to say is not the father? So that's why in this case, blood group can only help you to eliminate those possibilities which cannot happen. For example, a person of blood group, the father can't be blood group A because this person is blood group B. You see? So if it's blood group A, there is no way how the boy is going to have blood group B. So if there is another man who is blood group B, so blood group A, that one is wiped out. You understand? And then another man who is blood group O cannot be. So blood group A and blood group O, they are wiped out. Why? Because if it is small I, the boy is blood group B. So there is no way how this boy is gonna get a B from the father who is blood group O, yet the mother is already known is blood group O. You see? So that's the meaning that you can't use blood group to conclude the father of the individual. Because you are blood group B, you can't say that this is my child because this child of mine is blood group B. There are so many people who have the same type of blood group, and the possibilities of having these are many. I hope you understand me better. Now, how do you write this? It means that this one is only blood group A and blood group O are going to only be wiped out. So the blood group A and blood group O, they cannot be the father. So you get those 2 marks already. And then this blood group, the male A can give this allele for B to the boy. So it can be the father. Or this one also can also give one allele for B and gets another one from here. Also can be the what? Can be the father. That's why we cannot conclude using blood group who is the father of this boy. As I repeat that, blood group can only be used to eliminate the possibilities which cannot happen in a situation. Who is the biological father of the boy using the DNA profile? Now you have to check and see. You will see that it is this and this. You will see that is male what? Male 2. Give the reason is because the remaining bands of DNA profile match with the male. The remaining, they must match. If there is one which is not matching, then automatically it's not the father. Might be a relative, but might not be the what? The father. Remember, all the genes this boy or this baby is having is mother and the father. So if you take out all the mother's genes, because the mother is known, now the remaining bands of this boy must be those ones of the father. Let me show you what I'm trying to say. This mother, okay, this one is not for them. Yes, this one is corresponding. So this one is out. This one corresponds. This thing is now jamming. All right, corresponds. This one corresponds. Oh, this one corresponds. Yes. This one is not matching. So this one is not matching. This one is not matching. This one corresponds. Is out. This one is not matching. This one is not matching because this is not. This one is matching. So now, all of those ones which have X must match with the male who is the father. That's why I'm saying that the remaining bands of the boy, after those ones of mother have been removed, they must match with the father. Okay, this one, yes, it matches. You see, this one, yes, it matches. You see, this one is matching with this. This one is matching this. That's why we say that individual 2 is the father. Male 2 is the father of the boy, because the remaining bands of the boy have matched with the male. With male 2. All right. Now, let's go to just one example of a pedigree diagram. Pedigree diagram. Yeah, pedigree, where are you? Pedigree. And then I show you how you can use it to answer and also to score a mark. They are saying hemophilia is a that statement is enough. This statement, hemophilia is a sex-linked trait. Hemophilia is a sex-linked what? Trait. Meaning that if it is a sex-linked trait, what is it? It means what does it mean? It means that you're gonna use X and Y. Please, if they don't say is a sex-linked in exam, don't use X and Y. You're gonna get it wrong. Is a sex-linked disorder. Don't use X and Y if it is not mentioned in the question. So here it is mentioned sex-linked. Or sometimes they don't say sex-linked. They say that hemophilia, the gene is carried on X chromosome. Still that is sex-linked. If it's not mentioned, please don't even bother. Don't even waste your time. Don't even waste your time. to let me show you here. One of the deafness in human is carried on a single allele. The diagram below shows, yeah, done. They are not talking about sex-linked. However much they talk about Paul, and you know that Paul is a boy, Liz is a girl, Mary is a girl, John is a boy. However much you know that, please don't use X and Y. Please don't use X and Y. So if they don't mention is sex-linked, don't use X and Y. If they mention sex-linked, then you talk about X and Y. How many generations are here? There are 3. 1, 2, 3. They are saying that, and then number 2, it can be caused by, something can be caused by a recessive allele, or it can be caused by a dominant allele. Anyway, let's continue with this question. They are saying, state what is represented by a circle or a diagram. I wanted to tell you that sometimes we don't bring that a circle is a male, a box is a male, a circle is a female. Sometimes don't give you that key. You have to go to the exam when you know that. All right. Now they're saying, what are they saying? Okay. Now they are saying, this is a boy, this is a... How many individuals? Let me brainstorm you. This is affected with hemophilia. Don't say he's sick. Is sick of what? Sick of what? Malaria, ne? Sick of what? Sick of hemophilia, or is hemophilic. All right. How many kids are produced by P and Q? They are how many? It's 1, 2, 3, 4, 5. I know many people are gonna say that. It's not. They are not 5. 1, 2, 3, 4, 5. No. This one is an intruder. This one intruder. This one got married. This one got married. This. So there are only 3 kids. How many generations are here? 1, 2, 3. There are 3 generations. You understand? What is the phenotype, the genotype, or the phenotype? They can ask you what is the genotype or phenotype of P or Q, whatever they can ask. But if it is shaded, it means that that individual is affected. Now, they are saying that state what is represented by a circle. We have said that the circle means a male. State the number of general A. We have talked about them. There are 3 generations. 1, 2, 3. Offsprings P and Q. State the number of offspring P. The same thing is this, and this. This one and this ones, they are only and only got married to that. So they are intruders. State, they're saying that let us give the letters of only male who are having hemophilia. Letters of the female. Female is T and the female is U. Those are the only 2 people who are having hemophilia. Why? Because they are shaded. And then lastly, the genotype of R. The genotype of R. Now, how do we find the genotype of R? Look at the babies or look at the parents. First, look at the babies. It can help you. If you see that the baby is, one baby is hemophilic, then automatically the mother is gonna be heterozygous. So the mother is gonna, remember that here you have X and Y. Y doesn't carry anything. Y doesn't carry anything. So it's X and Y. So Y does not carry anything at all. All right. I think now, okay, so since this one is sick, it means that, and is a boy, so it's going to be like this. It's going to be, this is a recessive allele. So if it's a recessive allele, obviously this can't come from the father, because the father already gave the what? The Y. Therefore, this came from the mother. So it means that the mother, since is normal, but a carrier. So the mother is going to be, or R is going to be capital A, capital X, capital X, capital H, small h. And that becomes the answer for that. Yes. Let's continue now. Let's continue. So we are done with this. We are done with this. Sex determination and biotechnology. How do I explain sex determination? Sex determination. Sex determination It's like describing a karyotype. When asked you to describe sex determination, it's like describing a karyotype. But here, if they ask you the genetic cross, you have to do the cross. Ne? So, for example, if it's a cross, you tell us that P1, F1, everything, ne? Male crossing with a female, yeah, and then you tell us that male is X. You tell us the phenotype here, ne? And then you tell us the genotype. Male is X, Y, and then female is XX. Then you cross. Then you tell us meiosis here. You tell us gametes. They come out here. They come out, and then I'm gonna explain. I'm gonna show you how to write it if they say using a genetic cross, and then also show you how do you describe if they ask you to describe. Then these are gametes. These are gametes. Then they cross, then they cross, then this and this, cross Y, and then this and this, they cross like that. So this is the offsprings, meaning that you have 50% chance of having boys, and 50% chance of having girls. What about those who have girls, or boys? Yes, even if you have a boy here, the next time you have 50% of a boy again. Even if you have another boy, the next generation, you have the next reproductive cycle, you're gonna have still 50% chance of having a what? Having a boy. So it depends on how do you roll your dice, and how many chances do you have to have a boy. They are 50, but you are just lucky that you have what? Boys, or you are lucky to have what? To have girls. So in this case, we are saying that 50 boys, 50 girls. Yeah. If they ask you to describe, you tell us that individuals, humans are made up of 46 chromosomes. Ne? The 23rd pair is called gonosome. It's either X and X or X and Y. If X and X, if a male sperm with X fertilizes the female ovum, it will result in X and X. The individual will be a female. If a male sperm having Y fertilizes the ovum, remember ovum is always X, the individual will be X and Y, and therefore will be a male. There are 50% chance of having a male and 50% chance of having a female. That's how you can describe sexy linked. So let's finish up the cloning, and then we go to evolution. We finalize it with the evolution. All right. Biotech. Cloning. What is stem cells? What are stem cells? Stem cells, these are cells which have ability to divide to form other body structures. Imagine you are just a sperm. You are just a sperm, ne? Sperm fused with the ovum. You formed a zygote. The zygote, remember this paper one formed a ball of cells, ne? Which called the morula. You see, a morula is a stem cell. Because here, if I ask you, show me the eye, show me the legs, show me the chest, show me the, you can't show me. It's just a ball of cells. So these cells, they have ability to divide again, to form other body structures. You understand? Yeah, you form other body what? Structures. And then, yes, you become, yes, you have the eyes, you have the ears, you have everything. So we call them stem cells. Those are cells which have the ability to divide to form other body structures. All right. Give me 2 examples of stem cells. Umbilical adipose tissue, bone marrow. So you have to know those stem cells. Sometimes we bring this as an extract. Let me check and see. Let me check and see if I have any question regarding to stem cells. This is gene. Calculate the percentage of, okay. This is gene. Oh, I skipped something which is very important. That is the dihybrid cross. Yes. Let me leave it there so that I don't forget again. Dihybrid. I want to show you how do you identify the genotype. How do you identify the genotypes? Using the gametes, because that is a very problematic question. Okay. So in this case, I can't find the question there. All right. Now, let's go to cloning. I only want to discuss about cloning here. Ne? Yes? About the what? IA. Cloning. Yes? I have sheep A. I have sheep B. Ne? I get a cell of sheep A from the skin. Let me say somatic. Somatic. And then this one I get it from ovum. Ne? What I do, I remove the nucleus of this. I go with the nucleus. Ne? I throw away this shell. Well, here, I go with the shell, but I throw away the nucleus. I fuse them using electrical method, and then I form a cell which looks like a zygote. It looks like a zygote. Now, this zygote, I put it into another animal, a foster mother animal. Yes? Yes. And then what I do, I produce a lot of these animals, and these animals are identical. Question: why are all these animals identical? They're identical because when this forms a zygote, it forms a ball of cells. Now this ball of cells, now I divide these balls. Now I'll have small balls, and then the small balls I keep on going and then insert them in different other sheep. Ne? So it means that because I use the same genetic material, obviously these ones are going to be identical. It's because it's the same genetic material which was used. Now, a question: will these babies look like B or will look like A? They will look like B. Why? Because I use the nucleus of B. I didn't use the nucleus of A. Why did I use the nucleus, the shell or the casing of B? It's because I wanted this to look like a zygote, because what is outside is an ovum. You understand? It's like drinking alcohol in a cup. I won't say that you're drinking alcohol. I will say that you're drinking tea. So because the outside is a casing of the nucleus, therefore this will look like a zygote. And because this one is diploid, 2N, they can ask you how many chromosomes are found in this. Because it's a somatic cell. Somatic cells are always diploid. You see? Somatic cells are always diploid. Someone is asking me to do the part of evolution. I'm coming there. I'm finalizing genetics here. And then I'm coming to evolution. So because this one is what? Is a somatic cell. Therefore, automatically, this is going to be a diploid. And then, because it's brought in the a casing of the ovum, so it's going to look like a what? A zygote. And now I split them in two small pieces, and then I insert them in different sheep, and each sheep will produce identical Identical what? Identical shape. Yes, I think I'm done there. Sorry, I'm done there. All right. Now, lastly, on genetics is this question of the dihybrid cross. Identify what is Z. You just have to know. Cross this, and then you write what is Z. What is the genotype of the parents? Now, how do I identify the genotypes of the parent? Genotype of the parent. Look at the gametes. You'll find the genotypes from there. Start with F. For example, you write the F. If it is repeated, leave it. Is there any other small f? Yes, the small f is there. Then go to capital H. Yes. Is there any other small h? Yes, is there. Done. Look at them. If there was no, if all of them were capital H, ne? All of them were capital H, ne? For example, capital H, even here capital H, I would have said, do I have F? Yes. Do I have small f? Yes. Do I have capital H? Yes. Do I have small h? No. They are all capital H. Done. That's how you can identify. Simple, no stress. They're saying that number of the genotype that could result in offspring that is short finger, and short is dominant, ne? And then this one is recessive. Because it's recessive, I need to have with the small h. As long as I have capital H, is 1, is 2, is 1, is 2, yours, and then yeah, is 3. So there are 3. And then allele that for continuous hairline, allele, not genotype. So it's gonna be small h. The genotype of the child that is heterozygous recessive for both alleles. So is this one, which is small f. Yes. So this dihybrid cross, simple. You should not go to the exam when you don't know anything. Welcome to evolution. Now, evolution. Okay, evolution. Let's look at evolution by natural selection. This one does not have too many questions, which I'm gonna speak about, but it has many questions in human evolution. However, these days we are setting evolution by natural selection than human evolution. I think Mutasi, now we are on you now. Ne? Share the video so that now people can join for evolution. Ne? Because they find a lot of challenges with the evolution. Evolution, what is a hypothesis? What is a theory? You have to go when you know the difference. Ne? Yes. Hypothesis basically is a tentative statement which is subjected to be tested. It can be proven correct or wrong. And then a theory, basically what you need to look at is a statement which has been proven by facts. You understand? Situation which is being proven by facts. So it means that it must have evidences to support it. Then it becomes a what? It becomes a theory. Now, before I go there, I think this one is a little bit clear. The 6-mark questions or 7-mark questions, they are going to come from here. Number 1, theories of evolution. Number 2, that is Lamarckism or Darwinism. Number 2, speciation. Number 3, present day evolution. Now this difference between Lamarck and Darwin, yes, but present day evolution. You can't go to the paper without, because these are 6 marks. 6 marks each. Yeah, I told you, you go back, you come back and then you tell me what you found there. Ne? Yeah. All right, let's continue. Theories of evolution. Lamarck's theory, Darwin's theory. Lamarck, what you need to know is how to apply it, but I'm just going to talk about it. Ne? I'm not going to take too much time on it. I will spend the last few minutes on evolution, human evolution. But I'm gonna explain everything so that you don't lose anything. Lamarck's theory. State 2 laws of Lamarck's theory. It's law of use and disuse. That's number 1. Number 2 is law of inheritance of acquired characteristics. So it means that Lamarck has 2 laws. Number 1, law of use and disuse of a structure. Number 2, law of inheritance of acquired characteristics. Why is Lamarck's theory not accepted scientifically or by scientists in the world? Number 1, there is not enough evidence to support his theory. Number 2, organisms don't change because they want to change. Organisms change not because they want to change. You understand? Yes. For him, he says that if you want to be tall, you just become tall. If you want to be short, you just become short. No, organisms don't change because they want to change. Number 3, what is the difference? You have to know how to use how how Lamarck's theory is applied. For example, how did the big elephants with the big trunks came up with the big trunks? Using Lamarck's theory. So meaning that the more you use the structure, the more, at the beginning, all elephants had short trunks. Then you tell us that these elephants, they use their trunks frequently, maybe to defend themselves or to reach the trees, and then their trunks became bigger and longer. They passed their genes. Yes, they produced babies with big, long trunks. But if you are using natural selection, let me just use this as for natural selection. You have to start with, there is variation among the elephants. Those with long trunks and those with short trunks. Those with long trunks, they could survive. Yes? Why? Because they are able to defend themselves. They could reach the tall tree plants. While those with short trunks, they could not survive. They died in large numbers. You understand? This is called natural selection. This is called natural selection. I think. Now, those with big trunks, they pass their genes to the next generation. After many generations, all elephants had big trunks. Let me show you. Let me see if we can get any question here. But you can't go to the exam without knowing that. Using sex, show gender in human, how it is. I see. I explained this. I explained this. You see, 6 marks. 6 marks. Okay, when I find it, I'll try to show you that example. Speciation. Now, students confuse speciation with natural selection. What is speciation? Speciation is the formation of new species. Please don't confuse those two. Look, speciation is the formation of new species. What's a species? It's a group of organisms that can interbreed, meaning that they have similar characteristics. Group of organisms with same characteristics that can interbreed to produce a fertile offspring. What is a population? The population is the group of organisms of the same species that can interbreed to produce a fertile offspring. That's it. Now, speciation is the formation of new species. When they talk about speciation, they will bring about, for example, one area, and then this area is divided into 2. You understand? And then after that, if you try to mix them, they will form different organisms. They will form different organisms. They will form different organisms. So now, let me explain to you. You have a population of A. So you have to tell us that there was one this mark. There was one population. Yes. Now the population is divided into 2 by a geographical barrier. You have to tell us the geographical barrier. Let me show you, because I saw a question here. Yup. So what is the geographical barrier here? The geographical barrier, Describe speciation of Poto and lemurs. How did they become different species? There was one population of these organisms. There was one population of these animals. Did they talk about the ancestor? If they talked about the ancestor, then you have to speak about it. Scientists believe that Poto and lemur share a common ancestor. They didn't mention the ancestor. So you have to tell us that, don't say that there was one population of Poto and lemurs. They have not yet become Poto and lemurs. No. So they have not yet become those new species. You have to start with what they started with. There was one population of these animals which shared a common ancestor. Yes. And then what happened? The population was divided into two subpopulation by a geographical barrier. What is the geographical barrier here? Ocean. So you don't need to tell us by geographical barrier. Tell us by the ocean. If it is a river, tell us by the river. Unless the question is just open. And then now, you formed two subpopulations. That is east coast of Africa and Madagascar. And then what happens? There is no gene flow. There is no, this one can't come this side. Gene flow means that there is no sex between them. There is no gene flow between these populations. Whereby these two, they experience different environmental conditions. Are we together? So because they experience different environmental conditions, natural selection occurred differently. Oh, someone is asking me about punctuated equilibrium. I will come there. They formed, I'm going to explain it. Different environmental conditions, therefore natural selection occurred differently. Whereby they became genotypically and phenotypically different. And if you remove the river, if you remove the ocean, they cannot mix to interbreed. Therefore, they formed two new species of Poto and lemurs. They formed two new species of Poto and lemurs. You can only talk about these two new species. Don't talk about two species before you talk about genotypically and phenotypically different. If you talk about it, you're gonna lose marks. You're gonna lose marks. Even if someone is marking, it will pain, but nothing we can do. You will lose the marks. So what do? Is you only talk about different species after talking about genotypically and phenotypically different. If you don't do that, and you talk about you form two different species, that the population was divided into two different species by the ocean, you are gone. You have lost the marks. The paper has killed you. You didn't kill it. I think you understand what I'm saying. Yes. So now, I repeat. Let me repeat it for you. There was one population of a common ancestor. One population of common ancestor. The population was divided into two subpopulations of Eastern Coast Africa and Madagascar by what? By the ocean. There is no gene flow between the two subpopulations. You see, they are still populations, not yet new species. Remember, a population is a group of organisms of the same species. And then now, they experience different environmental conditions. Natural selection occurred independently or differently. And then now, they became genotypically and phenotypically different. If you remove the ocean and you let them mix, they cannot interbreed to produce a fertile offspring. Therefore, two new species of Poto and lemur have formed. They can bring when they are more than two or more than three. So in this case, you tell us three new species have formed. So don't stack on two new species. Don't stack on one new species. So two new species. So I'm coming for punctuated equilibrium. Charlotte, yes, you are saying hi. Are you still here? All right. Now, let's go to punctuated equilibrium before I go to present day evolution. Oh, let me just finish the present day evolution and then I'll talk about the punctuated equilibrium. If I forget, you remind me. Present day evolution
it deals with the bacteria, virus, black peppery moth. We are talking about present day evolution which is taking place, which we see. You understand? DDT on mosquitoes. Yes. So, you know, if they are bacteria, it means that you're going to use antibiotics. Use the same principle of natural selection. Please don't confuse natural selection with the speciation. So in this case, you will say that there was one population of bacteria. These bacteria are separated by the variation which is there. So now when you're explaining, you say that there was one population, sorry. You say that there was variation among these bacteria. Those which are resistant to antibiotics and those which are not resistant to antibiotics. Then he now tell us that the bacteria which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant to antibiotics, they die. Those which are this is called natural selection. Those which are resistant, they pass their genes to the next generation. So the next generation will have more bacteria which are resistant. And after many generations, the antibiotics will become ineffective to bacteria. Let me repeat. There is variation among the bacteria. Those you can use for mosquitoes about DDT. You can use for virus about the antiviral or ARVs. There was one. There is variation among the bacteria. Those which are resistant, you have to tell us the variation. If you don't tell us the variation, you lose the mark. There was variation among the bacteria. Those which are resistant and those which are not resistant. Those which are resistant and those which are not resistant to antibiotics. Those which are resistant to antibiotics, they survive on the application of the antibiotics. While those which are not resistant, they die in large number on application of antibiotics. Those which are resistant, they pass their genes to the next generation. After many generations, the bacteria are going to become ineffective to antibiotics. Simple. There is no way how you're going to skip. Oh, they're going to bring a lot of things which you don't know here. Never. What is the difference between artificial selection and natural selection? And what is the difference between Darwinism and the Lamarckism? Difference between artificial selection and natural selection. You know that artificial selection, what is basically on artificial selection is what is the selective force? First ask yourself, what is the selective force? Selective force in artificial selection is human. While in natural selection, the selective force is nature. Is the selective force. You understand? Yes. And then Lamarckism and Darwinism, the major difference is Lamarckism, organisms change because they want to change. But in Darwinism, organisms change due to the environment. Yes. Then number two, in Lamarckism, individual in the population change. Like if me, I want to be big, I become big. If I want to be small, I become small. Not the whole population. But in Darwinism, only the whole population change. Remember, when Corona came, if you want or not, they say that you have to put on a mask. Natural selection. You don't put on a mask, you die. Whether you want or not, you have to do so. You understand? Therefore, that is natural selection. Is it clear? So, in Lamarckism, individual in a population change. While the Darwinism, only the whole population will change. So you can also talk about other differences between the two. Let me talk about punctuated equilibrium. And then after that, I go to human evolution. Someone is asking me, how do I draw a pie chart. How do I draw a pie chart? I'll go to the last question. That is the last question, question 3. The pie chart. How do you draw this at the end of human evolution? So that just I show you how do I identify independent variable, dependent variable. How do I get it from the question? How do I know the control variable? How do I know the conditions which are supposed to be put in place before you start? All right, punctuated equilibrium. It is very simple. Gradualism versus punctuated equilibrium is gradualism versus punctuated equilibrium is saying like this. You have two scenario. Punctuate gradual. When you say gradual, it means that it's slow. The change is slow. While punctuated equilibrium, the change is fast. You have periods of no change and periods of change. The change is rapid. Periods of no change, periods of change, periods of no change, periods of change. This is punctuated. Punctuated. What this is it? Gradualism. Here you require a lot of time. A lot of time. Here it occurs in short periods. So punctuated equilibrium, you have periods of no change, period of change, period of change, period of change. And in most cases, the punctuated equilibrium is caused as a result of mutation. While this one is changed, is caused as a result of natural selection. Yes. Natural selection here, yes. While here, mutation. Why? Because you just wake up in the morning and then you have something different which you didn't have before. Mutation maybe occurred. So that is punctuated equilibrium, or the other side is gradualism. This marks the beginning of the end of evolution by natural selection. Let's kill the human evolution now. Human evolution. Human evolution, guys, is the most important part where students sometimes lose marks is a phylogenetic tree. I don't know why, but we'll talk about it. Someone is asking, do you do calculation for pie chart? Yes, you do the calculation for pie chart before anything else, because you can't do percentage in a pie chart because the circle is not in percentage. All right. Let me just see if I can find here a question. The first question in human evolution can be, to which family do humans belong? To which family do humans belong? They belong to family Hominidae. Hominidae. Differences between, let me tell you what are the short questions, guys, in human evolution. The short questions which you're gonna find, and then you have to give the answers. 6 marks. Number 1, it is out of Africa hypothesis. That's number 1. Number 2 is, so here you have to tell us the evidences. Evidences of that is fossil. You have to tell us the mitochondrion, DNA. Sometimes you call it genetics. Then you have to tell us cultural evidence, and then they can also talk about the differences between humans and apes. Yes. And then also they can talk about similarities between apes and humans. There is no 6 question mark they can bring here. Nothing. I repeat, nothing. Unless it's gonna come from heaven. Okay, now let me explain. There are some things which we changed a bit, and you have to know That's why if you check my book of distinction material, you'll find out that it's always updated each and every year. It's not static. It's always a version for that year. There will be a version for 2023. All right. Now let's continue. They're saying that, now look at this. Position of foramen magnum. They're saying which diagram represents a skull? There is a difference between a skull and a cranium. A skull is everything which is encloses the head. That's a skull. But a cranium is only the part which encloses the brain. Only the part which encloses the brain is what you call cranium. But a skull is everything which encloses the what? The brain. I think I'm clear there. Number 2. Number 2, they're saying that which diagram represents the skull of a bipedal? Now we have 3 factors which 3 factors which affect or which do the bipedalism, of which they can ask you the advantages of bipedalism. You can cipher, you can hold the organisms, you can hold the objects. Sexual dimorphism is exposed. You can see the sex organs. They can be shown because you are walking, yes. And then the body is lifted. You lift the body out of the ground, yes. So it means that less heat is gonna be reflected to your body. There is small surface area for heating of your body from the upper side. So those are some of the advantages of bipedalism. But now, what are the factors which contribute to bipedalism? There are 3. The first one. Why is this thing doesn't want me to write? I will write even if doesn't want me to write. Okay. Number 1 is shape. You look at the shape of the spine. Shape of the spine is what? Is shape. And then you look at the pelvis. Pelvis is short and wide. And then number 3, these are factors which affect bipedalism. Short and wide. Is wide, and short. So if you look at it, is wide and short. And then the third one is position of foramen magnum. It is in forward position. Yes. Now before I go to explain this, what is bipedalism? We changed this. You don't just say that bipedalism is walking on 2 limbs. Which limbs? Hind limb or forward limbs? You have to tell us walking on 2 hind limbs, or walking on 2 lower limbs, or walking on 2 feet. But we prefer walking on 2 hind limbs. If you're using limbs, it must have the word hind limbs. So, how is the shape of the spine able to for bipedalism? Shape here is S shape to allow flexibility. That's number 1. And then pelvic girdle is short and wider to support the upper weight. Position of foramen magnum is in forward position to allow the spine connect vertically. So basically, that's what you are supposed to tell us. Then they are saying that the next question is tabulated visible. They are saying visible. Don't bring about anything else. Visible difference between the upper jaw and the lower jaw of A and B. Check upper jaw, lower jaw. They're only looking for this. You see, in most cases now when we bring these, you have to know the differences. So in this case, you talk about the canines here are well developed. They are not well developed. There is one student who is saying that this one has canines, this one has no canines. No, the canines are there, but these are well developed, then this one are less developed. This one is rectangular shape. The jaw is rectangular shape. While this one, the jaw is what? Is C shape or U shape, C shape. Then if you look at here, you have spaces between the teeth. There are spaces between the teeth, large spaces between the teeth. Here, no space between the teeth or small space between the what? Between the teeth. Now, they can ask you, now I'm going to start firing you questions. How is the brain size related to intelligence? How is the brain size related to intelligence? The brain size related to intelligence, or how is the skull related to intelligence? How is the brain size related to intelligence? If the skull is big, it is going to accommodate more brain cells. The higher the brain cells, the higher the intelligence. If the brain size is big, you have more brain cells, and then this is linked to intelligence. That's why the previous organisms, for example, Ardipithecus, they had a small skull, therefore they had a small brain capacity, therefore the intelligence was also small compared to us. And when you are explaining this, when you're explaining the brain, there is something which we are looking for. If you don't mention it, you don't get the mark. The brain of human is not bigger than the brain of the elephant. It's only bigger than the brain of the elephant if you are comparing it with the body size. If we have a big brain compared to the body size, compared to the elephant. So when you are comparing, when you're talking about the brain or the cranium, you have to talk about compared to the body size. Please, when you talk about cranium, comparing them, then you compare it with the body size. I think I'm clear there. Who discovered Homo sapiens? You have to know who discovered it. I think I should drop. We have a short video which is talking about it. Who discovered the Homo sapiens? Tim White. Oh, who discovered Ardipithecus? Tim White. Tim White is Australopithecus. Sorry, is the Homo sapiens. Now, who discovered humans? Is Tim White. Yet is also Homo sapiens. He discovered himself. No, he discovered the oldest skull of Homo sapiens. Is it clear? Yes. So, you need to know, don't go to the paper without knowing 3 species discovered in South Africa. South Africa. At least 3. Don't go without knowing the 3 species which belong to Australopithecus africanus. Please don't. Which is Mrs. Place, Taum, Child, and then Little Foot. These are the 3 Australopithecus africanus discovered in Africa. What is the transitional fossil? A transitional fossil is that fossil which has the characteristics of the ancestor and the next generation, or the children. It means that it has not got extinct. It's still in transition. You understand? Which one of the following is a transitional species? In most cases, we look at Carabo. Australopithecus sediba is the transitional species between Australopithecus africanus and Homo habilis. So that is the only transitional species or fossil we talk about. However, even Homo naledi is also a transitional what? A transitional fossil or species. Now, question. Give the difference between the skull of humans and other primates. There is something which I want to talk about Prognanthus, because when you are marking that one, humans, Prognanthus means protruding jaws. So the other primates, they are Prognanthus. Humans are not Prognanthus. Don't say they are less prognanthous. No, if you have it in your book, yes, I know it's there, but we will not give you a tick. Humans are not prognanthous. Protruding, ne? Yes. While the other ones are prognanthous. Flat face, sloping face. Those ones are fine. Foramen magnum in forward position. We don't want anything else. Forward position. Foramen magnum in backward position. Don't tell us that foramen magnum is in the center. Center where? Foramen magnum is in forward position. Foramen magnum is in a backward position. Those are the things which changed, and then you needed to know this before you go for your paper. Because if you make a mistake and you mess them up, you're gonna lose a mark. Yet you knew the answer. It's just to rectify those few small mistakes. Lastly, describe, state the out of Africa hypothesis. State the out of Africa hypothesis. We say that modern humans, the word modern must be there. Modern humans originated from Africa. Modern humans originated from Africa and migrated the rest of the world. Originated from Africa and migrated. If you don't write the word modern, it's gone. The mark is gone. Give 3 evidences to support the out of Africa hypothesis. That is fossil evidence, mitochondrial DNA, and cultural evidence. Now, remember I said that this can come in this 6 mark. Yes? So how do I use cultural evidence? How do I use the mitochondrial DNA? How do I use fossil evidence to do this? Now, fossil evidence, you need to talk about few aspects. You have to talk about Adipithecus. You have to talk about Australopithecus. You have to talk about Homo habilis. You have to talk about Homo erectus. You have to talk about Homo sapiens. This is what you're supposed to talk about. Tell us that Adipithecus and then Australopithecus and Homo habilis only found in Africa. I think. Then when you reach here, you say that oldest, and also when you reach here, you say oldest fossil of Homo erectus and Homo sapiens were found in or discovered in Africa. I repeat. The fossil of Adipithecus, Australopithecus, and Homo habilis were only discovered in Africa. Only in Africa. Fossil of Homo erectus and Homo sapiens, the oldest fossils were discovered in Africa. That's how you're supposed to write the answer. Now, what about mitochondrial DNA? Here in mitochondrial DNA, we use the mutations which occurs in a mitochondria. Why do we use mitochondrial DNA? Because it has a small DNA, so it is easy to focus on it. But it's rare to find mutations there. So if the mutation occurs, we can use it to trace the lines of what? Descent. And if we trace them, it brings back to Africa. That's why it's called the mitochondrial Eve. And then cultural evidence, you talk about the oldest tools were discovered in Africa. And those are called the Odowan tools. Odowan tools. Yes. As I'm winding up with the today's class, so that you go and get your distinction, please, if it requires you to repeat this, to rewatch it, I'm telling you, I've tried to discuss each and everything which you're gonna find in the paper. You understand? If I've forgotten something, it might be only few, but you have 90%. I'm saying 90%, meaning that, what does it mean? It means that over 130 marks, you have the marks. All right. Let me conclude with the pedigree. Then I will just talk about few of the experimental question, and then lastly I'll go to the terminologies. Just only talk about the terminologies, and then we conclude for this class. I thank everybody who is attending, who is in my class, and I wish you the best. You must get a distinction. Let's try to finish up. Now, pedigree, not pedigree, phylogenetic tree. Before I go to phylogenetic tree, how do you write the scientific name? I think phylogenetic tree I need to use a diagram here. Let me just put out the diagram here, which I can use. Okay. All right. Let me use this. It's fine. Let me check if I have any phylogenetic tree here. This is the thing I was trying to explain before. Describe one other structure difference in bipedal and quadrupedal. You can talk about. Oh, okay. I gave the difference, ne? Short and wide, the factors which affect that. Okay. You have this now because I tried to explain it. Okay, let me use this. I'm gonna tell you all the questions which can come here. How many genera are indicated here? How many genera? Genera comes from the word genus. How many genera? Here is it's getting to 6 o'clock. To 6 o'clock. I don't know whether the electricity is going this side. Ne? Just in case that it has gone, at least you have everything. Ne? Yes. When you see me offline without saying bye bye, it means that it has gone because I didn't check anything. Describe the process of natural selection. Now you see this one. You just explain. You just explain. You just explain. Now I explained it. You don't use any example, ne? Yes. Okay. Now name. How many genera are indicated here? Genera is genus if it's one. Genera when there are many. You count the first name, but you don't repeat them. For example, 1 Australopithecus, Homo 2. Do I have any other? Nothing. So it means that I have only 2 genera there. To which family do humans belong? To which family do all? To name the family to which all the represented organisms belong. To which family do we belong? We belong to Hominidae. Yes. Then they are saying describe the cultural evidence used as a tool. You see, I explained it. Yeah, all these tools are other ones. Ne? So it means that how does it support the out of Africa hypothesis? It supports the theory of human evolution. Simple tools were discovered with the simple skulls, and then which means more advanced tools with the more or larger skulls, which means that they had the ability to modify these tools, and were found in the upper layers. How long did okay, now let me start. How long ago did the most recent common ancestor of this and this existed on Earth? Now, when they say how long ago, ne? For example, did the Homo ergaster existed? You come here. For example, if this is Homo ergaster, I come and draw where it appeared and where it got extinct. So it means that I will say 2 minus 1, which means, how long ago? No. How long ago did they appear? This is how they appeared. It means that it's going to be 2 million years. How long ago did they extinct? It's going to be here. So it's going to be 1 million years. How long did they exist? How long did they exist? That's a question. It means that I'm going to say where they appeared and where they got extinct. And then I said 2 minus 1, I get 1. But here it's going to be million years. It's not going to be million years ago. It's going to be million years. I think I'm clear there. Here you say million years ago. Here you say million years. Sometimes we always mark this unit. Then now they are saying which species first used the tools? When they ask you which species, if it is not indicated here, it is Homo habilis. That's why it's called Handeman. Which species first walked on 2 limbs? If they ask you species which first walked on 2 limbs, if it's not indicated here, it's gonna be Homo erectus. I repeat, Homo erectus. Then, which species are competed for resources? In this case, you'll find out that if this species and this species, they exist at the same time. If you draw a line like this, you see that this one and this one, they touch each other. It means that they exist at the same time. It means that they competed for resources. So maybe you'll say that Homo ergaster and Homo habilis. So it depends on the species which existed at the same time. But it must be having a line where they touch each other. Not exactly, but they must have a line where they touch each other. Then now, they are saying, what are they saying? They are saying, explain the possible reason why Homo ergaster was placed between Australopithecus afarensis and the Homo. It means that this is a transitional fossil. Yeah, it's a transitional. It's a transitional fossil, meaning that it has the ability, has the characteristics of this organism, and then it has also characteristics for this. All right, explain the possible reason why Homo ergaster. Where is Homo ergaster? Homo ergaster is here, and then Australopithecus afarensis is here, and then Neander is here. So it means that this is a transitional fossil. Yes, it has this one. Homo ergaster has some characteristics of this and also some characteristics of that. Because it didn't get extinct, therefore it's still going. Therefore it is a transitional fossil. How is the brain size compared to intelligence? I explained that. There's no questions they're gonna bring about this and then you can't answer. Yes. Lastly, how do I identify independent variable and dependent variable? Those who attended my paper 1 class, I think you know exactly. Just write ID. ID. And then after that, X, Y. I think. After that, you tell us what is ID. Independent variable, not identity card. Independent variable, independent variable, dependent variable on x-axis. So when you're drawing, independent is x-axis is here. So independent variable must be here. It must have units, please. Then this one, dependent variable, is this y-axis. So dependent variable is here. And then now, please put the title. It must have both the independent and the dependent. Put units here, please. Don't forget about that. And then now, because if you forget, you lose the marks. Scale, please. Don't say 2, 4, 7, 8, 10. No, the range is on the same. Here is 2, here is 3, here. You have to use the same scale. 2, 4, 6, 8, 10. You understand? If you miss them, you lose this and this. And then, now, the problem, now you have got to know that independent is X, dependent on Y. But how do I identify them from the table? In most cases, the first column, however much, how many columns you have, is ID still. The first column in most cases is independent. And the second column is dependent. Please don't miss up. Yes? The second and the third is always dependent. So now, how do I plot? How do I plot and how do I draw a pie chart here? If you are drawing a pie chart, please convert this into degrees. For example, if I have maybe 10, 20, 20-ish. Not technology. 20, 30. For example, this is Africa, Asia, maybe Europe. Then they say, so to put this on a pie chart, you have to say 10 plus 20 plus 30. So it gives me, this is 50, 60. 60. That's the total. So to do this, I'll have to say 10 out of 60 times 360, because this is the pie chart, pie chart, which is 360. So it give what you get here. You do that for 20, you do that for 30, then you plot. Protractor measure for this, then you change the protractor, start from here now, measure, then the remaining one will be for that. Then you put the title, then you shade whatever you shade, you put a key here, you get the answer, get the marks. So please don't mess it up. And then now, that is a pie chart. But now there is one for line graph. If it is on the same axis, and then the bar graph, it is if it is a bar graph, yes, for example, you have 20, 30, 40. Ne? Bar graph, you plot this and this, you get that and that. Then plot this and this. Then after that, give a space. Ne? Plot this and this, maybe 20. And then plot this and this, maybe 50. Then a space. Now, these bars must be the same. If this bar is not the same as this, you lose a mark. If this bar is not the size must be the same. These spaces must be the same. Ne? If those spaces are not the same, you lose a mark. That's how kids lose mark with these graphs. Then, if it is a line graph, it is a little bit different. Because a line graph, you have to first plot one by one. Line graph, look. Line graph, this and this. But in most cases, to get a line graph, you have to be with the figures, maybe 10, 30, 40. So that this and this, you plot, you get a tick there. This and this, you plot, you get a tick or a spot there. This and this, you plot, you join. Then if it is two graphs at the same time, now this and this now. So you first finish one column and another column, but independent must be kept. Now this and this, then you plot. This and this, then you plot. This and this, then you plot. Yes. Now, how do I know that my graph is wrong? We don't have a graph which runs like that. Some people have, it means that you swapped the axis. If you still have time, redraw it. We don't have a graph which runs like that. No, this is not met. Yes. So the graph must either be straight, going, if it's going up, it must form a wave, or it's going down, but this so-called parabola, what? No. We don't have them. Once you see that, you just know that you have swapped the axis. It means that what was X must be Y, what was Y must be what? Must be X. Please take note of that. Last,
Guys, thank you very much for joining our class. Let's talk about the memorandum of or suggested answers of this paper you wrote. I know that some of you smashed this paper, most especially if you were among the people who were following us. Yeah, I hope you managed to score high. And then don't be challenged by anything. For example, maybe my answer is a little bit different from your answer. Yes, it could be, but it could be different answers. I cannot discuss each and every answer which is most likely to be in this paper. So, guys, let's start without wasting time. Our paper, so that we are able to get it right. I thank everyone who shared, who were on this discussion of the prediction, the late revision, everything. Thank you very much. May God reward you, and we pray that you have a successful life. If things don't go the way you expect them to go, please still don't lose hope. If one door closes, obviously there will be another door opening for you. Just keep opening the doors. You will be able to get one which is right. All right. 1.1.1, we are saying that which female scientist took X-ray diffraction X-ray of DNA molecule and concluded that DNA is helical shape or helix shaped. In this case, all of them, these people, all of them were among the people who discovered the structure of DNA. They just talked about one name. James Watson is a man. Francis Crick, they were working together, is also a man. Rosalind Franklin is also a man. So the answer is, did I say Rosalind Franklin? Maurice Wilkin is a man, and then Rosalind Franklin is a lady. Is the lady who died of cancer. Yes, so the answer, the most correct answer in this case is supposed to be B. A cell with the 14 chromosomes undergo meiosis. In this case, they're saying a cell with 14 chromosomes. They didn't say that this is a homologous chromosome. They just a cell with 14 chromosomes. They didn't say that a cell with homologous chromosome. So it means that they're just talking about 14 chromosomes. If they're homologous, then automatically it means that there are 7 chromosomes. So a cell with 14 chromosomes undergo meiosis. What is the number of chromatid? They're asking for the chromatid. Remember, each chromosome is having 2 chromatids. So they're asking what is the number of chromatid in this cell at the beginning of meiosis. So they didn't say homologous, therefore it is 14 times 2, so that it gives you 28. I know that some people wrote you are 58, but they didn't say homologous. They said just chromosome. You understand? If they had said homologous, then you would have said 56, but they just said 14 chromosomes, which means that it's just a single chromosome. Yes. So the diploid of it, meaning that it is 77. Yes. All right. So the most correct answer becomes C, which makes it to be the most correct answer in this regard. They're saying that inheritance of blood group are two. Remember when we are talking about on the prediction about the blood group, I said that blood group is controlled by one gene. Yes, one gene, 3 alleles. Yes, 3 alleles, and then we say that four phenotypes, phenotypes, and 6 genotypes. 6 genotypes. One gene, that's gene, just a gene, and then gene for blood group. 3 alleles, allele for A, allele for B, and allele for O, and then 4 phenotypes, that is A, B, A, B, A, B, A, B, and O. And then 6 genotypes, we saw them. So we come here, we say that 2 phenotypes, that one goes out. 3 phenotypes, we said there are 4 phenotypes, that one goes out. 4 phenotypes, correct, and 3 alleles. You see 3 alleles, so this one becomes the most correct answer. 4 phenotypes and 2 alleles. No, it is 3 alleles. So the most correct answer in this regard becomes C. So you get 2 marks there. Yes, guys, don't panic because you're not getting right. Yeah, there are other questions where you're gonna get them right and still you stand to score a distinction. Yes. The following statement relates to cell division. Okay, crossing over, they're saying contributes to genetic variation in the population. Okay, they're saying that produce cells with the haploid number of chromosomes produce somatic cell. Diploid number chromosome cells with the diploid number, this is very rare unless it's fertilization. Somatic cells only, not really. So okay, well, let's see what they're saying in this regard. They're saying that which one of the following combination of statements refer to meiosis? Meiosis. Obviously, meiosis result in haploid, so not diploid. So this one, whenever there is 3, is off. So this is off. This is off. So it's off. So expect to have this and this. They're saying that produce somatic cells. Meiosis doesn't produce somatic cell. Meiosis produces gametes. So therefore, anything which has 4 is out. So automatically, we only remain with B. So B becomes the most correct answer. Then we go to 1.1.5. They're saying that some and beds are believed to have originated from common ancestor. The shaded part of the diagram below shows the distribution of these species. So we have these shaded parts. So the distribution of the species. So in this case, what are we looking at? The type of evidence for evolution. The type of evidence for evolution that is represented that is represented in the diagram is, in this case, we are seeing that different lives are there, so therefore it is bio, because the different geography of the world therefore becomes biogeography. So A becomes the most correct answer. They're saying that the plant with yellow which is recessive, round which is dominant, is crossed with the green which is capital letter which is dominant, with wrinkled which is recessive. So meaning that you have capital Y, small y, and then capital R, small r, capital R, small r. So in this case, what are they asking? They're asking that which one of the following are possible genotypes of the parents one. So which parents resulted in the formation of this? So definitely I will need to have one which has the capital letter and one which has the small letter. Yes, so that when they combine, they form this, and then this. So if you look at this one goes out, not really, because of yes, this one, it goes out also. Yes, this one goes out. So the most correct answer, because this one will come, bring it this one there, and then this one brings that there, then this one brings that